স্টার্ট করো
সম্পূর্ণ শেয়ারিং এটা রাইট হ্যাঁ হ্যাঁ স্টপ করে দাও এটা বলুন স্টপ হ্যাঁ হ্যাঁ
A very good morning to all. Am I audible?
Yes, yes, you are clearly
Yes,
audible.
ma'am.
A very good morning to all the delegates, speakers, and participants. I'm delighted to have you here once again on the second day of our international conference, eConsus 2021. At the very onset of today's session, please allow me to publicly express my gratitude to Dr. Amitabh Basu, Principal of our glorious college, the Chief Patron, and the Coordinating Committee for all their hard work in planning this conference. To commence this session, we have with us an esteemed erudite professor, Dr. Subir Chandra Dasgupta, as our first keynote speaker. I'm elated to introduce him with a deep sense of pride and humility. He has been my teacher and a role model since college days, a very dynamic personality, because of whom I developed a strong sense of affinity towards genetics. He is presently acting as a professor and head in postgraduate department of zoology, Maulana Azad College, Kolkata. He has obtained his master's degree in zoology in 1981 from Calcutta University and PhD in 1989 from School of Tropical Medicine. Beginning his career as lecturer in Darjeeling Government College in 1985, by dint of his dedicated work and passion for research, he steadily rose to become professor of zoology in 2010. During his tenure of work, he has published more than 45 research articles in peer-reviewed international and national journals, 3 books in his area of teaching, and more than 40 conference presentations. He has 1 international and 1 national patents. He has successfully undertaken research projects sponsored by national bodies like UGC, DST, DRDO, CSIR, and TRF. Professor Dasgupta is an active member of many scientific bodies, which include Indian Science Congress, Indian Pharmacological Society, Indian Biomedical Research Association. He is a fellow of Zoological Society, Kolkata. Being a government college professor, he is a nominated government member and representative in many academic bodies of this area of teaching. He is a member of many academic he is a member of many academic bodies. Till date, he has guided 12 PhD students. Dr. Dasgupta's researches mainly include identification and characterization of bioactive compounds from natural resources, animal venoms, toxins, and its therapeutic uses. I heartily welcome you, sir, to go on with your interesting presentation.
Okay, thank you. Am I audible?
Yes, sir, you are audible.
Okay, okay. Just 1 minute. I'm sharing my slides. Is it visible?
Yes, sir, it's visible.
Okay, okay, okay. So, very good morning to all of you. First of all, I express my sincere thanks, Principal Dr. Amitabh Basu, Asansol BB College. Yes, it is a very famous and very old college, for inviting me to speak something as a keynote speaker in the second international e-conference on revisiting strategies for sustainable development. So, and also the IQAC of the Asansol BB College, all the faculty members of this college, especially the zoology and the Zarka, who is my direct student at Maulana Azad College, and one also teacher is there, Shashwati. She is also my direct student from Maulana Azad College. So what I'm going to present today is my work with my mentor, Professor Gomes at Calcutta University. So I sincerely carry out a very small research at the department with my mentors, because without an university collaboration to carry out research sitting in a college is really very difficult. So my entire work with Professor Gomes creates a lot of interest among the students, also in the publication list, also from the funding agency, from the different national bodies. So the title of my today's lectures is the diversity of the vertebrate skin derived bioactive compounds and its therapeutic importance. So the vertebrate skin, especially here, I am going to present the three species skin. One is the amphibian skin, which is usually very common to all of you. That is a Bufo melanostictus. Nowadays, the name has been changed, Dattophrynus. And another is a snake-headed fish, the shoal fish, which is readily available in the common market, murals commonly known as. And the third one is the snake skin. So I just pick up this all three model because the skin is not just an mere the outside protection of the animal body, but it contains severe bioactive compounds. So this is my three major area of research. One is the Bufo, excluding parotid gland, because as a zoologist, all of us know the parotid gland is somehow toxic. So we took the skin excluding the parotid gland. And the Chana striatus, commonly Bengali name is shoal mach. So it is a snake-headed fish known as the murals. And the third one is a Naja Naja. So that is a snake skin. So that is the last work at Maulana Azad College. Already the students are awarded PhD. So that's a very interesting finding. I'll share with all of you this my findings. So the natural products from the plants and an animal origin have been used as a potential pharmaceutical target as their rich source of the bioactive compounds. So the skin containing a lot of important bioactive compounds. So more than 100 products in the clinical trial, mainly in the field of cancer biology and as an anti-insecticides. Several bioactive compounds have been isolated from the skin of vertebrates, especially the amphibia, snake-headed fish, and reptile. This is the first comparative analysis regarding the bioactive compound present in the vertebrate skin extract. Now, coming to the first group, that is amphibian. Amphibian skin is morphologically and biochemically, physiologically a complex organ which fulfill a wide range of function necessary for the amphibian survival. So parotid gland is a toxic, and during monsoon this gland activity is much enhanced for their prey act as a food to neutralize the predators, the insects what the amphibian took as a food. And that necessary for the amphibian survival, including respiration, water regulation, anti-predator, antimicrobial, lot of functions. The famous scientist Daly, if you search in the Google, so amphibian skin or amphibian bioactive compound, and the scientist name is Daly. The Daly has at least 150 patent, and all these are with the amphibian, right? So there are a lot of amphibians throughout the world, among which I think all of you have known through the Discovery Channel. The poison dart frog is a very dangerous in the amphibian kingdom. So the amphibian skin contains the various bioactive molecules, is peptide, protein, steroid, alkaloid, that possess the potent therapeutic activities like antibacterial, antifungal, antiprotozoal, anti-diabetic, anti-neoplastic, analgesic, and sleep-inducing property. I here just to mention one or two properties among the from the amphibian skin. One is anti-neoplastic, another is a sleep-inducing property. So research on the amphibian skin derived biomolecules can provide a potential clue towards a newer drug development to combat various pathophysiological conditions. So this is my experimental design with this Bufo melanostictus. So first of all, we have to carry out the what are the toxic dose, so determination of LD50. Then the effect of this tote skin extract on the sleeping time and the body temperature, because tote skin contain a very important bioactive compound which induces sleep in the animal model. That work has been carried out with the collaboration with a JNU, Jawaharlal University, because in the life science department they have a sleep research laboratory. Professor B.N. Mollik is there. So we have a strong collaboration with him. So the sleep inducing factor characterization has been detailed worked out from this skin of the amphibia. I'll show later on in my slide. Another is a hemorrhagic and hemolytic activity of the tote skin Pharmacological activity, the blood pressure, respiration, isolated smooth muscle, nerve muscle, and heart and auricle. Here I am like to say one thing. My PhD supervisor is actually from an MBBS, right? Professor Shitesh Lahiri. He was the director of the School of Tropical Medicine. So, and as because he is from the medical faculty, MBBS, and especially from the pharmacology department, which is a very important area of medical science. So, our research is almost oriented towards a therapeutic approach. And being a zoologist, I also know the different screening of the drug through the blood pressure, respiration, isolated, because the physiology students are well aware with this technique, not the zoology student. They have the chymograph, they have the system of recording the blood pressure, respirations, and smooth muscle contraction, nerve muscle contraction, etc., etc. So I just trained myself during my PhD days in that laboratory, because my laboratory is the department name as the Department of Pharmacology. I obtained my PhD from School of Tropical Medicine, Department of Pharmacology. And as because my supervisor is a medical person, so I have to know the details activity of that pharmacological activity. And the next one is an immunomodulatory and the anti-neoplastic activity. Now, these are some results of this Tote skin extract. The extraction procedure is very simple. We collect the tote from the physiology department, because in physiology department at that time, I'm talking about the year around 1991 to etc. In that time. So the tote has been extensively used in the physiological practical frog rectus muscle, etc. So we just collect that tote from their practical. We isolated the skin excluding parotid gland, put it in the methanol, and another reason just simply water. So we have a two extract. One is a methanolic extract, another is the aqueous extract. TSC for Tote skin extract was found to 400 milligram per kg in IV route. So by which we can determine what is the toxicity or the level of toxicity of that extract. And it has been found that the aqueous extract is rather more toxic, that is a 50 milligram per kg IV. So high respiratory rate and hypokinesia, convulsion, hind leg paralysis, protrusion of the eyeball. So these are all the phenotypic observation in a mice or rat model that actually gives us a clue what is the nature of the drug and the functions. Normal behavior resume after 4 to 5 years of observation. That means after inclusion of this drug to the animal model, that all this activity has been shown for, and duration of the stay is 4 to 5 hours. TSC 40 milligram per kg is a non-hemorrhagic compound, significantly increase the sleeping time. That is a very important observation by the percentage, that is 212%, and the rectal temperature caused the hemolysis. So increasing of the body temperature also induces the sleeping time. That experiment is a very easy. We, the students of zoology, actually do not perform that experiment, but if you have the permission from animal ethical clearance, because doing or performing this experiment, you need the rat model or mice model, and you need an animal ethical clearance, because this type of experiments need some crucial point. So what we have done, we isolated the extract from the toad skin, and that gives simply injection the intraperitoneal to rat and mice. This is our experimental protocol. In the control, we put the saline and standard the drug which induces the sleep. That is in our market, the alzolam is there, diazepam, paxam. So that also given to the intraperitoneal. If you compare the sleeping time of all these three agents, you find the mice of the rat which have the toad skin extract, the sleeping time enhances vigorously, right? So that is our simple experiment. From that, we carried out a series of experiment with this toad skin extract. So these are the pharmacological activity, role on the cat blood pressure. In cat blood pressure model, it induces initial hypotension followed by the rise of BP. The graph also shows there. Initial, these are all recorded in the chymograph. And atropine 3 milligram per kg antagonize the toad skin extract induced hypertension. So that graph also shows the doses is there. This is the role of rat respiration that produces respiratory apnea. So this straight line, this is a recording carried out with the rat respiration, but the straight line in between the graph, it is a apnea. That means temporary cessation of the respiration within 1 minute of the introduction of the TSA, but it ultimately returns. So temporary apnea is also observed with this extract. Other pharmacological activity that is isolated on the smooth muscle, isolated smooth muscle. There is a contraction with the guinea pig ileum, that is a smooth muscle, the contraction. That means it gives us a clue that the toad skin extract has a contractile property that will act in the smooth muscle. So role of toad skin extract on isolated heart. This is isolated heart preparation. Once again, I'd like to say that all this methodology is almost related with the pharmacological, mainly related with the medical science. But as because I work in this medical oriented laboratory, the pharmacology, so I have to know this all this detailed procedure, because that actually gives us a clue for the drug development. So on isolated toad and the guinea pig heart, toad skin extract decrease the heart rate and increase the amplitude of the contraction. Amplitude means the length of this heartbeat has been increased. On guinea pig auricle, this is heart, this is auricle, it causes complete blockade at a high dose and lead to a cardiac arrest. This is a cardiac arrest, that is a complete block in this auricular contractions. So toad skin is a very slow contractile element. Toad skin extract also isolated in nerve muscle preparation. Here the two model we have choose, that is one is chick biventral services preparation, another is a rat phrenic nerve diaphragm. So these are the two experimental protocol. This methodology is almost standardized for a pharmacologist. So here they also show the produced a complete blockade of the electrically induced twitch response in chick biventral services, but no effect on the rat phrenic nerve diaphragm. It is a nerve muscle preparation. The phrenic nerve is there and the diaphragm is a muscular dome. So the phrenic nerve and the diaphragm has a no effect with the toad skin extract. Now, toad skin extract have a strong immunomodulation effect because it increases the lymphocyte and the macrophage counted. It's macrophage migration inhibition assay. It is a dose-dependent inhibition takes place. This is a neutrophil chemotaxis assay. This is a standard casein we have used. This is in presence of casein. The neutrophil usually migrated, but it has been found that toad skin extract also induces the neutrophil migration. That gives us a clear idea that this TSA extract have a strong immunomodulation activity also. So this is an anti-neoplastic activity that we have worked out in two leukemic myeloid leukemia cell line. One is U937, another is K562 cell. This is a standard controlled drug arabinase, and this is a toad skin extract. You find this in the inductions with the toad skin extract that significantly decreases the activity of the U937, also the K562 cells. The effect on toad skin scales on the cell cycle phase is also significant. It inhibits the cell cycle G1S, G2 phase, and that paper have been already published in the Toxicon International Journal of Toxicology in 2006. So the toad skin also the neoplastic activity, again that apoptotic like activity has been worked out with the FACS analysis, flow cytometric analysis with the annexin 5 binding assay. Here you find this is a control slide. This is a panel A. This is a panel for U937 and this is for K562. So A is a control panel, B is a standard arabinase C, and this is a toad skin extract. You find the toad skin extract, the binding with the annexin 5 is maximum enhanced. Similar activity also showed with the K562 cell line. So now the second we have carried out the confocal laser scanning photomicrograph of this U937. As we all know, the apoptosis also takes place, and one of the hallmark feature of the apoptosis is a membrane blebbing. So you just noticed with this the staining after the staining of the red rhodamine B. So it has propydium iodide. So it has been found that the membrane has been already blebbed here. There's a two cell line is there. One is a electron microscopic picture, a confocal microscopic picture with a, and the scanning electron microscopic picture. So in this is a intact cell, this is a control cell, and the membrane blebbing actually takes place, and that cell line is again scanned with this electron microscopic observation. The similar observation is also found. Then the purification, then we have tried to purify the sleep inducing factor. That work is actually with a collaboration work with JNU. These students actually carried out this her PhD thesis, and along with my mentors, we have purified this compound, the purification of sleep inducing factor, with nomenclature read SIF, SIF. And interestingly, that SIF have recorded the EEG, EOG, and EMG. So all the detailing analysis has been carried out, because carrying out an EEG, EOG, and EMG in the rat model is not so easy. We are here in the pathological laboratory. The human patients are often being tested with EOG, EEG, but carrying out this EEG and EOG in a rat model and compare and study with that is not so easy, especially in the West Bengal. So naturally, we search for a good laboratory who is actually working on this sleep-inducing factor, and Professor Birendranath Mollik is there in for JNU. He is very kind enough to collaborate with us, and I personally visit in his lab at the JNU. Is a tremendous. We cannot dream about it. The sleep research laboratory, and all these experimental parameters have been carried out there, and we found a profound changes of this EEG, EOG, and EMG of by the SEF, that is a sleep-inducing factor. Then we carried out a detailed analysis of the brain biogenic amine, because as you know, the sleep is related with so many bioactive compounds, namely as serotonin, noradrenaline, GABA, MAO. There are a lot of compounds is when there. So the MAO activity, we found there is a severe changes have been takes place at the time progressing. So the effect of sleep on the brain MAO activity on the male albino mice, and here is a biogenic amount that causes a significant changes in the brain biogenic amine also. So the presence of the sleep-inducing factors in the amphibia has been established, and that sleep-inducing factor may causes some effect also in the human being, but because all the experiment have been carried out in the rat model. So we propose that a sleep-inducing factor is almost there, and our observation has been taken by the famous book in the amphibia, The Biology of Amphibia by the Dwellman and Trube. In that book, they have mentioned that yes, that the snake skin containing the bioactive components. Now, the second objective is a Chana stratus, that is commonly known as a shoal fish. The murrels, the snake-headed fish, occupy a very important position in the ecosystem and also the environment. The fish skin accumulates different kinds of the toxic compounds in different environmental conditions. The identity of some of these compounds and their functional status have been explored. The fish Chana stratus, previously it was named as Ophiocephalus. Later on, it has been changed by the Chana stratus. Locally known as a shoal fish is a striped snake-headed fish, commonly known as the murrels. Right, generally in West Bengal, the peoples remove or burn the skin of shoal fish before consumption. Bajare jokhon amra jai, so if you purchase this shoal fish, usually the shoal fish skin has been removed. It has been postulated that if you consume the shoal fish with their skin, that causes a hemolysis of the RBC. So that information we have gathered from a traditional book of Shantals of India, that is present in the Asiatic Society. That is a treasure house of the very old conception in Indian scenario. So the cause of the such removal or the burning of the skin is not actually known, and no scientific information is available. So our attempt has been first attempt to prove yes, the snake skin the fish skin containing some terrible bioactive compound which is not at all good for the human health. So the general practice of the skin removal is carried from the one generation to another, and on the belief and that perhaps the skin has some toxic effect. To establish the shoalfish skin extract toxicity, this present study was carried out. So the fish skin accumulate different types of components. So these are extraction procedure of the skin. We just remove the skin from the live fish purchased from the local market. So just carried out the TLC. This CSC, that means Chana strata skin extract. So after elution, proper elution, we have isolated a factor which is a terrible cardiotoxic factor. Again, to prove an bioactive compound having cardiotoxicity, you have to carry out this isolated heart, isolated auricle, guinea pig auricle. So series of experimental protocol already standardized in the pharmacological method. So we have to carry out series of experiment, and that actually proves us or gives us an indication that the shoalfish extract, skin extract, contained a bioactive compound that is cardiotoxic in nature. So we isolated the cardiotoxic factor, and that cardiotoxic factor has detailed characterized and characterized. So this is a hemolysis. You look, the percentage of the hemoglobin is terrific for the 12 hours. The control is 13. But after 12 hours, that hemoglobin comes to 6.50. So usually our laboratory suggests consume shoalfish. That is very good, but do not consume the skin of the shoalfish because it causes the hemolysis. So all the data is drastically reduced what you observed in that level. So RBC, WBC, the total decrease of this complete hemogram of this nature. This is a cardiotoxic factor, shoalfish skin extract. This is a normal amplitude of the isolated guinea pig heart. This is a guinea pig heart, and this is a time in minute when you. This is the application of this fish skin, and after certain time you find that total heartbeat has been stopped. So it is a terrible cardiotoxic factor. It is isolated guinea pig auricle. Again, the same thing. Auricle beat is also stopped. So the cardiotoxic factor is present. And now we go for a detailed analysis of the biochemical of the cardiotoxic factor. That experimental work, being a zoologist, very honest speaking, I do not have any idea regarding this chemical structure analysis of this the compound. So what we have tried, so we make a collaboration with the Indian Institute of Chemical Biology, Indian Association of Cultivation Science. In our team, we have a good biochemist is there, Dr. Debashish Bhattacharya, structural biochemist. Now he has retired and guest faculty at my college. So with his collaboration, we have carried out a series of detailed experiment to prove the cardiotoxic factor of this SFAC. So IR spectra analysis, this is NMR analysis, and that compound, and this is a FAP MS spectrum analysis. All these analysis actually infers that all spectral studies revealed that the cardiotoxic factor was an aromatic alkaloid compound with a NH3 carbon double alkaloid and hydroxyl group functional group, and having molecular weight is 413 kilodaltons. So next come to the bioactivity of the shade snake skins, and that is also interesting area, because as a zoology student, if I ask someone what is the function of the shed skin, why the snake shed their skin, common answer is that simply for the growth and or simply for the excretion. But our experimental result actually do not tally with the conception of the zoological standpoint. We find and we prove that this shed skin containing terrible bioactive compound, and even it is a toxicity is there. So I personally collect this shed skin under the permission from the Department of Forest, Government of West Bengal, because as you know, the collection of the shed skin from the wild is not permitted, because that snake skin is being imported to other country for different purpose. So carrying out with this research with the snake skin, I need a permission from the Wildlife Department. They permit us to collect 3 or 4 snakes shed skin. So for my experimental analysis, I choose the naja. So the shed skin is considered is considered as a biological waste product among the zoology student. The traditional medicinal system of different parts of the globe use this shed skin for various ailments without any scientific validations. In Indian Ayurvedic medicine, the cobra shed skin ash is used to coal in women and pregnancy disorder. That is our clue. I have already mentioned we just find or obtained this clue that shed snake skin containing is a terrible bioactive compound from the book Shantals of India, written by the Boding. That book is present in the Asiatic Society Library. From that book, we found that the snake skin, shed skin, is actually a treasure house, and they usually give that the extract to the Chantal woman related to pregnancy disorder. So used as a folk remedy, including Chinese and tribal medicine. With this information, this is our first attempt to establish the proper scientific validation and its medicinal importance. So I have already published a good review regarding this activity of the snake body parts and used as a medicine throughout the world. But in our India, it is to capture the snake and to kill the snake is a highly objectionable thing. But if you collect the fresh dead body of a snake, you are able to carry out so many research which have a strong therapeutic indication. So I published that paper in the Journal of Zoological Research. Snake blood is used in the rheumatoid arthritis. Snake meat has been used to snake skin disease, pain, intestinal hemorrhage. Fat has been used, has been anti-inflammatory, antimicrobial. All this has been used in the Chinese medicine. Snake bile is the appetite stimulant. Snake venom is a very common, all of you know, even in homeopathic medicine, snake venom is extensively used. That is Vipera 30 is one of the drug used in the homeopathic medicine. Naja 30 is a homeopathic medicine. So snake venom and Kalajar, fibrinolysis, even the latest study shows that the snake venom is a very much effective in the leishmania treatment. So that is an anti-leishmania activity also found in the Naja venom. And the snake skin, that is also used in carbuncle, psoriasis, glaucoma, and pregnancy related disorder. So with this aim, I have designed my experimental work with this. So the main activity, the bioactivity studies, as because in the book they have mentioned that is related with the pregnancy disorder. So my focus is to carry out the whether there are any reproductive toxin present in the snake skin. So the first activity is the virgin animal estrous cycle, hormone, cytokine, histopathology. And the second part is a purify and characterization of the active compound from the snake skin. And the bioactivity, that is the hormonal level, cytokine level with the active fraction. Also with the biomorphometry changes of the puff, because the pregnancy related disorder is also being focused with this Naja skin extract. So some part of the biomorphometrics of the puff has also included in this design. So we have collected the Naja shed skin. The very interesting finding I have noticed that if I collect the skin from the captive snake and also from the wild, the result is absolutely different. So what is that? I'll show. Look this slide very carefully. Now I have analyzed the heavy metal content of the shed snake skin. So if I collect the skin from captive, I usually I collect the captive snake skin from the Calcutta snake parks near Modhamgram, Badubagan. The Deepak Mitra is there. Unfortunately, he was no more. So he provided some of the skin. With the skin is usually from the snake which is in captive condition. Also, I have collected the snake skin from the wild, from the forest, right? Interestingly, I found when I carried out the experiment with the wild snake skin, they contained of a huge heavy metal, presence of huge heavy metal, so lead, cobalt, nickel. But if I perform the same experiment with the captive snake skin, the heavy metal occurrence is very poor. I do not know what is the exact reason. One of my possible conclusion is that possibly the snake absorbed the heavy metal from the ground and that forced the snake to shed their skin. So that experiment I have carried out in a nanogram level with a collaboration with National Institute of Oceanography, because in that institute at that time only they have the instrument that is known as the ICP-MS, because atomic absorption spectrophotometric determination is not accepted in the present day paper. So I carried out the entire heavy metal extraction with the help of National Institute of Oceanography. The ICP-MS data shows that the shade snake skin contain huge amount of lead, cobalt, and the nickel. This is the total fat content, because skin is also containing huge amount of fat. This assay says that is snake shed skin effect on the virgin estrus cycle. So the simple experiment I have performed, I show the next slide. This slide is very important. This is my first experiment with this shed skin. Look very carefully, this histological slide. This is the control slide of the ovary having a clear Graafian follicle is there. The model is a rat, but this is a day 5 treatment with a shed skin extract. So you find there is a no mature Graafian follicles, and here after 10 days all the follicle is atreatic follicle. That means if there is some compound is present that directly acting on this ovary, so there is Graafian follicle is not there. So this anti-fertility like structure have been developed histologically that have been proved. This is the lining of the uterus. The uterus is normally is a thick lining the control. In the day 5 the uterus tissue has been torn, and in the day 10 it is disoriented. The vanish of this uterine gland, it is distorted follicle have been there. So this is my first experiment at Maulana Azad College with this snake shed skin, and I found that there are some compounds are present in the shed skin. So what I am trying, I would like to purify that compound which is present in the shed skin. Simultaneously, I also carried out the hormone level determinations. Also some other activity. It has in some analgesic activity, is a pro.
Sorry to interrupt you, sir.
Yeah,
You
yeah,
need
yeah.
to wrap up now.
Right. So antioxidant profile has been also scanned. This is the purification profile. I have isolated 2-factor NNSS2, NNSS1, and that character has been details analyzed and characterized. This both the fractions are details analyzed. Interestingly, we have found this following amino acid present. That is again collaboration with Indian Institute of Chemical Biology. The total fractions have been characterized. The FTIR analysis, it has been found this amino acids are different. They have aliphatic stretching. They have the beta sheet hydrogen bonding. So detailed characterization. After detailed characterization, I again repeat the same experiment. This is histology and ovary that confirm that yes, both the fractions have the same thing. Carried out this apoptotic marker, whether there any apoptosis will takes place, that increases the caspase 3,9, Bax decreases caspase 8. So similar experiment have been performed with the urinary marker. A lot of urinary marker. I just try to focus the whether any reproductive toxin is there or not. I proved that yes, there is a reproductive toxin. The same experimental design with the NNS1 and NNS2, the same experiment. Here it is a biomorphometric changes. So the puff head length, puff neck length, puff tail length, etc. The significant changes in the prenatal and the postnatal development of this puff. This is same experiment with this NNS2. The Graafian follicle, no follicle is there. So all these are the atreatic follicle, but here it is NNS2. The same experiment, the same protocol with 2 different fractions. So similarly, I have carried out at present, this work is also going on. I am carried out with the female line, but the same experiment, that reproductive toxin has been also, existence of that toxin is also proved in the male line. There is no sperm, right? So that will destroy all the sperm. This is a simple histology of the ovarian tissue that has been carried out. The morphology of the sperm head, that work is still going on by our postgraduate students, so for their project work. So the lot of changes have been also takes place in the male line. So this is the last slide, that is a possible mechanism of actions through estrus cycle. I proved that during this cycle, all these hormone level changes, and that will probably act through the apoptosis, because it has a marked action on caspases, BCL2, Bax, etc., etc. Thank you very much.
Thank you, sir.
Thank you, sir. That was a great piece of information we received from you. So we have a few questions.
Yes.
Um, you were talking about isolated heart.
Right.
Just out of curiosity, I'm asking,
Right.
how did you carry out
Yeah.
that
So
kind of experiment?
usually, if you take the isolated heart, we prefer the animal is a guinea pig, right? So after anesthesia, we just isolate the heart, totally heart, and put it in a separate chamber where artificial perfusion has been given there, because this is an experimental setup of a good pharmacological laboratory. This is a very common methodology of a medical science in their first year, second year pharmacological experiment. So isolated heart can be isolated from the body and put it in a perfused chamber, and the beating is recorded through a electrical system in a recording drum. The instrument known as a kymograph is a very common nomenclature for physiologists.
Okay. Another question we have from the audience, that is, did you observe any abnormal effects on your experimental models after
Yes.
administration of such bioactive compounds?
Yes, there are a lot of negative activity because it causes the pyrexia. The temperature of the animal body rises, but we still do not know why this animal the body temperature is rises, because in human body this temperature is being controlled by the hypothalamus. So possibly the bioactive compounds acting on the hypothalamus system also, but we are not going to dip into that model. That is a very negative effect of this bioactive toxin. The body temperature increases.
Thank you very much, sir. That was a brilliant presentation. It was a great privilege having you with us. Thank you.
Okay, thank you.
Moving on to the next speaker. Now, if one takes a closer look at the alchemy of an achieving person, two distinct virtues pop up, and those are perseverance and hard work. Our invited speaker, Dr. Gargi Bandyopadhyay, is a living idiom of such an achiever. At a very young age, she has accomplished herself as a research scientist in the Department of Biology, Yeshiva University, New York, USA. She obtained her master's degree in physiology from Calcutta University and did her PhD in biophysics and molecular biology from Jadavpur University. Dr. Bandyopadhyay pursued her postdoc from Department of Biochemistry and Molecular Biology, University of Maryland School of Medicine, Baltimore, USA. She is actively engaged in doing research in the field of cancer biology and stem cells. Her current project pertains to gene expression profiling of invasive breast cancer cells taken directly from patients. We are keenly interested to hear from you, ma'am. Please carry on with your presentation.
Can you hear me?
Yes, ma'am, you're audible.
Okay. So I'm trying to share my window. Can you see my?
Not yet, ma'am.
Not yet. Yeah, it's open. Can you see my screen? My slide?
No, ma'am.
What's going on?
Your screen is visible, madam.
So there is no slide.
No slide.
No slide. Okay. Okay. I'm again then sharing. It's doing. I'm doing my entire screen, or so now?
Right now, madam, nothing is coming at the moment.
No slide at all?
No slide. Your screen is not shared.
Cancel.
I believe you have to open your slides first, madam, and then.
Okay. I am now opening. I open my slide.
Yeah, and madam, then there is this icon
window.
You can see in the center, present now, an arrow which is pointing towards the top.
I believe now you can see. Can you see?
Not yet, madam. present now. Yeah, madam, it's come.
Yeah, okay. Okay. It's clear?
Absolutely, madam.
Okay, fine. Sorry for the interruptions. Okay. So, thank you for the invitation. And I am Gargi Bandyopadhyay. As she told about me, I am doing right now, I'm doing as a research scientist in Yeshiva University, New York. And our team, our university, we have a collaboration with Albert Einstein College of Medicine, that is also in New York. So the John Condellis lab, our lab is jointly working with the John Condellis lab in Einstein. So basically my work is on the cancer stem cell, breast cancer stem cell. So breast cancer is nowadays is mostly reported cancer among the women in worldwide, and ultimately it's caused the morbidity or mortality. And it's leading cause of cancer, breast cancer is a leading cause of a second cause of a cancer related death in women in USA. So, and the TMM, my study is based on the TMM, tumor microenvironment of metastasis. That is a unique site, how the cancer stem cells is related to this site. My total few of my studies, the part of my studies I am going to discuss. So going to the next slide. Epithelial plasticity and stemness. How the EMT, epithelial mesenchymal transition, is related to the invasive cancer. Epithelial mesenchymal transition is a very important biological process that involves in many biological development and embryonic development, wound healing, but whereas it involves in various invasive cancer promotion and progression of the invasive cancer cells also. So our department, the main group of the Condellis lab, they are working in a particular protein, that is a MENA protein. Is a mammalian enabled protein. It's a kind of actin binding protein which is actually involved in this EMT process. And ultimately, there is various type of cancer, means MENA high expression is correlated with the breast cancer, colon cancer, in the cervical cancer, the various type of cancer. Condellis lab is actually basically working on this MENA expression, MENA protein, how it's involved with the breast cancer, how it's involved with the cancer promotion and progression. So, and this we saw that there is that is also that this group also working on a particular unique site. This is called TMEM, tumor microenvironment of metastasis. Is an anatomical structure present in the primary tumor cells of the breast cancer patient, which ultimately helps for the cancer cells for the metastasis. What is metastasis? Metastasis is a complex multi-step process by which the cancer cells detached from the primary tumor cells, and it detached from the primary tumor cells and it enters to the blood vessels. And through the blood vessel, these cancer cells are then transported to a different organ and from the secondary tumor. Ultimately, it's called the metastasis. So, so our group is mainly looking for this cell, this unique site. This is called TMEM. How it involves with the tumor metastasis, and it is already shown that the MENA high expression, MENA's cancer cells is highly expressed in this TMEM site. The details of the TMEM site I'm going to discuss in my next slide. So this it's known that it is already proved that MENA cells are involved in the tumor, the cancer cells metastasis, and this cell is highly expressed in this area, this TMM area. So now I came to the picture, the stemness and TMM, how cancer stem cells are related to the TMM. What is cancer stem cell? Breast cancer stem cells or cancer stem cells are a very small population of the cancer cells that which has the self-renewal property, and which ultimately has the promotion and progression of the heterogeneity of the breast cancer cells, and ultimately it again form the cancer tumor in a different organ. And most importantly, most of these cancer stem cells are chemo-resistant. So is there any involvement with these cancer stem cells with this TMM site? This is our main agenda. Means that there is a study that in that Condelis group already shown in this PNS paper, that the invasive cells has the higher number of cancer stem cells compared to the average primary tumor cells. So how we will know that what is the cancer stem cells? Is well-known breast cancer stem cells marker are CD44 positive, CD24 negative cells, ALDH1, CD133. Those are very well-known breast cancer stem cell marker. So this is, here we can see that this is a growing tumor cells, primary tumor cells. Tumor cells are detached from here, and it now is coming to this TMM site. So what is TMM? TMM is consist of three kind of cells. One is the endothelial cells of the blood vessels. The another one is the macrophage cells, and the other one is the cancer cell, the tumor cells. Without help of these two cells, the cancer cell cannot enter to the blood vessel. This is our finding. In different papers, we showed that without help of these two cells, cancer cells cannot enter to the blood vessels. And this is a sign signaling paper. It was shown that this TMM score, there is a particular the TMM. Our pathologist, Dr. Maya Octe, she is actually doing the scoring part. That the TMM score is involved, is correlated with the MINA form of the fold change. Means the MINA higher expression means more TMM number. So the cancer cell which is connected to the TMM, whether it has the stemness or not, that we can do in by the several ways. That is, the first we collected the cancer patient samples, patient's FNA sample from the hospital. With the patient's FNA sample, we undergo with a couple of experiments. One is the flow cytometry. Another one, the in situ hybridization, fluorescent in situ hybridization, which is called FISH, and QRT-PCR. The other way is this one, this is the fresh tissue. We did the flow cytometry, FISH, QRT-PCR. The another unique assay that we developed is here. I'm going to discuss in the later slide. This is item is called the in vitro trans-endothelial migration assay. That assay we performed. And this one is the FFP tissue. Same, we collected the FNA samples and we do the FFP tissue section and do the immunohistochemistry staining and this TMM score. It is the TMM score done by our pathologist, that how the patient, this TMM score differ from patient to patient. And this is the unique site of the TMM, that is the cancer cell is attached with these blood vessels. So initial question then came, is there a correlation between the TMM density and the % of cancer cell expressing stem cell marker in patients? Do item competent cancer cell express stem cell marker? Can cancer stem cell marker be included in vitro by co-culture of the TMM components? How can we block stemness induced by the TMM components? So identification of the breast cancer cell expressing stem cell marker from the patient FNA sample. As I told in the previous slide, that after collecting the patient FNA sample, we undergo the flow cytometry. The flow cytometry, it is shown that the patient who has this is here is a data I cannot show. Maybe this is a data. This is patient sample who has the higher CD44 positive cells has the higher TMM score compared to the patient who has the lower TMM score has the lower number of CD44 positive percentage. And this is that's a triple immunohistochemistry staining. It's showing the TMM site, the endothelial cells, tumor cells, and the macrophage cells. So this is our in situ hybridization data. It's showing that the cancer stem cell marker, the CD44 positive cells, CD133, ALDH1 is significantly high when it increases with the number of TMM. Means more TMM sites, more breast cancer stem cell marker. So definitely then the stemness is related to the metastasis. And this is our QRT-PCR data. QRT-PCR data also showing the same thing, that LDH and CD133 is more the patient who has the higher number of the TMM score sites. So, and this immunohistochemistry now the staining, I showed that the cancer cells, the green color is the ALDH marker staining. The cancer cell is close to the blood vessel. The blood vessel is red color, is colored with the CD31. So the cancer cell which is attached to the blood vessels has the more stemness compared to the cancer cell which is the far away from the blood vessels. So definitely that means the cancer cells which are related with the close to the blood vessel has the is TMM site has the higher stemness. This is we confirm by the image software analysis is a pixel intensity that the distant or the close to the blood vessel cells shows the high is a significantly higher ALDH compared to the distant site. So basically now I'm going to discuss about the in vitro trans endothelial migration assay. Initially I was hired to establish mainly this assay to isolate the cancer cells from the patient, the FNA samples, and undergo to the ITM assay. What is ITM assay? Is in lab artificially we developed an assay kind of the same item kind of situation. What the TMM situation. TMM is consists of, as I told before, that the cancer cell, macrophage cells, and endothelial cells. The same kind of situation we created in the lab artificially is called the item in vitro trans endothelial migration assay. So what is the assay? Just in brief, the 48 hours before when the cancer, the FNA samples come to our lab, 40 hours before, I placed cultured a particular number of HUVEC cells, is say about, I think the 120,000, 150,000 cells, HUVEC cells on the back side of a filter. I just keep the filter upside down and I plated the 150,000 HUVEC cells on the bottom of the filter and let the HUVEC cell grow for 48 hours. After that, when the cancer patient sample, FNA sample came to our lab, I colored the cancer cells in the green. with the green seal tracker dye. And I have to add the macrophage, as I told before, that without the help of these two cells, macrophage cells and endothelial cells, cancer cell cannot cross the blood vessel, and here cannot pass the HUVEC cell membrane. So I have to add the macrophage cells. Here we consider the mouse macrophage cells. Is a MET1 cells, that is mouse macrophage cells, I colored with a red-green cell tracker dye. And there is a ratio that 15,000 cancer cells and 60,000 macrophage cells. This 1:4 ratio cells, I mixed together and put it on the top of the filter, where in the bottom of the filter already there is a tight layer of HUVEC cell already prepared. So now the assay is, I let the assay run for 18 hours. After 18 hours, then we collected the 3 population of cells on the top cells, these cells which is stuck to the HUVEC layer, and these cells which is in the bottom of the filter, 24-well plate cell culture 24-well plate. 3 population of cells, I took it to the Einstein, and there is a particular space, the facility, and just fine needle aspiration assay. With a fine needle, I collect only green colored cancer cell. It's a very tough job, and on a good day, I can collect only 7 cells or 10 cells per day per population. So 10 cells from here, 7 cells from the bottom, and 7 or 8 cells from this layer. So these 3 populations of the cancer cells I collected, and then RNA extracted from these cells, and this is a very low amount of RNA, because I'm doing with the 5 cells, 5 single cells, 7 cells, or 8 cells, something like that. So we have to then mRNA amplification. We do the mRNA amplification with the amplified mRNA, with the labeling and hybridization, labeling with the Cy3. After that, we do the hybridization, hybridize in a slide in the genome microarray, and then we collected all the gene profile. We do the all gene profiling of the top, middle, bottom. And I'm not going in details with this assay and this gene profiling, but interestingly, the bottom cells, these population of the cells show significantly higher stem cell marker compared to the top cells, means the loading control, whatever the cancer cell we loaded here is significantly higher in the bottom cells. So that means the stem cells are mainly responsible for this metastasis, the invasion of the blood vessel, and which ultimately cause promotion and progression of the breast cancer cells in a secondary organ. So summary of this study, the TMEM-rich breast cancers are also enriched for the breast cancer stem cells as observed in FNS. Cells with the transendothelial migration activity express stem cell marker, working towards a comprehensive intravasation signature. Still, we are working on this because there are multiple number of the many thousand number of the gene profiling we have. So I'm just working with only the cancer stem cell marker genes. So now that the co-culture part. Now the question arises that which molecule or which pathway involves the stem cells, stemness of this stemness of the breast cancer in this TMEM region. What is the molecule responsible? What is the pathway? So for this, first in lab I do the co-culture. I took 2 different breast cancer cells. One is MCF7 and MDR3. One is very well-known breast cancer cells, and it's already.
Sorry for the interruption, ma'am. You need to wrap up in a few minutes.
Okay, sure. So MDR231 is more aggressive compared to MCF7, and it's shown that when I co-culture with the macrophage cells, with the cancer cells, there is a significant increase of the 24 CD44 positive and 24 negative cells, which is a known cancer, breast cancer stem cell marker. But there is when the cancer cell is separated with the macrophage, with the filter, there is no significant increment. So that means the physical touch of the cancer cells and macrophage cell is needed for the stemness. And the similar study I did with the endothelial cells. Interestingly, endothelial cells shows in both situations, is a no physical contact or in contact, it increased the stem cell marker. So this is our probable pathway. And then I applied a few single molecule cytosine. I applied, interestingly, salinomycin is a known Wnt blocker pathway, is decrease the stem cell marker cells when I do with the co-culture. When I apply to the co-culture, is a salinomycin significantly decrease the stem cells. So might be then came to mind that Wnt may be responsible for this stemness. So right now, I am working with the Wnt blocking antibody that is called LRP6. We procured from Oklahoma State University. The LRP6, interestingly, LRP6 working far better compared to the salinomycin. That it's when we applied the LRP6 in very small amount, is significantly reduce the stem cell marker gene in the co-culture. So might be then Wnt is responsible for the stemness. So that I confirmed this again. These are our Wnt finding by collecting the conditioned media from the co-cultured. I do the ELISA, and we saw that in different co-culture medium that the Wnt is the cells when is co-cultured with the cancer cell, co-cultured with the endothelial cells and the macrophage cells, in the free there is the free Wnt is present. We do the ELISA with the Wnt antibody. So the animal study, this is pretty much in very small version of my work. I didn't now here present my animal study. We are basically now doing the 4 different groups of animal. We are injecting with the control animal cells with the tumor formation. The second group is the very well known, I applied the doxorubicin, the chemotherapeutic agent, and third one is doxorubicin plus salinomycin, and fourth one is the doxorubicin plus this LRP6. Interestingly, LRP6 showing a significant reduction of the tumor after 5 weeks of our the chemotherapeutic treatment. Summary: TMEM-rich breast cancer are also enriched for the breast cancer stem cells as observed in the FNAs. Cell with transendothelial migration activity express the stem cells marker. Components of the TMEM can induce stem cells marker via multitude of the interactions, whether it's juxtacrine or paracrine, means either it touch with each other or it's physically separated by the membrane. So Wnt pathways seem to play an important role in the induction of the breast cancer stem cell marker by TMEM components, endothelial cells and macrophage. And lastly, I want to acknowledge my supervisor, Dr. Sumanta Goswami. He is associate professor of Albert Einstein College of Medicine as well as Yeshiva University. And Dr. Maya Okte, she's the pathologist. As I told you, that she does all the TMEM scoring. So should I go back to the main screen and stop sharing, or I should go on this sharing mode? What should I do?
Hello, can you hear me, ma'am?
Yeah. Should I
Hello?
go to the main screen or stop
please come
sharing?
again. Please come again. What did you just now say? Yes.
Then I'm stop sharing.
Hello?
Yeah.
All right. Thank you, ma'am. So
main
that was a
screen,
very
yeah.
smart presentation.
Yeah.
Very smart presentation, quite informative and interactive. So I would like to ask you, ma'am, that we know that the stem cells, they are very quiescent, and in certain when cancer crops up, it then revives back, it comes to life and starts dividing. So ma'am, have you seen any upregulation or downregulation of any genes or something like that? Any particular genes which leads to the excessive proliferation of those stem cells leading to cancer? Oh,
Basically,
just out of curiosity, nothing else.
yeah, sure. So basically, we work with the mainly as you saw in my presentation, we work mainly on the CD44 positive cells and ALDH cells. Only for the QRT-PCR, we observe the CD133. As I told you, we in the gene profiling study, we have few hundred genes that are related to the cancer stem cells is upregulated in the population of the cells which is invested in the blood vessels or in the HUVEC cells. So we are still doing, or when you're still working on that. The other cells, other type of, we work on the ALDH, CD133, CD44, CD44 positive cells.
All right. So thank you, ma'am. It was a pleasure having you with us. Thank you.
Okay.
Have a good day. So
can I
moving
leave?
on to the, yes, ma'am, you can leave.
Okay, sure. Thank you.
Moving on to the next speaker of the day, I have the honor to introduce Dr. Saurabh Seal, a very prolific and generous personality. He has been a very workaholic person and has proven his worth time and again. He is currently an assistant professor at the School of Earth, Ocean and Climate Sciences, Indian Institute of Technology, Bhubaneswar, Odisha, India. He was awarded PhD degree in Physical Oceanography from IIT Kharagpur, India in 2012. He has a postdoctoral experience from Japan Agency for Marine Earth Science and Technology. His research areas are physical oceanography, ocean circulation modeling, and air-sea interaction. Dr. Seal has published more than 30 papers in reputed international journals, completed 6 sponsored projects funded by different research boards of India. He has supervised 5 PhD theses and more than 30 master's theses since he joined IIT Bhubaneswar. It would be a great privilege, sir, to hear from you. You may start now. You will be having 30 minutes.
Sure. Thank you, madam, for your nice introduction. Can you please confirm that whether this is visible?
Yes, sir. Clearly, you're visible and audible.
Okay. And you can also see my point, right?
You have to share it with the whole screen. It is not showing on the whole screen. Yes, now it is okay.
Yeah, now you can see.
Yes, sir.
Okay. Okay, thank you very much. The organizing committee for inviting me. And particularly, I thank the committee to choose a topic which is on sustainable development. So my talk will be a little bit different what we heard in the morning. And there is, I want to talk a little bit of the environment side and particularly role of oceans towards the sustainable development. So in the talk, I'll talk about that. What do you think about the oceans in our daily life? How those are related to sustainable development, and particularly cyclones. I just want to give some informations about this thing because this is important, and we all nowadays we are seeing the devastative nature of that. And there are just information I want to share, the ocean observations which are done by the different agencies, Government of India for climate services, and some take-home messages for the strategies for sustainable development. So here, you know that United Nations, they have mentioned the 17 strategies or 17 goals for particularly sustainable development, which is no poverty, hunger, educations, and there are many, but there are few things which is related to the oceans, which I will talk about: the clean water, energy, climate, and life below the water. So this is particular topic which is related to, very much related to the, or directly related to the oceans. All other things also somehow it is related to the oceans. So if you talk about the sustainable development, some points, it's a good health, good economy, good educations, renewable energy or clean energy, or good environment to live. As you know that there are all the populated cities in the world which is situated in the coastal regions for different regions. And to make a sustainable environment, we must look at the coastal regions and particularly for the oceans. So that will be some aspects I want to bring to you people to think about this. So what do you think about the oceans? It is something about the nice beaches, okay, so some place to spend our holidays. It is something about the foods or marine lives. There are something which is port and harbors where we have different things are being transported from one place to others, and also the navigations, and also the marine security involved in that. And also we talk about the clean energy or green energy, which is renewable energy we can also get from the oceans. But one thing is that, okay, we have also some the other version of the nature which is a bit destroying, but this is a natural phenomena, that is the cyclone. So if we note down, okay, what will be the particular topics which should think of. One is the transportation, food, energy, coastal and marine ecosystem, weather and climate for the sustainable development. Here is one stat, I took it from NOAA, USA. So they have come across with some values. They're telling that, okay, it's more than 50% the oxygens which we are consuming that is coming from the air, and it is the carbon dioxide also it is taken by the ocean. Okay, so and also there is the climate regulations are there. The 70% of the earth's surface we know, 30% is land and 70% is the ocean. So they are also playing an important role for transporting the heats from equator to the pole, and it's regulating the climate patterns. It is also the 70% of the US transportation that is coming through the land. And also we use the beaches, which is for recreation, for fishing, and for other purposes. And also the economy, which depends partly majorly on the ocean's part. We have foods, not only fishes, but there are some other peanut butters and soy milk we are coming from the oceans. And there are medicines. You can see there are some medicine listed. They have it is arthritis, other diseases. My previous speakers have been talking about the different medicines coming from the different animals and planets. So those will be useful. This is also the source of those things are from the ocean. So we can also think of that we are getting medicine from the ocean. If I talk the marine ecosystem, so there's a marine life. So we have certain species which are living, so which is controlled by the ocean temperature, salinity. So some species like some environment. So depending on their conditions, different species are available. Which some species lives in the upper ocean, some are in the deeper. And also particularly in the mangroves, particularly in the in the West Bengal, we have a large area of mangroves, which is naturally grown, and it is also useful for protection from the cyclones. Okay, so we should also think about the sustained ability of those things which will help to protect from our natural hazards. And there are also some corals and nice things which we also get from the oceans. And importantly, it takes the carbon dioxide, it relieves the oxygen to that. There are some atmospheric disturbance, not only from the cyclone, but there are monsoon depression, which brings a lot of rain to our Indian continent. It is mainly coming from the oceans. One thing I just want to clarify that why you need to think about the oceans and the environment. This is some physics I some diagram. I'm not giving anything numbers here, but if you understand the process, we are getting radiation from the sun. It is coming through cloud and it is going to the oceans. Now, if we are making this ocean polluted, or means the ocean will be trapped more at the surface and it will not be able to go down. So that time the ocean will be warmed up more. Even from the clouds, if there are some things that, okay, some radiation is going to the atmosphere and atmosphere is also radiating back. So if we are changing some composition in the atmosphere, which can absorb the more heat or reflect the more heat to our earth, so that time also will get more temperature. So there are some simple complex physics which is playing a role, and we should take care of these compositions of the atmosphere as the ocean to control these full radiation processes. Slightly deviation from thing can give a some larger fluctuations from whatever we are seeing. So this clean air, clean water will help to maintain that whatever the temperatures we have. And also this is some animations you can see from the ocean's currents for the Indian Ocean, which you can see the heat are being transported. The ships are following some routes which is going from one place to others. Now I'll come to a little bit of the tropical ocean, we generally see from our almost in the pre-monsoon time and post-monsoon time. During May, we felt the influence of the cyclone, particularly in the western East Manipur. So that time there was some storm surges, some flooding happened. So people need to aware of these things. So I want to bring some statistics in that. So here some cyclone, you can see this has happened in 1971, particular which hits the Bangladesh, and as per the record, the deaths were more than 3 lakhs. When it happened to the 99 cyclone, it comes down to 10,000. And then in last two, in Phony Amphan, it is close to 100. So we are looking for some day where this fatality will be very minimum and finally zero. So what is the improvement happen through these things? It's whether the cyclones have changed their strength, but it's not. There are studies we are telling that cyclones are becoming more stronger. But the thing is that whatever, if you can tell this is success to prevent the fatalities, again the natural events, but natural hazards we can say. So those things are not, you can't stop cyclone, it will happen. It's as I told, it's a natural process. It happens every year, everywhere. It's not only in the Bay of Bengal, it is happening in the Pacific, it is happening in the Atlantic. But the thing is, the strategies we have to make so that we have a we can counter those challenges. So if you call this a success, so it is coming from the observations. People are taking more observations. They are monitoring the ocean and climate, ocean and atmosphere. There are good models, particularly from discipline of mathematics, physics, mechanical engineering. They are coming with their knowledge of the fluid dynamics and they are applying to analyze the weather predictions. So that we are happening. But the most important thing that we have to have the awareness among the people, so that if some agency, government agency is telling me the cyclone is coming, so we should take care of that and we should follow whatever their instruction is that. So sometimes what happen, some regions like in West Bengal, we don't see that much of cyclonic effect compared to Odisha and Andhra Pradesh. But so people are a bit casual, okay, the cyclone is coming, it will not happen, it will not do anything. But what happened for Amphan and Iyas we have seen. So that awareness background should be there and people should be follow that whatever the instructions are giving them. And nowadays we are blessed with advanced information technology. We have all smartphones. We have a good network coverage, and we get SMSs, we get WhatsApp messages. We generally have different TV programs. Okay, so those things are coming, and which is helping us to encounter those conditions. So we need to follow that. We need to spread the awareness. We need to take use of the information technology so that we can reach maximum people. So there is some, that India Meteorological Department, which actually started from Kolkata in almost 150 years back. Now its headquarters moved to Delhi, but that time from British era, it is there, and the people. It is an interesting factor. People have studied from there. So I'm not going to very details of the statistics, but we should follow the instructions and guidelines from the IMD so that it can be followed. And then we have some tropical cyclones. We have seen that, which is it's, as I told, it's not happening in only the Bay of Bengal or Arabian Sea. There are more actually in Pacific and Atlantic. Okay, so they have some strategies, they have some program, and also our Indian community is coming up with a greater extent to spread the awareness and give the good prediction system. In our recent studies, we have seen that, okay, in recent times we have a cyclone intensity is increasing because there are SST is changing. This is a part of the climate change, and also we have seen there are some westward shifted of the cyclone tracks, which makes more landfall around our West Bengal and Odisha. So, and this studies could possible because of the ocean observations. So I want to say that how now ocean is happening in the deep oceans when cyclone is forming. Satellite is one thing, but we need to also know what is there in the actually deeper oceans. So there are different organizations. We have Ministry of Earth Science in India, and we have National Institute of Ocean Technology. So they deploy different buoys in different location in Arabian Sea and Bay of Bengal just to monitor the cyclone movement and the oceanic conditions. There are some profiles which calls Argo. There are some buoys which called Rama. So those things are taking continuous observation to monitor the oceans for weather and climate systems. Now, thing is that we can cloud the clean energy which we can get from the oceans. So one is we have the tidal energy in India, which is not that much famous now, but yes, we need to think of that. Although the east coast of India is not that much potential, but west coast, particularly in Gulf of Khambat, it's very potential zone is there to get the tidal energy. So use the tides for generation of energy. There is also energy is called that ocean thermal energy conversion. So where we can make use of the warm water at the surface and deep water, cold deep water to regulate the process of a chamber. So that through that one we can energy, and it is also in developing stage. Okay, so there are some energy which is already in Lakshadweep and these regions. People are already started using this ocean conservations, oceans energy conversion, that thermal energy conversion or technology there. There is also people are think that the osmosis power, that is we have a low salinity from the rivers and high salinity from the oceans. So that difference can be useful to generate some ocean energy. So as you know that United Decade of the Ocean Science of Sustainable Development, they declare that one. They have a visions that okay, the science we need for the oceans and whatever for our development we need, and also we need to transform that ocean science solution for the sustainable development, connecting people and our oceans. And there are some another ocean energy things is exploration, like we have a deep ocean polymetallic nodules, and also there are some hydrocarbons which are taken from the ocean. So this is another part of the ocean resources, and it's called like a blue economy. We can use the oceans for creating the energy and increase the economy of a country. So if you are telling the strategies is for, we need to go for a clean and healthy oceans. We can use the blue economy, we can use the renewable energy for the sustainable development, and also most importantly, we need a sustainable environment for better futures. Okay, thank you. So if you have any questions.
Thank you, sir. That was a very nice presentation. Sir, I
Yes.
just wanted to know on behalf of my audience
Yes.
that we have been noticing huge amount of pollution in the oceans, marine pollution that we
Yes.
say. So, is there any link between the increased pollution and the energy extraction or energy production? Is there any link between these two?
Yeah, marine
With
pollution.
rising pollution, does it impact the energy production in any way?
The energy in terms of ocean energy or other energy, I'm not sure, but yes, the marine pollution, what actually, as I told, if you are polluting the upper oceans, so there will be more trapping of the heat at the surface, so we can expect the more warm ocean. If it is more warm ocean, then it will also impact the environment, and also whatever the seasonal cycle, those things are being changed. Okay, so and also most which is the means worst part of that, it is impacting the marine lives. Okay, so which is the future source of the food, and that we are destroying. So in that said, that pollution is very a very bad hazards for the nature in terms of both from direct and indirect effect of that.
So that means it has impact, but that all those are indirect.
Yes.
Right. Yes. Yes. Another question we have is whether volcanic eruption from mid-oceanic ridges responsible for the destruction of marine ecosystem.
Yeah, that is a interesting question, but we can't say right now as of now because we don't have a observations for these things. But yes, it releases a lot of heats and particularly some of the minerals, but we don't have any in situ observation that how much it is being happening. So it is very difficult, but yes, there must be some impact on that, and we should look for more data before telling anything. Yes. Yeah, very interesting
Another
question.
question, yes,
Yep.
another question we have from a student, Ninad Bhagat. He asks, what is the scope of deep ocean floating wind turbines in Indian waters?
It's not deep. Sorry, madam, say again, please. It's deep ocean wind turbine.
Deep ocean floating wind turbines in Indian waters.
Yes. So thing is that there is the potential zones to have this wind energy in the offshore regions, but the thing is that whether the energy will be transferred to our mainland, because we are living in the mainland, so it is very difficult to get the energy. We can get the energy, but the thing is that how we'll transfer these things. Okay, so there is a challenges, but we don't have a floating turbine, but we have some offshore wind turbine which can generate more wind energy, and we have potential to get those, and particularly in the Arabian Sea and in the southern part of the Indian region, where the wind is relatively stronger.
All right, I hope you're satisfied with the answer, Ninad. Thank you, sir. Thank you, sir, for a nice presentation.
Okay, thank you, madam.
We now move on to our next session, which is the presentation session, and the first presenter. Excuse me. Yes, the first presenter we have on a list is Momi Jasmine. Are you there, ma'am?
Yes, I'm here.
All right. So, Madam Momi Jasmine is the presiding member of the organization SAS, Feel the Desire of the Nature and Society. She is here to share her experiences with us. We are delighted to have you here, ma'am. Can
Okay.
you can now share your screen?
Yeah, sure. Thank you. Is my screen is visible?
Not yet, ma'am.
Okay. Okay, let me try. Now is it working? Is it visible to you?
Yes, ma'am. It's visible now.
Okay, okay. So good morning, everyone. And thank you to BB College and the organizing committee for giving me the opportunity. Today I'm going to present one of our, my name is Momi Jasmine and I am a development practitioner and a president of an organization named SAS Feel the Desire of Nature and Society. And today I'm going to present one of our work on a pilot study on improving adolescent girls' social health by the intervention of NGOs. So here in this study, basically I will show the work of our organization. Mm, So in our this study, we have focused on the girls in their adolescence stage. So adolescence is a significant stage of human life. It is the time of experiencing new changes, physical, emotional, and intellectual. And according to World Health Organization definition, that adolescence is any person between the ages of 10 to 19. At this stage, adolescents develop some common characteristics like mood swing, identity crisis, value conflict, curiosity, and attraction to the opposite sex. With such characteristics, they are more prone to be experience risky behavior that can affect their social health and makes them vulnerable, especially girls in terms of reproductive health and rights. As the children enters the adolescence phase, they bring along with resources and vulnerabilities according to Bloom RW 2012 definition. It is the matter of concern. And social health is the ability to form satisfying interpersonal relationship with others. It can be said that to maintain good social health, it is important to adapt any situation, stress management, empathy for others, positivity, and power of right decision making. Teen pregnancy, substance abuse, sexting, and some major problems of adolescence which have influence on adolescent social health directly. And the key elements of social health include social support, social adjustment, social network, etc. So hence an attempt has been made on assess the social health status of adolescent girls and to find the scopes of intervention. The aim of this study is to work on improving adolescent girls' social health, includes health and well-being, interpersonal relationship, and that interpersonal relationship can be a relationship with parents, friends, or some intimate relationships, lifestyle, and overall social functioning of teenagers in coalfield areas of Asansol, West Bengal, India. Few objectives are to study the various laws and policies that contribute towards the social health of adolescent girls, to address the issues and challenges related to the topic, and to execute a program so that the social health standard of adolescent girls can be improved, and to develop strategies that can be implemented at macro level for maximum positive outcomes. In this study, the purpose of yes.
You may continue, ma'am.
Okay. So in this study, the purposive sampling is being used. Those individual belongs to the age group of 10 to 19 years of age and lives in coalfield areas of Asansol, West Bengal, India are taken. And a sample of 40 adolescent girls were taken from two villages of Dishidgarh. And to the tools we have been used, demographic information sheet to collect various demographic information about the sample, including name, age, gender, education, qualification, and marital status, etc. And a questionnaire for data collection from adolescents and their parents. The data has been analyzed by using the Microsoft Excel. The method of this program was as at the beginning of the study, a survey was conducted to measure the response of girls on various social health indicators. After need assessment, the NGO Assas FDNS has introduced a program named Jagriti for the improvement of adolescent girls' social health. Various awareness program and free classes on computers, spoken English, digital literacy, mental health, reproductive health and menstrual hygiene, mental health and etc. are offered to improve quality of lifestyle, health and well-being and overall social functioning of a person. After 6 months, responses are recorded again to analyze the difference. And the result we have been found that you can see from this diagram that showing the pre and post effect of program Jagriti on various social health indicators among adolescent girls. So when we were planning to implement this study, so we have prepared questionnaires on few topics and measured their level of understanding. So we have found that there were very less girls that are aware about only sexual health, and but mostly no one cannot say about the sexual health and right. Then we have also measured awareness on mental health, that if they know about the depression, stress management, and the process of that, if someone is in problem that they might need counseling or guidance. So what is the level of awareness about mental health and digital?
Deepika Gope. Deepika Gope, please mute yourself.
Okay, so digital literacy, which includes computer basic fundamental computers and also theoretical knowledge, and the things we need to know in this digital world, and awareness on romantic relationship, that includes that in which age one should be get married, and the relationships and all, and awareness on lifestyle related disease. So what we have found that more or less we found there is lack of awareness among adolescent girls, and it is quite natural also, as mostly no one talks on such issues with them. And the period of adolescence begins with the onset of puberty, a rapid changes in a rapid change in physical growth accompanied by continuous sexual maturity, and ends when individual assumes adulthood. And the adolescence period can be considered as a risky bridge also. During adolescent period, peer group of them plays a central role in their lives, which have very much influence in the behavior and attitude of the adolescents. Intimate relationship with opposite sex can become crucial in some cases. And under the program Jagriti, the NGO making the girls empower in such way that they can cope up with any situation and become aware of every malpractice which have direct influence on their social health. So in conclusion, I would like to say that nowadays we can see depression, addiction, suicide, relationship conflict, etc. are some common issues among all ages. It is basically starts from adolescence stage, depending on the environment they born and brought up, and few things comes from genetic heredity also. But it can be solved with proper strategy and intervention. Through the program implementation of Jagriti in a small level also, you can see the changes. There is a strong need of intervention at macro level also. changes in policy level to address the need and requirements can help to prevent many social issues to some extent. Thank you. Is there any question?
No question as such, but we are proud to have you people out there in our community. Thank you so much, ma'am. You people are doing a great job. It's a great initiative rehabilitating the vulnerable adolescents. Thank you again, ma'am.
Thank you.
The next speaker on the list is Partho Chattopadhyay, librarian, Hiralal Bhakat College. And the topic on which he's going to speak is introspective study on the school libraries of West Bengal, a state of India, a new perspective in 21st century. Are you there, sir?
Ma'am, I am audible. Am I audible?
Yes, sir, you're audible.
Myself, Partho Chattopadhyay, I am the librarian of Hiralal Bhakat College, Nolhati, Birbhum. Now I am going to present my paper through PowerPoint presentation. Madam, please wait.
Sir, can I help you in any way?
No, sir. I am trying. Some network related problem is there.
Okay, sir.
So actually, some sort of net problem is there. Now it is coming. Sir, it is now shown.
Not yet, sir. Not yet, sir. There is this icon in the bottom of the screen which shows an arrow which is up.
Okay.
Upward pointing arrow.
So now it is coming.
Not yet, sir.
Sir, actually now problem is there. So actually now I am simply talking.
Okay.
Some error is there. Actually, I am writing this paper from my survey on different schools of West Bengal and their learning habit of the students are being
Okay, sir. Go ahead. Go ahead.
Huh? Actually, school libraries.
Sir, go
Actually,
ahead.
school library is such a place. Actually, in our library system, there are three types of libraries. They are academic libraries, special libraries, and public libraries. And now I am going to present my paper on the academic library system. And school library is one of such the academic system. In West Bengal, the school libraries are established in the higher secondary school mainly. In the higher secondary schools, the school libraries plays a very pivotal role to render the education system throughout the school. Apart from their class study of the teachers, any particular teachers teaches on a particular subject. But from the library, the student can get various types of resources. From class 5, from standard 5 to standard 12, the library system is being followed in West Bengal in school. And in these schools, collection development policy is one very vital aspect, because basing on the collection development policy, a school library can foster their services. And I think that school library In our previous times, we found that when a lockdown period was there, then the open educational resources were the only companion to complete the whole teaching learning process. And some school librarians created their own e-resources to continue the teaching, not only from the open educational resources, but the hard copy resources, such documents on Rabindranath Tagore, Satyajit Ray, and any other eminent writers. And in India and in abroad, they are playing the many vital role for the continuing the teaching and learning process. And in my study, I observed that most of the school librarians are engaged to continue the teaching learning process easily. And the objective of the school library is to inculcate the reading habit among the school students. And school library is such a place where the teaching learning process is being continually done. And in West Bengal, the students are assigned various types of projects works, and my paper I made some suggestion that in the project works the student can use the public library in adjacent to their schools, and from the public libraries they can get more resources, and on the basis of their resources they can complete their project works and submit the same to the school. And in this way love and sympathy, and then a student can become a lifelong learner in future. Thank you, sir. Thank you, sir.
All right. Thank you, sir, for your talk. Is there any questions from the audience? You may ask.
Ma'am, some net related problem is there, and that's why I cannot complete this. That's why I cannot able to show my PowerPoint presentation.
It's okay. It's okay, sir. There are no questions as such. Thank you for your talk, sir. The next speaker on the list is Arendu Banerjee, who's a research scholar from University of Calcutta. And his topic of today's presentation is Sustainable Development, Migration and Aspiration of Human Life, a model-based investigation. Are you there?
Yes, thank you.
Yes, Arendu Banerjee, are you there?
Yes. Am I audible?
Yes, you're audible.
Okay.
All right,
I
sir,
shared.
you have 10 minutes.
Yes, thank you. I am sharing my screen. Is it visible to all?
Yes, it is clearly visible.
Okay, thank you. Thank you all for this opportunity. My topic is sustainable development, migration and aspiration of human life, a model-based investigation. Firstly, I want to describe the thing that the development and sustainable development are two different things. Only development and the economical growth and other form of developments alone are called development, which can be impacted on environment and our climate. But when it is in turn of sustainable development, this is also a development along with social inclusion and the respect for the environment. If we delink this development from the social inclusion and environment, then it will be a matter of self-sabotage. In this case, we can consider the example of a Roman Empire of ancient age. It is very known and it is very much discussed that Roman Empire failed because of internal and external conflicts.
Google Meet টা কোথায় আছে বলো তো, খুঁজে পাচ্ছি না। Google Meet টা কোথায় আছে খুঁজে পাচ্ছি না। Google Meet টা কোথায় আছে?
নেটই পাচ্ছে না এখানে তো।
Google Meet আছে এখানে ইনস্টল।
Is there
Achha.
any problem? Sorry.
কেন এতক্ষণ
Yeah,
কি
sorry
শুনতে
for
পাচ্ছিলেন না? আরে এতক্ষণ
Partho sir, please mute yourself. Yes, Arandu, you may continue. Sorry for the interruption.
Okay. Okay. We know that in the Roman Empire failed because of the internal and external conflicts on the time, but which is very uncommon. But the fact is the Roman Empire and the Republic both failed because they failed the sustainable development test. How that failure played out. A breakdown in a time-honored social norm, entrenched political polarization driven by economic inequality, repudiation of the common goods by elites, and environmental havoc leading to disease and disaster. That means the development of the then Roman Empire was not in the mode of sustainability. So the climate change and the diseases and the disasters, epidemics are always a matter of the failure of Roman Empire. And we know till date we are also in that state. That's why the sustainable development goals developed. Yes, there is some difference between the economy of the ancient Rome and us. Our is vastly wealthier, healthier, and more inclusive and more resilient. But which is the fact that we have to go for the sustainability in all means. And if we tends to understand the sustainable development with human life, we also find the human migration is a factor of this survival, or I can say the sustainable survival. Yes, the migration is a survival strategy sometimes, and sometimes it can be for the aspirational satisfaction. Now, if we define aspiration, that aspiration is a very complex psychological phenomena, which is a component of human desire. And people want to move from one state of aspiration to the higher state of aspiration in terms of dignity, good health, recognition, political power, ethnic grounds, religious stance, and many more. So if migration is a question of human subject, then aspiration and the desire can be related with these conditions. So if we consider that migration can be a level, or migration can be done for the level of a different level of aspiration, our main objective here to connect the tradition of ambitions or the aspiration with the tradition of migration. What is traditional ambition? Traditional ambitions are the two successive generations aspiration level. Here we want to model the different level of aspiration of two different generation, and in terms of probability, we want to show that this level difference between the two generations can be impacted on the migration levels. The basic structure of the probabilities of the interval transition is that if we consider two generations as G1 and G2, and we are saying that there are different level of aspiration in terms of I and J, we term the level of aspiration as 1, 2, and ... n, then we can say to enter in such a level of aspiration for a generation is Pi, and for the next generation is Pi dash, where and when we are moving from the level of aspirational state I to J, the probabilities are become Pij and Pij dash for two consecutive generations, with these two conditions that all the probabilities are greater than equal to 0 and the sum over all probabilities must be equals to 1. Now here we are modeling the interval transition probability instead of only transition probabilities. Here we consider semi-Markov process instead of only Markov process, because here we consider that the stay in any state of aspiration is a random variable, and that's where the different level of aspiration with the different level of migration is following a semi-Markov process. Here in a semi-Markov process, the main thing is to calculate the interval transition probabilities instead of the transition probabilities. Now, in our study, we assume Hij m. M is the time where Hij is the probability distribution function, where we can consider that it follows geometric distribution, because if some people or someone is moving from one level of aspiration to another level of aspiration is called a success, and if not, then it is a failure. And where this probability distribution is geometric, because we want that a successful aspirational level change, we want to consider that. So we describe this by Hij_m, and for the next generation Hij-m dash. Now this is our IPTMs, interval transition probabilities. Here the things that's W bar n is the waiting time matrix. That waiting time thing means if someone is moving from one state to another state, there must be some time to wait, and that is called the waiting time. And the distribution of waiting time with the average waiting time is W bar n. And this is the IP, the notations, the box notation is for the congruent matrix multiplication. We saw that in this section, the computation is only depends on the P box notation HM. H is the holding times. So we denote this thing as C, Cm and C dash M dash as the core matrix of our semi-Markov process. Now, if we consider that two generations core matrices are Cm and C dash M dash, then the probabilities of entering in aspirational state J from I in time M and M dash for two generations is Cij M and Cij dash M dash. Now the difference between two generations aspirations is denoted by kappa D, that is max, I and J, modular value of absolute difference between the Cij M minus Cij dash M dash. Here, if the differences, the Kd, kappa D measures the gap between the aspirational gap between two generations. If kappa D is tending to 1, then we can say that the gap of the aspirational level of two generation is high, and if this tends to 0, then the gap is low, which denotes that some people from the first generation is not so highly aspired than the next generation if the kappa D tends to 0. But if the kappa D tends to 1, that means that the generational gap, that is the second generation is more aspirational, or the first generation is more aspirational than the other generation. We can a data analysis, we perform a simulation study on this section, and we find some results that we present some very little tables due to time constraint. Here we generate 5x5 interval transition probability matrix with different time level and with different time waiting times. M are the different time level, and holding times are A's. So we get some probabilities, and if we see the difference that is estimated is this, which denotes that the gap is something measurable between two generations from the simulation study. And in this, if we consider the sustainable development in terms of the aspiration and the migration, we can see the gap, the probability of this gap of 2 generations aspiration level is continuously increasing. So we can say that people are migrated for either for their aspiration level or their survival strategy. If there be some sustainable development goals for the migrations of the world, then we can introduce some sustainable life for them. Thank you. That is my paper. This is my references.
Thank you, sir. That was a great deal of information we obtained from your paper. Now we move on to the next speaker. That is Professor Mahananda Kanjilal. And she's from Jogesh Chandra Chaudhary College. And she would be speaking on sustainable development and gender inequality. You have 10 minutes, ma'am. You may
Yes.
share your screen now.
Yes. Very good morning to everybody. Just a minute. Just a minute, please. Just one moment.
Madam must be facing some technical glitch. So we are moving on to the next speaker.
Hello? Hello, can you hear me?
Yes, yes, ma'am.
Yes, I'm ready.
Okay, okay, okay. No. Okay, you may continue.
Yes. Videota de eta tha. Can you see the presentation? Hello?
Yes, it is visible.
Hello?
Yes, it is visible, ma'am.
Okay, thank you. আমি তো এটা অন করবো। আমি কোথা থেকে চেঞ্জ করবো? এখান থেকে চেঞ্জ করি। এখান থেকে চেঞ্জ করলে পারবো না, আমি এখানে যাই। ওকে। Hello, good afternoon. Let me start with my paper, Gender Discrimination and Sustainable Development. I'm audible, I think?
Yes, ma'am.
Oh. Now, the topic of discussion today is gender discrimination and sustainable development. Now, first of all, Now, sustainable development lays emphasis on process of present development, but also analyzes its implication on future development. So the development that is going on in each and every countries of the world, the present development is important, but nowadays, a lot of researches are going on about the future sustainability of the development process. So therefore, nowadays, this issue of sustainable development has become very much relevant nowadays in each and every parts of each and every spheres of economics. Now, initially, sustainable development was mainly focused on environmental issues. But nowadays, it includes social, ethical, and other economical issues also. And here, the factor gender has become very much important in sustainable development. Sustainable development requires equal participation of women in all respect. And that's why gender development has become an inherent or integrated part of this subject. And social equity is linked to this gender equity. Now, just a minute. Now, if we consider the sex ratio in India in 2011, our last census, we find that some of the states we have undertaken, and here we find that only in Kerala and Puducherry, the sex ratio is more than 1,000. In other states like Tamil Nadu, Andhra Pradesh, Meghalaya, Odisha, Karnataka, West Bengal, it is less than 1,000. That is, gender inequality can be explained with this sex ratio in India. Now, next thing is some of the aspects of gender development or gender inequality can be reflected in female labor force participation rate in India. In India, labor force participation rate was 37 in 2006. But in 2012, in 2017 and 19, gradually it has decreased. So over the years, we find that female labor participation rate has decreased, which reflects gender inequality in our country. Now, if we compare the situation of India with other countries, we find that in 2019, in India, female labor participation rate was 20, whereas in other neighboring countries like Bangladesh, Bhutan, Nepal, Pakistan, Sri Lanka, it is more than India. Okay, so it also reflects that gender inequality exists in the case of female labor participation rate in India, and the world average is 47. Now, next we go to Global Gender Gap Report to 2021. From that report, it has come out that out of 156 countries, rank of India is 140, that is behind 28 countries. And the concerning fact is that for the first time in India, it is the position of India is in last 20 countries of the world. Now, what are the parameters that has been undertaken to measure this global gender gap? The parameters are participation of women in education, health, wages and salaries, and politics. And here it has come out, especially in the category of second and the fourth category, India is much behind of other countries. Now, in Sustainable Development Goal of United Nations, it has also come out that in 2020, the rank of India decreased below its previous level. And in this Sustainable Development Goal, gender development is a criteria for making this index. And here also again, India is found to in a position which is below Bangladesh, Nepal, Bhutan, and Sri Lanka. Now, in the case of gender equality, India's ranking is lower than, again, China, Sri Lanka, Nepal, and Bangladesh. So gender inequality rate, that is, men and women are not considered as equal in India, which can be found from these findings, international findings. And Human Development Report 2019, over there also we find that out of 189 countries, the rank of India is 129. Now next we go to the reasons of this gender discrimination in India. The first reason is, of course, patriarchal society, which is prevalent in India even in this 21st century. The second one is patrilineality, that is inheritance through male descendants. That is another reason. Number three is patrilocality. Usually married couples, they live with husbands' parents. And number four, socially girls are treated as burden due to some system of dowry at marriage. And the fifth one is conceptually it is believed that only sons will look after their parents. So all these are socio-economic reasons behind this gender discrimination. Now, gender discrimination prevails in almost all areas in India: social, cultural, political, economic, occupational, and educational. And this affects sex ratio, women's health, educational attainment, economic conditions, labor force participation rate. Now, what are the effects of this gender discrimination in society? First of all, female children are neglected in the matters of nutrition, healthcare, education, and other aspects. More girls die in India than boys. Crimes against women, like rapes, dowry dates, honor killings, are prevalent, very much common in India still now. Girl trafficking, this is a vital issue. We find that is crimes against women. We find this is one of the most important fact, that girl trafficking, 51% of the trafficking victims were children, of which more than 80% were girls, found in 2018. Child marriage is also in 2011, 12.15 million children are married, of which 8.9 million are girls. Female foeticide is also very much common. Infanticide, that is killing a female child, is also prevalent in our country. In 2011 census, it has been found that 82.14% males are literates, whereas 65.46% females are literate. So there is also a gender discrimination in the case of education. An unequal payment to female labor is also there. Now, what are the measures needed to reduce this gender discrimination or gender gap? First of all, education, creating attitudinal shifts. Simple traditional education is not sufficient to bring the social change or the attitudinal change. So proper education regarding gender discrimination is necessary. And continuous efforts towards breaking the myths. Even within the literate persons also we find this kind of traditional beliefs. So, and another thing is government campaigning. The positive attitude to change this social myth is necessary from the part of the government. And efforts are needed to change the perception of the society. It takes years and years after years to change the perception. So a perception of a particular social perception, we can say, the conservative attitude. And another thing which is also very much vital, that is women empowerment. Although women empowerment is not sufficient for changing this social the gender discrimination, but it is one of the most important aspects, of course. And development can have opposing impact on sex ratio. There is evidence that crimes against women decline with spread of education. Although in Kerala, the highest literacy rates in India are found to have large crimes on women. So this has also been found. So this is not sufficient, but this is of course one important fact, that is education, spread of education and women empowerment, that can help to reduce this gender discrimination, which will hamper the economic development, of course, and also the sustainable development of any economy. Thank you. Thank you.
Thank you for a nice presentation, ma'am.
Thank you very much.
We now move on. We now move on to the next speaker, that is Dr. Kamalika Chakraborty, Assistant Professor, Khatra Adivasi Mahavidyalaya, and she would be speaking on green economy. Madam,
Am I
please,
audible,
stop
ma'am?
sharing your screen.
Yes, you are
Yes,
audible.
ma'am. You are audible.
Okay.
Please share your screen, Kamalika. It is visible. It is visible perfectly.
Is my screen visible?
Yes, it is visible.
Yes.
Okay. So good afternoon, everyone. Myself, Dr. Kamalika Chakraborty, Assistant Professor of Economics, Khatra Adivasi Mahavidyalaya, affiliated to Bankura University. So today I'm going to speak on green economy. Now the United Nations Conference on Sustainable Development, which was held in Rio de Janeiro in 2012, promoted sustainable development to an internationally accepted concept. And this conference resolution pointed out that green economy is one of the tools to achieve sustainable development. So a green economy is one which ensures economic growth and development through investment in such economic activities that contribute towards less carbon emission and reduced pollution. And a green economy will also be one which will use the resources in the most efficient manner and will also be socially inclusive. So 5 principles on which green economy is based are: firstly, the well-being principle. So this principle says that green economy will help people to create as well as to enjoy prosperity. Next is the justice principle. According to this principle, green economy promotes equity not only among a generation but also across generations or between generations. Third, the planetary boundaries principle. According to this principle, the green economy safeguards, restores, as well as invests in nature. Fourth is the efficiency and sufficiency principle. According to this principle, green economy will support sustainable production and sustainable consumption. And the fifth is the good governance principle, according to which the green economy will be guided by an integrated, accountable, and resilient institution. So another common term related to this is green growth. Green growth means encouraging economic growth and development while ensuring that natural resources are not exploited indiscriminately. So green growth is actually not a substitute for sustainable development. The focus of green growth strategies will be to ensure that natural assets can be used in the most efficient manner so that they can deliver their full potential on a sustainable basis. Green growth helps to achieve sustainable development in the following ways. Firstly, by enhancing productivity, and productivity can be enhanced by using the resources in the most efficient manner and avoiding any kind of wastage. Also by promoting innovations and value creations and allocating resources in the most efficient manner so that they can yield the highest value use. Secondly, green growth can help to achieve sustainable development by elevating the confidence of the investors. This can be done through greater predictability at how governments deal with major environmental issues. Thirdly, green growth can also help to achieve sustainable development by launching new markets. That is, this can be done by stimulating demand for green goods, services, as well as technologies. Fourthly, fiscal consolidation can also be a solution. This is done by mobilizing revenues through green taxes and also through the elimination of environmentally harmful subsidies. And fifthly, green growth can help to achieve sustainable development by decreasing risks of adverse impact on growth. Here, by adverse impact on growth, we mean resource bottlenecks which may arise, as well as damaging and potentially irreversible environmental impacts. What are the ways to overcome the challenges to green economy? Firstly, implementing green growth strategies in a way that eliminates poverty, at the same time promotes employment opportunities and contributes towards a strong and sustainable economy. Secondly, better infrastructure should be provided to people. Thirdly, those health issues which arise due to environmental degradation should be addressed properly. Fourthly, productivity should be increased and costs should be reduced, but at the same time, it should be kept in mind that environment needs to be protected as well. And last but not the least, the strategies for green growth should be country specific. So there is no one solution for all countries. So the strategies for green growth should be taken in such a way that it meets the demand of the particular country for which the strategies are being devised. So thank you. I would like to thank the organizers for giving me the opportunity to present my paper. Thank you.
Thank you, ma'am, for the brief and crisp presentation. I would like to ask you what is this fiscal consolidation on behalf of the audience?
Fiscal consolidation is actually, ma'am, the two aspects. Fiscal policy has two aspects. One is the collection of revenue and another is the imposition of tax. So in order to ensure protection of the environment, we can try to devise such tax and subsidy policies which will eliminate those subsidies which may be environmentally harmful and tax on those areas which may promote the sustainable development aspect.
Thank you so much, ma'am, for shedding light on such a relevant topic that is currently gaining traction. This is Rajarshi. May I ask you something, ma'am?
Yes, sure, sir.
Yeah, ma'am. I was reminded by a certain concept which is called intergenerational justice. Listening to your talk. So, can I have your comment on that? The green economy.
Intergenerational justice, you are saying, sir?
Yeah, ma'am.
Actually, the entire concept of sustainable development is that we should meet the needs of the present generation. At the same time, keep in mind that our future generations should also be able to enjoy all the facilities which we are enjoying. So from that point itself, this intergenerational concept is coming, that we are exploiting the natural resources to meet our needs, but sometimes not keeping in mind that our future generations are being deprived, may be deprived of those resources. So this intergenerational justice implies that justice must be made not only to the present generation, but also the future generation. I hope I am able to convey my thought.
Absolutely, Kamalika. Thank you so much for your lucid reply.
Thank you.
I think it will help us all. Thank you so much, ma'am.
Thank you once again, ma'am. I would now welcome Dr. Poulumi Adhikari, Assistant Professor, Narajol Raj College, and she would be speaking on diversity of vascular plants on concrete houses and historical buildings in and around West Bengal, West Midnapur. Ma'am, you have 10 minutes. You may now share your screen.
Is my screen visible?
Not yet. Yes, it's visible.
Is it visible in full screen now? Am I audible?
Yes, ma'am.
Yes, ma'am, you're audible.
And is my screen visible in full screen mode?
Yes, it's visible.
Okay. Thank you. A very good afternoon, everybody. We have all seen the very many types of vascular plants, ponds, climbing plants, and trees, and also the very many types of angiosperms growing on the roofs and walls of old historical buildings and concrete houses in both rural and urban areas of West Bengal. So today, I, Dr. Poulumi Adhikari Mukherjee, Assistant Professor, Department of Zoology, Narajol Raj College, Vidyasagar University, am going to present before you on the topic entitled Diversity of Vascular Plants on Concrete Houses and Historical Buildings in and around West Midnapur, West Bengal, India. To begin with, the old historical buildings and concrete houses in West Midnapur district are the foundations of the community's most sustainable form of development. And the unique character of each of these heritage buildings in West Midnapur effectively embodies much of the essence of the economic, cultural, social, as well as environmental sustainability. However, negative impacts on environment through unplanned development may weaken sustainability as a whole. Therefore, assessment of environmental conditions is very much necessary for initiation of any sustainable development. To achieve this goal, identification of the major positive aspects, its optimum utilization and monitoring is necessary. The symbiotic relationship between local ecology, social structure, and economic conditions of any habitat should thus be taken into account. As mentioned before, that vegetation in cities includes a notable spontaneous component that grows voluntarily without human assistance on adventive or rural habitats. They occur on a wide range of site conditions from remnant natural to semi-natural and artificial along the heterogeneous scale. Such vegetation often embodies natural elements with ecological value in built-up areas in terms of plant species composition accompanying wildlife and ecosystem processes. Particularly, the walls and roofs of concrete houses and buildings are familiar and conspicuous structures, yet they are given very little attention from a biological perspective. And this is very unfortunate as they also support a great diversity of flora and fauna as well. The walls particularly provide demanding conditions for plants because of their natural calamities like exposure, drought, and anthropogenic activities. Deposition of dusts, organic substances, suspended particles of air and atmospheric moisture are responsible for the formation of thin film of soil substratum on the walls and wall fissures, leading to the development of a specialized flora. And this man-made habitat gradually influences on a wide range of plant species to colonize on it. Though the man-made walls represent extreme environments with edaphic factors such as low availability of water and nutrients and exposure to wind and sun, still some plants show the capacity to grow over the walls. The presence of climbing or creeping plants, large plants or shrubs growing on the walls of the houses and buildings drives a bullet-headed root into crumbling masonry, thus causing damage and disintegration of the material, which, if forcibly removed, can pull off a weak surface of brickwork or plaster, thus causing movement of the foundations and subsequent cracking of walls and roofs. The data concerning the diversity of floral changes are scarce and fragmentary, yet there are only a few specific studies on the floral species growing on the walls and roofs of concrete houses and buildings. Hence, keeping this in mind, my aims and objectives of my study was conducted on wall and roof flora. First of all, to identify the different vascular plants that are growing in the crevices of the roofs and walls of the historical buildings and concrete houses in and around West Midnapur district, to understand the rural environment and floral diversity of old heritage houses scattered in the district, and to explore their effects on the walls and roofs of those buildings. Finally, also to investigate the floral diversity on those sites undergoing reconstruction, restoration, and maintenance activities during this period of study. Our results will help to establish the regularities and trends concerning the origin and dynamics of the roof and wall flora at the studied sites, as well as to devise some techniques to remove this nuisance in terms of property damage, thus maintaining the historical buildings and old heritage houses of the concerned areas. So the methodology which was applied, that the houses and buildings were randomly selected in the district and the various plant species going on them were sampled and examined. A total of 150 houses and 10 historical buildings, including the different Rajbari of located in West Midnapur, were surveyed throughout the district from a period of November 2019 to October 2021. And out of them, 60 houses were marked as new, which were less than 50 years of age, and the rest were marked as old, which were older than 50 years of age. Furthermore, a questionnaire was conducted among the inhabitants to gain knowledge about whether they eradicate the unwanted plants growing on their concrete houses and buildings or not, and if the residents eradicate the weeds, whether they do it by self manually or by hiring labor, and if labor is hired for cleaning, then how much is cost for such cleaning purposes. Also, plant abundance was determined by calculating the average number of vascular plants recorded, and one-way ANOVA was carried out to determine whether there is any significant difference of plant abundance in between all the sites, and there is any difference of plant abundance in new as well as old group of houses and buildings. So the results which we got was that in vascular flora, we got one species of pteridophyte, but while the rest of the plant species were angiosperms. The most dominant plant species were identified from the Amaranthacea, Asteraceae, Moraceae, Poaceae, and Fabaceae families. But we did not find any significant variation of plant abundance along the sides or in between old and new group of houses. These are the list of the different vascular plants which we found on the historical buildings and concrete houses in West Midnapur, both in old and new and both as well. And Here are some of the photographs of the most abundantly occurring vascular plant species, both in the historical buildings and concrete houses in and around West Midnapur. And these are the photographs of the least abundantly occurring vascular plant species on the buildings and concrete houses in West Midnapur. So from the survey of the residents, it was found that 67.67% correspondence in new houses clean by hiring labor, and 33.33% of them do not clean at all. However, in cases of old houses and buildings, we got 3 cases. They clean the plants either manually or by hiring labor, or they do not clean them at all. And the average cost of cleaning every time and the average annual cost of cleaning are also not significantly different in both between these two groups of houses, that is new and old groups. So this is the differential maintenance which we got from
Sorry to interrupt you, ma'am. You need to wrap up now.
Yeah, I'm just coming on to the conclusion. The growing on the concrete grown plants, both in the new and old houses in West Midnapur, and these are the data which we got. So coming on to the conclusion, the negative interaction which is occurring between the vascular plants growing on concrete houses and humans in rural as well as urban areas is a least explored area. And we should throw light on this topic, on this critical issue. Our study shows that the most abundant plant species identified among the vascular flora on both concrete houses and historical buildings were primarily angiosperms, and they inserted their roots, resulting in cracks, ultimately damaging and harming the materials and also the binding capacity of bricks and cement. Not only this, the root remains acted as substrate for the microbial activity, and these also caused harm to the building materials. Thus, it can be concluded that they are not only responsible for the biodeterioration of walls and roofs, but they are also the slow poison for building life. Our study also shows that the concrete grown plant community is more or less similar in both new and old group of houses in the district, but their way of approach of riding the menace is a little bit different. On one hand, the residents clean the concrete weeds of older houses, while the owners of the new houses mainly clean them by hiring professional laborers rather than manual one, and also the data is not significantly different. Therefore, our present study helps to identify the problems faced by the inhabitants and also tries to give an insight that how it can affect the city dwellers financially. However, a detailed and extensive investigation is due on this issue. Thus, in conclusion, it can be said that the interaction between vascular plants and human engineered habitat is everlasting, and it is not a very easy task to get rid of it completely, and it needs immediate attention and management as well. We hope our current study, delivering detailed information regarding the diversity of vascular plants, will certainly draw attention of the scientists to emphasize on further research work on this burning issue, to implement some control measures to eliminate the nuisance and provide a successful approach to ensure a more sustainable development in older heritage buildings and houses. These are some of the references which I have consulted for this paper and study work, and I am highly grateful to Bongamoti Hasta, Assistant Professor of Botany Department of Narachal Raj College, for identifying the plant species. I'm also thankful to my students of Zoology Department of Narachal Raj College for helping me in taking the photographs of the vascular plant species growing on the houses and buildings. And finally, I wish to thank all the authors of those literatures we have consulted for preparing this paper. Thank you all for your kind attention.
Thank you, ma'am, for such an interesting presentation.
Thank you.
Our next speaker is Professor Ruchi Sharma, and she is from Jagran Lake City University, and her topic is focusing on community engagement for ensuring sustainable development. We are glad to have you here, ma'am.
A very good afternoon to each and everyone present here. I'm Dr. Ruchi Sharma, and I'm going to present my presentation. I'm sharing the slide now. Kindly let me see whether it is visible or not. Kindly let me know if it is visible. The slides are visible.
Yes, ma'am, it is
Yes,
visible.
ma'am.
It is visible. So
Yes,
I
ma'am.
will be doing it on slideshow. In case if it gets stuck in between, kindly let me know. Okay, thank you so much. So when we are talking about the sustainable development, the topic which I have chosen to present paper on is focusing on community engagement for ensuring sustainable development. The objectives, is my second slide visible?
No, ma'am. Ma'am, you have to full screen your your presentation.
Pardon?
You need to full screen your presentation and
Okay,
please
okay,
share your screen, ma'am. Your screen is not shared now.
okay. Is it visible now?
Yes, ma'am, it is visible. Make it full screen mode.
Now, I hope it is visible.
Yes, 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 it is visible.
Okay, thank you so much. So the objectives of my research is to understand as how the community can be engaged to accomplish the goal of the sustainable development. The research methodology which is being used is I have used the secondary data to come up with a particular proposed model which I will be sharing with you all. When we are talking about the development, many a times it has been seen or being looked forward for the economic development, but the true meaning of development is not just only limited to economic, because many a times in order to fulfill the greed or the economic targets, the unplanned use of natural resources, they are being done, and as a result it has got the negative impact on our environment as well as on our society. So unsustainable development is a major threat which is being faced by our society, and as being rightly mentioned by Dr. Amartya Sen, economic growth without investment in human development is unsustainable and unethical. We cannot say that we are a developed society just only on the basis of the economic development. The human society is being exposed to various vulnerabilities, and now as we are seeing the present crisis which we are facing nowadays is COVID-19 pandemic, and coming with the new variants, new waves which are there. So it has affected our sustainable development adversely. So we can, at times it feels like that we, the human beings are in a dark tunnel where there is no light at the end, and how to overcome this particular, to cross this tunnel. So for this, the sustainable development is the answer. And for the first time, the sustainable development term, it appeared in UNEP and IUCN in 1980. The most commonly definition which is being used is given by Bertland Commission report, which says that sustainable development is the development that meets the need of the present without compromising the ability of the future generations to meet their own needs. There are the various dimensions, basically focusing on the 3 dimensions which was highlighted by Kahn in his research in 1995: economic sustainability, environmental sustainability, and social sustainability. So keeping these 3 dimensions only, I'm proposing my model in my research paper. In order to promote the sustainable development, a well-planned, resilient, community-inclusive framework is needed. And for the effective implementation of it, the strategies need to be identified. And for this, the strategy identification phase, especially in the present scenario, we have to look into the COVID-19 crisis, where we are focusing more on health improvement as well as economic recovery. Because of the lockdown, the economic, there was, a setback in the economic development. But at the same time, we need to be very careful in our planning approach, where in order to scale up of economic activities, we should not go for the unsustainable development, because it will be going to have an adverse impact on our society as well as on our environment. So keeping this all in mind, the research paper, it focuses on proposing a model which is named as PRIME. The model PRIME focuses on understanding the importance of community engagement in dealing the problems of unsustainable development. And it facilitates empowering local communities to play active role in the achievement of the sustainable development goals. And the implementation of the model, it will be going to help us in achieving in enabling the capacity of local community for ensuring the sustainable development, so that after we are in December 2021, as we are moving through the tunnel which I reflected earlier on, that is a dark tunnel. So when we move into 2022, at least some bright sunshine is there based on our approach. The proposed model in the research paper highlights that in order to cope with the present crisis and to promote sustainability, we require social change in the society, and which talks about the model where the P stands for participation, R stands for resource mobilization, I for inclusion, M for monitoring, and E for empowerment. So it's a cyclical process, being as in the model you can see there is a cycle where we are talking about the different stages and one supporting the other. So the first P, which stands for participation. Here we are talking about to strengthen the capacities of the individuals and communities to mobilize and help themselves. In this way, dependence on the government is minimized, and the ordinary people, that is the common people, will be going to rediscover their potential for cooperation and mutual endeavor. It focuses on bottom-up planning system. This bottom-up planning system, where local level plans would initially be formulated by the people, those who will be going to have participation right from the planning stage to decision making until the implementation stage. Next is R, which stands for resource mobilization. It refers to all the activities which are involved in securing new and additional resources for sustainable development. It also involves making better use of and maximizing existing resources which are available. A lot can be achieved when people from different parts of the community share a common goal and actively participate in both identifying needs and being part of the solution as well. It uses deliberate participatory process to involve local institutions, local leaders, community groups, and members of the community to organize for collective action towards a common purpose. It is characterized by respect for the community and its needs. So one stage, that is participation, is promoting resource mobilization. From resource mobilization, we are moving ahead to the next, which is I, which stands for inclusion. Here, I does not stand for just one individual, but here we are talking about we, I involving the we concept, including each and every community member. So we are talking about the inclusive society that looks forward to respect for human rights, cultural diversity, and democratic governance, and upholds the principles of equality as well as equity. As a process, it enables all the citizens' participation in all the activities that affect their lives directly or indirectly. Moving ahead, next is M, which stands for monitoring and evaluation. It is a process through which whatever we have planned, the planning is there and how it is being moving ahead to reach the sustainable development goals. It needs to be assessed to understand if we are lacking somewhere in between, so that accordingly the correction can be done then and there, rather than finally going at the evaluation stage and looking back and reflecting that we have lost major time in just implementing the projects which were not able, or which cannot be related to the development or goals at all. So when we are talking about the monitoring and evaluation, it acts as an accountability tool for most social impact projects as well as interventions. Now, all these combining together, participation, resource mobilization, inclusion, monitoring, it automatically empowers the community, where we are talking about the community engagement. So it refers to the process of enabling communities to increase control over their lives. It implies community ownership and action that explicitly aims at social and political change. Community empowerment is a process of renegotiating power in order to gain more control. And again, more empowerment, the moment the people, the community feels that it is empowered, then they will be going to move ahead to the participation. They will be going to, it is a cycle. They will feel more connected. They will feel more participative to take initiative in the achieving the sustainable goals. So to conclude, we need to be conscious of the social developmental agenda because the awareness is very necessary. The development pathway of society is marked by community engagement. Therefore, it is important to be aware about the opportunities as well as challenges so as to leverage the opportunities and counter the challenges to achieve sustainable development goals and enhance the efforts to build resilient sustainability. So at last, the key suggestion, which was based on the proposed model, it exhibits the importance of the recovery process to be resilient, and also we should not focus on achieving prosperity at the cost of exposing society to vulnerabilities. And here in this picture, we are coming out of that tunnel, hopefully in 2022, moving ahead to the target of achieving the sustainable development goals. Thank you so much.
Thank you so much, ma'am, for your wonderful deliberation, which was very much in line with the theme of our conference. It was very good to hear from you. Thank you again. The next speaker we have with us is Pawan Kulkarni, who is an academic coordinator, GIBS Business School, Bangalore, Karnataka, India. He would be speaking on an analytical study on industrialization, innovation, and infrastructure for achieving SDGs in India. Yes, sir. Are you there?
Can you hear me, ma'am?
Yes, yes. Yes, you can now present your screen. You are having 10 minutes.
Yes, ma'am. Sure. One second. Ma'am, can you see the screen? You're able to see the screen?
Yes, sir. It's visible.
Yeah. Good afternoon, all. It gives me immense pleasure and privilege to present an analytical study on industrialization, innovation, and infrastructure for achieving sustainable development goals in India. It is completely related to Sustainable Development Goal 9. My co-authors are Ashrit Bharadwaj, who is MArch second year, Department of Art, Design and Architecture, University of New South Wales, Sydney, Australia, and Dr. S. Evanarani, Madam, Department of Decision Sciences, Jain University, CMS Business School, Bengaluru, Karnataka, India. Before I start my presentation, I would like to deliberate presentation that regarding the inclusive and sustainable industrialization, together with can unleash the dynamic and competitive economic forces that generate employment and income. They play a key role in introducing the promoting new technologies, facilitating the international trade, and enabling the efficient use of resources. However, the world still had a long way to go to fully tap this potential. Least developed countries in particularly need to accelerate the development, particularly to development fully tap this potential, and in particular need to accelerate the development of their manufacturing sector if they are to meet the 2030 target and scale up the investment in scientific research and innovation, especially in our country. Introduction. The Sustainable Development Goals, as we know, it encompasses all the key development sectors, including education, health, sanitation, employment, infrastructure, energy, and environment, and set time-bound targets to achieve them. From a global perspective and in its own cause, the need for India to achieve these targets remain imperative. Significant progress has already been made across the country in recent years. The tempo of the progress must be modulated in an evidence to backed manner of the Sustainable Development Goals. And to understand the objectives of the study is to understand the Sustainable Development Goal 9. Goal promotes investment in innovation and reliable and resilient infrastructure, which are crucial drivers of the economic growth and development. And to analyze the aims at promoting increased resources using efficiency and greater adoption of clean and environmentally sound technologies and industrial processes. And the another objective is to recognize the importance of promoting sustainable industries and investing in scientific research and innovation, which are all important ways to facilitate the sustainable development.
Sir, we lost your voice. We can no more see your screen, sir.
Ma'am, 1 second. I think some technical issues are there. 1 second, ma'am. I will come back. Ma'am, now can you see the screen, ma'am?
Yes, sir. Now it's
Yeah,
visible.
I'm
Now
really
it's
sorry,
visible.
ma'am. There was
We,
some
we
technical
are running
issue.
short of time, sir. You have
Okay,
8
okay.
minutes.
Yes, ma'am. There are different targets which are set by the Sustainable Development Goals by the United Nations Organization and UNDP to develop the quality, reliable, and sustainable and resilient infrastructure, including regional and trans-border infrastructure to support economic development and human well-being with a focus on affordable and equitable access for all. And another important target is to promote inclusive and sustainable development, industrialization, and by 2030 significantly raise industry's share of employment and gross domestic product in line with national circumstances and double its share in least developed countries, and increased access of small scale industries and other enterprises, in particular in developing countries, to finance services, including affordable credit and their integration into value chains and markets. And by 2030, the target is to achieve the upgrade infrastructure and retrofit the industries to make them sustainable with increased resource use efficiency and greater adoption of clean and environmentally sound technologies and industrial with the perspective capabilities. Because of shortage of time, I will be directly coming to the findings of my study. And before findings, there is something called as Sustainable Development Index score every year that will be released by Niti Aayog of Government of India for Goal 9 and all other goals of Sustainable Development Goals range between 24 and 72 for states, between 23 and 66 for union territories, and Gujarat and Delhi are the top performance among the states and the union territories respectively. 6 states and 1 union territory bagged a position in the category of front runners. Score range between 65 and 99. Front runners, the states as per 2021, Statistics is Gujarat, Tamil Nadu, Punjab, Goa, Haryana, Maharashtra, and Delhi. Performers who are between 50 to 60 crores. It's out of 100. Karnataka, Himachal Pradesh, Kerala, Telangana, Uttarakhand, West Bengal, Andhra Pradesh, Sikkim, and Puducherry. And these all are the aspirants which are need to upgrade themselves and they have to perform even more properly. And when it comes to India achieving the sustainable development goals, especially when it comes to sustainable development goal 9, industry, infrastructure, and innovation, it has improved over the period of time. In the year 2018, we were ranked as 57. In 2019, 52nd, and 2020, 48th rank. India ranked among 131 countries in the Global Innovation Index 2020 by WIPO. And when it comes to 98% targeted habitations covered with all weathers road under PMGSY, 16.1% of total gross value added in from manufacturing sector, 12.07% total workforce employed in the manufacturing sector, 84 have mobile connection for every 100% in India as per 2020-21 statistics, and 55 have internet subscription for every 100%. This is the current situation of India in achieving sustainable development goal 9. And when it comes to findings of the study, when we analyze the sustainable development goals, especially sustainable development goal 9, the first important thing comes is infrastructure, how we are going to improve the infrastructure and what is the current situation of the infrastructure, especially when it comes to road connectivity. Good connectivity is fundamental requirement for industrialization development, as we all know. When it comes to the Pradhan Mantri Gram Sadak Yojana was launched by the government of India to provide connectivity to unconnected habitations through all weather roads. At the national level, close to 98% of targeted habitations have been covered as per the statistics given by the Niti Aayog. 7 states, namely Goa, Gujarat, Haryana, Karnataka, Punjab, Rajasthan, and Tamil Nadu, have achieved the target of providing 100% connectivity under PMGSY. Meghalaya has recorded the lowest performance since only 381 out of 603 targeted habitations. See, when it comes to the manufacturing, is very important for any country. If any country wants to grow and to be listed in the developed countries of the world, the manufacturing and the industry growth is very important. The value addition by manufacturing sector of the total gross value added in India in 2018-19, the share of manufacturing sector was 16.1%. The target is to increase this to 25%. 5 states, namely Goa, Gujarat, Himachal Pradesh, Sikkim, and Uttarakhand, 1 union territory, Puducherry, have already achieved the target. And employment in manufacturing sector is also very important. When I was studying and with my team, we were able to make out, as per the periodic labor force survey, 12.07% of India's total workforce was engaged in manufacturing sector in 2018-19. Among the states, highest share was in Gujarat, 20.8%. 3 states, 1 state, namely Sikkim, Meghalaya, Arunachal Pradesh, Lakshadweep, had less than 5% of their labor force employed in the manufacturing sector during this period.
Yes, sir, you may continue. Hello?
Yes, ma'am.
Hello?
Yes, ma'am. 100%. And when it comes to innovation score, innovation is very important in the current business world and competitive world. Why it is so important when we ask the industrial growth, the science and technological growth should happen with the innovation. Innovation is important because we famously say that today the inventions, what we do tomorrow, it will become a mud pot. That is the competition level what we have in our country and across. So India has been ranked 48 out of 131 countries. It's a very good ranking by the World Intellectual Property Organization in the Global Innovation Index 2020. With a score of 35.59 out of 100, state union territory scores have been sourced from India Innovation Index from 2020. Among the major states, Karnataka ranked first with a score of 42.5%, followed by Maharashtra and Tamil Nadu. Himachal Pradesh topped among the northeast hilly states with score of 25.06 and followed by other states like Uttarakhand, Manipur, Meghalaya. Delhi topped among union territories. And when it comes to logistics, the logistics is very important for any country's growth because the World's Bank's Logistic Performance Index in 2018 ranked India as 44 out of 160 countries with a score of 3.18 and have been source logistic ease across different states leads. 2019 report released by Ministry of Commerce and Industry, Gujarat with a score of 3.62, the best performing state, while Nagaland has scored only 2.28. Chandigarh leads in union territories. So when it comes to internet density and mobile tele density, see the presently for every 100 people in country, 84% have mobile connection, while only 55% have internet subscription. Notably, the mobile density is over 100% in 11 states like Goa, Haryana, Himachal Pradesh, Karnataka, Kerala, Maharashtra, Mizoram, Punjab, Tamil Nadu, Telangana and Uttarakhand, and 4 UTs Chandigarh, Delhi, Ladakh and Lakshadweep. With 200 internet subscription for every 100%, the Delhi tops the country in the internet subscription density. Among the states, internet access is highest in Punjab, 84.32% subscription per 100%. Bihar has the lowest number of mobile connections and lowest internet subscription. And however, the global manufacturing growth has been steadily declining even before the outbreak of the COVID-19 pandemic. The pandemic is hard and causing disruptions in the global value chains and the supply of products. The innovation and technological progress are the key to finding lasting solutions for the both economic and environmental challenges what our country is facing, such as increased resources and energy efficiency. Global research and development as a proportion of GDP increased from 1.5% in 2000 to 1.7% in 2015 and remained almost unchanged in 2017, but was only less than 1% in developing regions. So in terms of communication infrastructure, more than half of the world's population is now online, and almost the entire world's population lives in the areas covered by mobile network, and it is estimated that is 2019. And it is very important to see, in conclusion, I would like to tell the sustainable development goals is need to be achieved properly by the end of 2030. The coronavirus pandemic has revealed the urgent. The Asian Development Bank notes as the critical infrastructure in the region remains far from adequate in many countries. Despite the rapid economic growth and development the region has experienced over the past decade. So the highlights make the infrastructure in India resilient to the disasters and climate change will require an additional investment in our country. This sum may need to even greater in some sub-regions such as the Pacific, small island and developing states. So in the conclusion, I can tell when it comes to India, India is performing very well. As we have seen the rankings, the statistics, the conclusion I can say that India is performing very well. In order to top, it is very important. By 2015, agreements signed by all the countries across the world. By 2030, they all have to achieve, including our country. In our country, the Niti Ayoga is taking care of that. And the important thing is they have to develop the quality, reliable and sustainable and resilient infrastructure, including the green values in the system, especially in the corporate sector. It may be education institutions. You take up any organization setup, it may be a small scale industry, sole trading concern, partnership concern, Hindu undivided family, joint stock companies, private companies, public companies, any other companies, a small scale industry to the large scale industry, everybody should concentrate upon the sustainable development goals, especially the green economy. They should concentrate green infrastructure, and above that, the green energy should be used in order not to harm the environment and country. With a focus on affordable and equitable access, equality is very important. As we speak on this many times, but achieving the equity is not a simple deal for any country. But when it comes to our country, all the state governments, as we have the federal structure in our country, state government and central government, unifiedly they are making the constructive efforts that there will be affordable and equitable access for all the facilities should be given to our people. And by 2030, we can say that we should upgrade our infrastructure and retrofit industries to make them sustainable with increased resource use of efficiency and greater adoption of clean and environmentally sound technologies and industrial processes, with all countries taking action in accordance with this respect. So the country, especially the India, is already making a constructive and a very positive efforts in order to achieve all the sustainable development goals. But when it comes to sustainable development goal 9, right from the liberalization, privatization, globalization which has happened in the year of 1991, from that the country has developed enormously, and the country has given the notable contributions towards the world development and achieving the sustainable development goal. If the country goes in the same pace, taking all the states together, because few states are doing very well, few states are in medium range, few states are not performing well. So in considering the states which are not performing well, they should also come in the first lap. So in order to achieve that first lap, they should be a top performers. So the state government and central government both should make a constructive effort and industrial development. And equitable distribution of the population can only happen by the industrial growth of each and every district of that state. For example, there are many drawbacks. When it comes to Karnataka, you can take all the industries, especially IT industries, are completely concentrated in Bengaluru. There is no equitable distribution of IT industries. Even the government is not suggesting for the companies to set up wherever they want. But the problem is the equitable distribution. But in the same time, if we take example of Tamil Nadu, even there every district is doing very well because every district have their own advantages, and small scale industries, medium scale industries established in Tamil Nadu. So these all things has need to be achieved, and West Bengal is doing very well when it comes to, you take recent statistics from past 5 years, it has been enormously grown. It may be West Bengal, it may be Gujarat. Gujarat is a top performer, and all other states are doing very well. But the important suggestion is this: the equitable distribution of the employees and the people person can only happen when there is equitable distribution of the industries across the India. And we have Make in India, Startup, Stand Up India. There are many ways of doing business, many initiatives taken by the state government and central government in order to uplift the infrastructure, industry, and innovation. And the important thing is we should encourage the youths of this country, which is very important. Because many people say when the unemployment is there, in the general statement, today's youths are useless. I say they are
Hello.
not useless, they are used
Apologies for
less.
the interruption.
Should
Excuse
be
me, sir.
taken care.
Apologies for the interruption. Could you please wrap it up your presentation within a few minutes, please?
Sure, sir.
Yeah.
When
Please
it
go
comes
ahead.
to youths of this country, they are playing a very important role when it comes to industrial output, infrastructure, and innovation. Because when it comes to industry, the setup of the industry, the startups should start in our country. Then only the industrial development can happen. And when it comes to infrastructure, infrastructure can be built by the constant effort of the state government, central government, and whom they are giving the contract and who is going to execute it, and they are getting the employment of the same. Innovations can be done only by the youngsters. They should promote, because young people of this country should be used properly in order to ensure the sustainable development goal 9 will be achieved the maximum. So in the end, to conclude, I can tell that sustainable development goals, whatever the statistics we have elicited from the different reports of the government of India and across world, how every country is performing, the parameters fixed by the country. By 2030, the sustainable achievement of sustainable development goals it is not
Hello.
a natural choice. It is the only
Hello, Pawan
choice
sir.
for the
Hello.
entire world. Yes, sir. Thank you, sir.
Yes, sir. You have to close your slide right now.
Yes,
We
yes.
cannot
Yeah,
proceed with you.
I am closing, sir. sustainable development goal, achievement of sustainable
No, sir.
development.
Sir, excuse me,
goals
sir.
outcome
You have to stop your
is the only
presentation. You have to
choice.
stop your presentation and close your
Thank
slide.
you. Thank you all.
Yeah.
Thank you.
Thank you again, sir, for your relevant presentation. The last speaker of today's session, first half session, is Dr. Tarun Ghosh. He is an assistant professor, Agu Kamini Chandra Prakash Mahavidyalaya, and the topic he is going to present today is advanced measurement technologies and new concepts to support sustainable development. Sir, can you hear me?
Yes, ma'am. Good noon
You
to
may now
all.
present, you may now share your screen. You have very less time. You have to wrap it up within around 8 minutes.
Visible, ma'am? Hello? 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 Am I audible? Hello?
Yes, sir.
My screen is visible now?
Yes, sir.
Okay, thank you. Myself, Dr. Tarun Ghosh, Assistant Professor, AKPC Mahavidyalaya, Bengai, Hooghly. My today's topic is advanced measurement technologies and new concepts to support sustainable development. Pollution and environmental hazards are global problems now and pose a big challenge to the environmentalists. Accordingly, the necessity of the moment includes controlling global warming or the mean temperature of the globe and its changing trend, ensuring potable water as well as water for agricultural purposes and domestic use, making available of the clean energy for industrial and domestic use, developing eco-friendly or green technologies. Moving towards this goal involves considerable cost which poor countries are not in a position to afford. To fight it back, an overall green economic policy taking care of the inabilities of the poor nations is required to be operational globally. They will maximize benefits in terms of environment protection and fulfillment of human needs for the money invested on the development of the basic needs and desirable amenities. To be able to act in right direction, we must have enough data encompassing the entire globe, the reality on the ground as well as on the upper atmosphere. It requires adoption of the advanced technologies like use of satellites to have the data like content of ozone, oxides of nitrogen, sulfur dioxides, carbon dioxide, etc. in the atmosphere in local areas throughout the globe at different all altitudes and therefore the integrated result over the globe. Improvements and further developments are on in this regard. We also need to appreciate new concepts related to sustainable development. The concept of virtual water is a new development and likely to lead to far-reaching consequence in
Your
the
screen is
long.
not visible, sir.
Yes,
Excuse me,
ma'am.
sir. You lost your screen. Your screen is not visible, sir. It got all black.
Now visible? Hello.
No, no, not visible yet. It isn't visible.
Visible now? Hello.
No, sir, not visible. Yes, it's visible now.
Visible now?
Yes, yes, yes.
Just a minute. Visible, ma'am? Hello? Hello? Hello?
Yes, yes, sir, you may continue.
Visible now?
Yes, sir, it's visible. You may continue.
And improvements and further developments are on in this appreciate new concepts related to sustainable development. The concept of virtual water is a new development and likely to lead to far-reaching consequences in the long run in our lifestyle and the pattern of food supply chain. Okay, thanks for waiting. Thank you, ma'am. Hello. Hello. Hello.
Yes, sir. Hello.
Yes,
Hello.
sir. My lecture is complete. Hello.
Yes, sir. Sir, your screen is not
Hello.
visible. Yes, sir. Your screen is not visible. You are audible, but your screen is not visible. It is completely dark.
Okay, sir. My lecture is complete. Completed.
Okay, sir. Thank you, sir.
And
So,
sir, I have a request. Sir, I have a
yes.
request, please. Sir, I participated in the previous eConsus 2020,
All
and
right.
my work has been published in the. But
Yes, sir.
till now I have
Yes, sir.
not. Till now I have not got the hard copy of the book. So I have sent mails 2 times. So
All
I
right, sir.
request
Sir,
you.
yes, sir. So you will be receiving the hard copy of the conference proceedings of the last year very soon. I will escalate this matter to the concerned people. So here we will like to end our session for this, and we will again meet at 2:10 p.m. sharp for the final session of the conference. So we will have a lunch break at now, and we will again meet at 2:10 p.m. sharp for the final session. Thank you all.
Good afternoon, everyone. Am I audible clearly?
Yes, Manoj, please go ahead.
Yes, sir.
Thank you. Myself, Dr. Manoj Pal, Assistant Professor, the Department of Geography, Banwarilal Bhalotia College, Asansol. It is my great privilege to welcome you all once again in the eConsus 2021, Revisiting Strategies for Sustainable Development, organized by our college. We are fortunate enough with the honorable presence of our keynote speaker, Professor Shobosachi Bhattacharya, and Dr. Atin Odhikari, and invited speaker, Dr. Pitha Bhattacharya. Now, I would like to take the opportunity to introduce our first keynote speaker, Professor Shobosachi Bhattacharya. Professor Bhattacharya is a renowned professor of Agricultural and Ecological Research Unit, Indian Statistical Institute, Kolkata, West Bengal. He had completed his MSc degree in statistics from the University of Calcutta in the year 1998, and he was awarded PhD degree from Jadavpur University in 2004. He was postdoctoral fellow at the Iowa State University, USA from 2004 to 2005. He received the gold medal in Global Research and Design for the Best Innovative Research in 2009 as a researcher in General Motors India Research and Design Lab. His scientific career has been specialized in applications of statistics and mathematics in ecology, epidemiology, and agriculture. Now, please may I request Professor Bhattacharya to share your views towards sustainable development with us. Over to you, sir.
Yeah, so am I audible?
Yes, sir.
Yes,
Okay.
you are clearly audible.
Okay, so thank you, Manoj, for inviting me in this opportunity to share some of my research thoughts. And as I understood from your team that there are a couple of students in this workshop or in this venue or in this online platform. And also there are some faculty members, those who are actually participating in this online meeting. Actually it's a mixed audience so far I understood. And most of them actually have that biology, microbiology, and sociology, like this type of structures. And some of them have mathematics and statistics background, but the number is very few. So it's difficult to plan the talk because its time is only half an hour. And I put my title as escalating role of statistics and oblique maybe mathematics. So for that in ecological framework. So let us share first my slide and then we'll discuss. Okay, I hope so. My slides are visible.
Yes, sir.
Okay, so thank you all. So with this preliminary introduction, so I, Professor Shobosachi Bhattacharya, am working in Indian Statistical Institute, and my lab is AERU lab. As you can understand that AERU means Agricultural and Ecological Research Unit lab. By training, I'm a statistician, but presently I'm working in that biology environment or the ecological framework. So I would like to share some of my thoughts, what I just discovered in some of my previous research with my students, PhD students. But I would like to put it in a very lucid format so that you people can enjoy it. And I'm trying to avoid most of my slides with mathematics. So with a nominal mathematical background and not putting huge mathematical style, I would like to start my lecture. Okay. So as you just see my title, the role of statistics and mathematics in ecological framework, I think there is no need to explain. And but today I have chosen a selective part of that ecology is mainly is a population ecology. So if you look at the ecology, so there are many parts or many paradigms. So the first paradigm of ecology is a population ecology, okay? And that population ecology term actually associated with the very, very important word is called the population growth. So the growth mechanism of the species, growth any species of any taxonomic groups like bird, mammals, insect, bony fish, anything. So actually those growth phenomena, the species growth phenomena are associated with the population ecology stuff. So if I will call the ecological framework, then actually it is associated with population ecology problem and rather the population growth problem. So before going to deep dive on that population ecology part, I think the population growth part, I think you should have some preliminary knowledge. So that's why I thought that it would be better to spend some time on the population ecology part or the population growth part. And before that, you can see that there are a couple of papers which came up in recently in last 2 to 5 years. You can see that the growth of the Artemia species. Artemia is a planktonic species. So is a zooplankton, growth of the Chaetosaurus species. So all are ecological phenomena. Now you see that the growth term is common all the publications. And rather, not only in the ecological theory, but also in epidemiological models, that growth phenomena are very common. So you see that even in COVID-19 pandemic models, so we use the population growth dynamics model to describe the epidemiological problem. So the flavor of the ecological problems, we have used it in the epidemiological setup. So now you understand that the ecological problem actually associated with the population growth dynamics. So not only this low species, but also if you can look at the fish species like Sidinus migala is a common Indian fish, is called in that freshwater fish. So the new growth curve model actually came up with in 2014 with my student. So you see that with this few publications, what I would like to say that the population ecology or the population biology is closely associated with the growth dynamics of the species. So today I have chosen a specific topic for that population ecology because it's a very huge, I mean to say the span. So in that case, I will only cover the cooperation aspect of the population ecology. And as your workshop is, or the conference, whatever you say, the e-conference, that is associated with the sustainable development. So I think it will be better to describe or to spend some time on the species extinction and the suggested conservation management. Because as the conservation management policy or the schemes has a close synergy with, or have a close synergy with the sustainable development of any ecosystem. So you see that in the first bullet, I have written that the existing growth curve model to explain the ecological phenomena of cooperation. So naturally a question arises that, okay, so the growth curve model, the population growth model, which are available in the common literature, whether these population growth models are enough to explain the cooperation movement of the species? The answer is no. So the existing model before 2014, so most of the model actually associated with the competition theory, not the cooperation. So I should raise two names. One is a Charles Darwin and another is a Hamilton. So I think you people have biologic background, so you know the name of Charles Darwin is a very famous natural scientist. So who spent his whole life in that natural science, and another is Professor Hamilton. So what is Charles Darwin's theory? Everybody know the theory of natural selections and the struggle for existence. That means, so from the layman language, if somebody don't know the word, the natural selection, or the or this particular theory of Charles Darwin, so you can lucidly say that, okay, so weaker species die, stronger species alive. That is the basic theory of the Charles Darwin. But Hamiltonian concept is little bit different. It's based on the altruism rule and also the cooperation mechanism of the species. So if you look at the Charles Darwin book, in the Origin of Species, 6 chapters, so you can see that there actually Charles Darwin suggested that sometime when the species cooperate each other, not only the intraspecific cooperation, but also the interspecific cooperation, then my rule or my concept of struggle for existence may not be valid always. So then the Hamiltonian concept will be actually more appropriate. So based on this, actually we'll discuss our today's lecture. And just before going to that, what should I say, that the deep dive, before going to deep dive in that particular problem, I think you need a little bit knowledge of the density dependent and density independent growth curve models. So it's very simple. So if you just classify the growth curve model into two ways, one is density dependent and is density independent. So one is called the size covariate and is called time covariate. These are the terminology. I think it will be better to, if you just look at this particular slide, then you can understand what is density independent and what is density dependent. So the left hand side of the equation, you say this is a rate. If somebody have some biology background, so if you are not familiar with the differential term, but you can actually think that 1 by xt dxt dt, that actually a rate. That rate has some special name, is called the relative growth rate, or the RGR. So if RGR is constant, there is a density dependent. What do you mean by the RGR? RGR means the relative growth rate or the per capita growth rate. So you have an initial size of the species, and dx dt is actually the rate of change of the species, or rate of change of the size of the species. So in that case, with respect to initial size xt, what is my rate of change? So if it is constant, then it is density independent. Why it is density independent? Because as it is constant, it's not proportional to xt. That's why it is actually density dependent. Now you look at the second bullet, it is density dependent. Because if you look at the right hand side of the second bullet, 1 by xt dxt dt, you see that b into 1 minus xt by k. So it means it's depend on xt. And also if you have the RGR function for the third bullet, you see that it depends on the time. The right hand side will depend on is depending on the time. So it's called the time covariate model. So this is not a very difficult mathematics, it is just the expression I have used here to explain the relative growth rate of the population dynamics. So just look at the RGR profile and the size profile. You can avoid the left-hand side column. So you see that the population size is in the x-axis for the first RGR profile graph, and the y-axis is RGR, that means the relative growth rate. You see, for some data, it's linearly decreasing. And for some data, for the second blue and red diagram, you see, is non-linearly decreasing. And in the third, you see that is non-linearly decreasing, but in a sharp mode. And here I have plotted not the size, population size in the x-axis. Instead of population size, I have plotted the time. Again, I should repeat that RGR profile I am observing here. That means the relative growth rate, how the relative growth rate of the species is changing over population size. Some noise are coming. Could you please mute your noise? Everybody. Oh my God. Could you please mute your mobile?
Sir, please continue.
Hello, am I audible now?
Yes, sir.
Yes, sir.
Yes.
Hello.
Please.
I
Sir,
don't
you are-
understand whether people can listen.
You are
Am
audible.
I audible still now?
Yeah, yeah. Sir, you are perfectly audible. And may I request all the listeners to please switch off your microphone?
Yes, please
Sir,
switch
please
off.
go ahead.
Okay. So if you look at that RGR profile, for the first graph, you see that it's linearly decreasing. This is graph, and this is actually so, and if you look at this or is linearly non-linearly decreasing rapidly, or it is non-linearly decreasing not rapidly. So with this few introduction, so the population regulation problem is actually depends on density dependent and density dependent growth mechanism. Fine. And if you see couple of publications came up in last 10 to 20 decades with an exciting title, you see that the population regulation is a bankrupt paradigm. A monumental obstacle to progress like this. So it's a dogma, no? So it's a fate. So why 2001? And there are actually a couple of publications through the concept of RGR profile. How? So,
Sir, your voice is breaking.
voice is breaking. Let's. Is it okay now?
Yes, sir.
Okay, as we discussing that the population regulation. So the population regulation problem, actually we are searching for the relationship between the population size and the growth rate. That is maybe the relative growth rate, as I discussed in two of my earlier slides. And I told you that the Darwin's principle of natural selection actually not always hold. There is a theory which came up in 60s, is called the Hamiltonian theory, and where the cooperation actually is a very, very important ecological or the species phenomena for explaining the population regulation problem. So if we look at some of the species, bob white quills, so they actually coming together or they huddling together to actually to in when there is a winter. So there is a problem if you not stay in a group, that you are more exposed with the temperature because it's a huge winter season. So that's why the bobwheat quills huddling together to fight low temperatures. Similarly, if you just look at the emperor penguin chicks, they're huddling to fight in the cold. So they are all there cooperating. Another example is the African wild dog. So that is here. So here also the cooperative breeding. I think you have some idea on cooperative breeding. What is cooperative breeding? Cooperative being actually that alloparental care. So what is that? That even if after breeding, the parents are not only caring the chicks or whatever maybe the babies. The other members, other group members are also supporting. It's very disturbing. Other species, I mean to say the other group members of the same species, they are also helping the babies. They are not at all the parents. So that is called the cooperative breeder. So cooperative breeding. So cooperation is a fundamental aspect that you can understand that in this particular population regulation problem. So what is our aim? We are mathematician, we are statistician. Actually, there are some existing models of population regulation. Some of the population regulation models are logistic model, Gompertz model, theta logistic model, etc. So we need to develop some new models so that cooperation phenomena can be incorporated. So that is the aim, and today I will devote only this in my lecture. So there is another example of cooperation is called the cooperative hunting and the reciprocal grooming. So I should say one name. So who was our seniors in Indian Statistical Institute. So I can raise the name of Professor R.L. Brahmachari. I think many people know the Ratanlal Brahmachari's name. So actually he spent couple of times, I mean to say he spent 12, I mean to maybe 12, yes, 12 times he went to Africa for doing some research on that lion. So what actually was his finding? That when lion group would like to hunt, the optimum size of that group is 7. If you have more group members, then there is a problem of group sharing, food sharing. If you have low number of members, then there's a problem of hunting success. So you see that here also the cooperation is a big issue. So Darwin's theory of competitions along with the Hamiltonian's theory of cooperation together actually can explain the total population regulation problem. And the reciprocal grooming is another example. So that is my main slide that I would like to spend some time on that. That the relationship between the population size and the relative growth rate as I previously defined it as RGR. Now in this slide, it is defined as a per capita growth rate. It's same because with respect to initial size, how my population rate are changing. And that's why in mathematically, I have written it as 1 by xdt dt or 1 by ndx dnt dt, whatever it may be. So that simple mathematics 1 by xdt dt is not an issue. But the point is that the per capita growth rate and the right hand side is the density dependency. These relationship actually play a big role to explain the population regulation problem. Now, why it is important? So one can predict the pattern in risk of extinction of the species and the demographic history and the different behavioral phenomena of the species. So as I told you that if I plot X or N, whatever may be, so in this slide I have written it as N. So the previous slide I have written is X. So that is actually the population size. And in the y-axis you have the relative growth rate. And you see that there are various patterns with a various value of theta. So what is that theta? You see in that model I have written that this is actually the instantaneous growth rate, and this is actually the term is called the negative feedback, because when the rate is changing, the relative growth rate is 1 by n dn dt. That is when theta is less than 1, then it behaves like this way, and if theta is greater than 1, then it behaves like this way. That means the theta can be used as a regulator for this density dependent relationship, or the PGR and the density relationship, or the RGR and the density relationship. So it is regulator because actually it is regulating the relationship of N and PGR through the value, to the different value of theta. So now there is a paper by Sibley et al. In Nature. It was published in 2006. And that paper says that most of the PGR density relationships are concave in nature. That means concave upward. In mathematical language is called convex curve, but we ecologists, we tell this curve as a concave upward curve. You see, this is concave upward or the convex, whatever maybe you are comfortable, you can say. So what the Sibley's conclusion? Sibley's conclusion was most of the PGR density relationships are concave in nature. Now there is a natural question why it is so. Because most of the species actually would like to spend most of its lifespan around the carrying capacity. What is carrying capacity? The maximum capacity of the system. So a system can accommodate, suppose k people, k number of people. So that k is called the carrying capacity of that particular community. So most of the species actually behave in this pattern, this fashion. Most of the species behave. So they will try to come around the carrying capacity quickly, and then they spend most of his lifespan here. Because if the carrying capacity is 100, that means the species actually can be at the steady state at the population size 100. So the species would like to spend most of his lifespan around that value 100. So the species will start with the value 0, that is the population size here, you see. So 0 actually is not possible. So at least there are two members, one is male and one is female. So each species start with these two members. So after reproduction, it will gradually moves to a number 100, and the aim of that species would like to spend most of his lifespan in that 100 value. And the way they will try to reach that value 100 by the relationship that is called the concave upward relationship. That is a finding in Nature in 2006. And what we did, so what we are thinking, that the overall growth mechanism of the species actually composed with two factors. One is the natural growth rate of the species towards unlimited growth phenomena. Another is the negative feedback of the species, where the environment is pushing on you. So environment cannot allow you to grow unboundedly. Okay, so you have one portion that is your wish to grow unboundedly, but the nature is not allowing you to grow unboundedly. So there are two opposite forces. One is the natural inherent affinity of the species for the unbounded growth, and another is the law of limited growth. That means the nature is actually creating some pressures on you that you cannot grow unboundedly better, because there are many species. You have to follow some rationing algorithm, because I cannot allow you, nature cannot allow you to grow unboundedly. So these two are the main two forces. And if you look at your previous curve, previous models, like whatever I explained, that is see, this is the existing model. So the growth rate is dxt dt and xt into 1 minus xt by k. So xt is the natural inherent propensity of the species towards unlimited growth, and 1 minus xt by k is actually the negative feedback term, what the nature is pushing on you. So these two opposite forces actually acting on the species. But there is a third factor, which is called the cooperation. When you cooperate, when a particular species cooperate amongst their conspecifics, then what happens? Then there must be some new factor that should be incorporated in the species, in the model. So how can I incorporate this? Actually, this is very simple. That was my previous model, and I actually multiplied with n power gamma. So you can see that you can ask me, sir, how you got that n power gamma? It's very simple. That when operation is increasing, that can be defined with the parameter lambda, then you have the more chance of increasing the relative growth rate. So this is the left hand side is your relative growth rate. You see, this is your relative growth rate. So if gamma is greater than 1, then what happened? That n to the power gamma is increasing with time. Because if time increases, then nt also increases. Then what happens? The n to the power gamma also increases. So actually, it's a very simple mathematics. But if you incorporate this into the model, then that model is sufficient to explain the cooperation term. Because again, I can explain this. So if you have dn dt, then if I multiply n, then n into n power gamma 1 minus this. So this is your negative feedback term from the nature. This is your cooperation, and this n is your natural affinity towards unlimited growth. Every species have a tendency to go unboundedly. That particular behavior can be captured by this n. And n to the power gamma is the cooperation activity. And 1 minus n by k power theta, that is actually the negative feedback term that the nature is pushing on you. So based on these three phenomena, actually your growth function or your population regulation can be defined. Fine. So if you in your x-axis, if you plot n power gamma, n power gamma, and your y-axis if you plot per capita growth rate, you can see The first part is due to cooperation. The red curve, look at the red curve. The first part is due to cooperation and the second part due to disoperation. Because when you are done with cooperation, when your actually the need of cooperation is finished, then there is no need to cooperate. Then the portion of disoperation will come into the picture. Now look at the green and the purple line. So that lines actually, just remember that the existing models, that is always is telling you that growth rate is either linearly or the non-linearly decreasing, either in a concave upward way or in a convex way or in a concave downward way. But here you see that it is primarily increasing and then decreasing, and that actually reflects the principle of Hamilton and the principle of Darwin's together. So I don't know whether I have time. Still I have time. Time or I'm done. So if I have time, I can spend some more thing. So Manoj, so how much time
Yes.
I have?
Yes, sir. You can continue.
Okay. So actually the point is the cooperation parameters gamma is very important for the conservation management tool. So how it is important? Because you see that naturally, by intuitive way, you can think that if you have more cooperation, then there is a less chance of extinction. Is it always true? Is it always true? I don't think so. So more cooperation sometimes is not beneficial, but it is detrimental. In ecology, this phenomenon is called overcrowding effect. So if you just go to the examination hall, so it's very common that somebody because many of you from the audience have the teaching background, so you know that the students have a tendency to cheat. So if there is a less cheating, I mean to say only 2-3 people are doing that, then there is a no chaos. But many people are doing that, so that's actually a cooperation theory. There may be a problem because some teacher may rusticate some of the students due to bad manners. So overcrowding or the over cooperation sometimes is detrimental rather than incremental. So in natural phenomena, in ecology, you can see that sometimes that over cooperation actually leads to the chaotic regime. So how? So I think that can be easily demonstrated by that bifurcation diagram. So that bifurcation diagram is a very nice tool to represent the different what should I say, different stage of the species. So how that, suppose this left hand side diagram, you see that there is a parameter RD in the x-axis. So that may be some parameters RD. Okay. And that just I'm explaining the chaotic theory. So what happening when RD is increasing, that means the instantaneous growth rate is increasing. Then up to certain value of RD, you see that there is a single line. That means the species are in steady state. That means the top graph, you see that primarily it is increasing and then it is actually reaching to the steady state. Now you see that if there is a bifurcation, that's why it's called the bifurcation diagram, after some value of R, 1.99, then what happens? Then there is a limit cycle. What is the limit cycle? That means there is a cycle, or like this. And after a value 2.5 and more, then you see that the period is increasing. So primarily it was a 2-period image cycle. Now it's a 1, 2, 3, 4-period image cycle. You see, so that's why there is a nature of cycling attitude. I mean to say, limit cycle attitude, but the period is changing. But after some value of R, what happens? You will be reaching to a chaotic region. So where there is no guarantee that whether you are you are going to up direction or you are going to down direction. It's a totally chaotic way you are doing this. And actually this region is here. So for the value 3 R equal to 3 or more, you see it's all in chaotic region. That means there's no guarantee that whether after time point t, what should be your position. But here you have a specific, after time point t, you have a specific value of your xt or the population size. So that's why these explain that the more value of R is not always beneficial. So if the reproduction rate is high or instantaneous growth rate is high, then all time it is not beneficial. Sometimes it may be detrimental. Similarly, it is the case of the gamma. So when you have the cooperation and when you have more and more cooperation, then it may happen that you are going to the chaotic region. But there is a debate that whether chaos is really detrimental for the species. There are two communities. One is the Berryman group and there is the Allen group. That somebody is telling that chaos is a precursor for extinction. Somebody is telling no, chaos is not a precursor for extinction. What we observed through the model that actually the risk of local extinction increases as the dynamics becomes more complex. The probability of global extinction decreases in the chaotic region. What do I mean by the local and global extinction? Local extinction means that if the species are available in Atlantic, in Pacific, some aquatic species, suppose I'm just discussing. So suppose the herring fish, which is a very beneficial fish for the Atlantic. So you see that in Atlantic Ocean, some region, the species may go to the extinction. That is called the local extinction. But somehow it may happen that species may globally extinct. That is called the evolutionary extinction. So actually the chaos invites the local extinction, but the risk of local extinction increases as the dynamics becomes more complex, but the probability of global extinction decreases in the chaotic domain. So I think that is the, and that thing I think we can. So you see that the same diagram I have plotted for the cooperation model, whatever we proposed, and here also you see that the same thing, that for some value of gamma, this is actually going to the chaotic region. So we have a global population. This is my last portion of the talk. So if you have a global population dynamics database, so that is maintained by the Global Population Dynamics Database NERC Center, and that is Imperial College of London. And these are the data set of abundance of species, around 5,000 species of different taxonomic group, birds, mammals, insect, bony fish, etc. And if we hello,
Friends committee side, sorry for the interruption. We already have few questions pertaining to your presentation, sir, and we are also running out of time.
So that's why I told you, no. So I don't need to cover this part. So I can finish this thing. So
yeah. So could we take the questions now? Or
yes, no problem.
Thank you so much. Sir, here is a question. Do the values of parameters pertaining to fishes and insects change with the time?
Is you so could you please repeat? Actually, is your voice is breaking.
Yeah, sure, sir. Do the values of the parameter,
Values of the parameter.
yeah, in relation to the beneficiaries and insects that you talked about, do they change with the time? This parameter, the values of the
No, no,
parameter.
no, no, no. Parameters actually, if you look at the parameters, mean it's not changing with the time. Parameters are fixed. Fine. So the point is actually what happens when you have the data set, when you estimate the parameter based on the data set, as I explained, you know, that xt or nt, whatever whether there is a size data available over time, then you can estimate the different parameters r, k, gamma, theta. Fine. So for some species you estimated gamma, suppose as gamma hat. So gamma hat is suppose 2.2. And now you have some theoretical value. You can say that this diagram, that bifurcation diagram, actually bifurcate when gamma equal to suppose 5.2. So the gamma is not changing. The theoretical value of gamma, actually I'm observing that how the parameter behaves based on different value of gamma. And when I have the estimated gamma value, and I can see whether the estimated gamma value actually falling in which particular region. So suppose this, I can explain from that, it will be nice. So you see that this is the theoretical value of the gamma. And suppose gamma star is your estimated value. Now you see that the estimated of gamma star actually falling in particular this region. That means you already entered into the chaos. That means you are in threatened mode or you are in chaotic mode. So there is no guarantee that the species can sustain after a certain value of gamma. You understand? Gamma is not changing. Gamma is a parameter.
Okay, sir. Okay, sir. Thank you. It gives me immense pleasure to thank you once again for such an insightful deliberation. Thank you, sir.
Thank you.
Now, I would like to take the opportunity to introduce our invited speaker, Dr. Pritha Bhattacharjee. Dr. Bhattacharjee is an assistant professor, Department of Environmental Science, University of Calcutta. She had 10 years teaching experience. She did her MSc in geology in 2002 and joined CSIR ISAB for PhD in human molecular genetics, where she developed her passion for studying environmental health at cellular and molecular level. She also worked as a pool scientist in CSIR ISAB and a decade of association of such a premier institute, she learned to transform from a researcher to an independent scientist. She joined Calcutta University as a faculty, and Department of Environmental Science gave her a real environment to grow further. At present, she is the head of the department. Her research lab primarily works in epigenetic research. She has 63 international publications with H-index 20. Major focus of her research lies with environmental epigenetics, occupational health, lifestyle disorder, and in silico-based prediction analysis. She wrote several book chapters and contributed a textbook for undergraduate students in vernacular language on environmental studies. Dr. Bhattacharjee has served as editorial board member and reviewer of a number of international journals. She supervised more than 25 postgraduate students from different universities in India for their internship. Under her careful guidance, 6 PhD students and 7 scholars are working in different roles. Now we are very much eager to hear from you, ma'am. What do you, ma'am? Please continue.
Thank you so much for giving me this opportunity. Hope I am audible.
Yes, yes, perfectly audible.
Uh, can I share? Yes. Can you see the slide?
Yes, sir.
Hello.
Yes, sir. Yes, ma'am.
Thank you so much. First of all, I want to thank the organizers from Banwarilal Bhalotia College, Asansol, for giving me this opportunity. Thank you, Zarka, especially inviting me and telling regarding this workshop. So today I will be happy to share few thoughts with regarding the topic environmental sustainability and its linkage to health. The slides are especially meant for the students. I hope they can understand. If they have any questions, they can put the questions in the chat box so that at the end of my presentation, I can try to give their answers. So we all know that what is about our common future, that we all have a common future, only a sustainable art. If it has to be sustainable, then we have to think the basic parameters. What do we mean about this sustainability? There's a development which meets the needs of the present without compromising the ability of the future generation to meet their own needs. This is called sustainability. That is, it should not be compromised our future generation. And to have a sustainable society, a sustainable environment, it has basically three basic pillars. One is social, another is environment, and another is economic. These three pillars intersect, three areas intersect with each other, and only the section where it meets three pillars, then only the sustainability comes. Sustainability has certain key components, and the main component is natural capital. Natural capital indicates the natural resources and the natural services. And we know the natural resources are renewable and some are not renewable. So whenever we think about certain development, the first... Hello? Whenever we are talking about certain development, we can say that the first thing that hits to us is the economic, and also at the second priority level, it comes to the social. But we ignore or we less prioritize our environment. So we must choose to prioritize the long-term sustainability over the short-term gratifications. And we have to calculate the true cost borne by the societies in future instead of just the price of the action and policies today. So whatever the policies are taken today, whatever in the terms of the price, we see the environment, we see the development, those all are economic centric mainly, but we ignore the environmental part. And I will give you one statistics regarding this. Is whatever we call about this sustainable development, we can understand that we are not sustainable yet. The countries are differ in their resource use and it impact on the environment differently. The developed, so-called developed countries that we say, that's a westernized country, they are developed with all the facilities, they use 88% of world resource. Just imagine, developed countries, they have a low population size. You have just have a lecture on population. So the world's population, where it's only 18%, the countries, they use 88% world's resource. and they cause 75% world's pollution waste. They cause the pollution, the tremendous pressure on Earth by polluting it. On the other hand, whom we call as the less developed countries, they have 80% world population, 82% world population, but they use less resource. They have the access to less resource. However, they're rich in biodiversity. So it's depend on the lifestyle, how we are utilizing to have a sustainable development. The thing which maybe is less taught or we ever less think, that's the ethics of sustainability. Whenever we talk about ethics, the first thing that comes about ethics is what is good, what is bad, what is right, what is wrong. And mainly we use ethics in the terms of that the ethics, the moral ethics, care ethics, we use ethics in these terms. But sustainability ethics, yes, there is ethics of sustainability. We have to know that during the development or whenever we are talking about the development, to have in a holistic manner what are the right things, what are the wrong things. Just see that who gives an estimation that approximately 24% of the overall global burden of disease is attributable to environmental factors. And this range that lies from 12% in the wealthiest country to 40% to the poorest country. So the wealthiest country, they are having access to most resources, they're using it widely, they are polluting it, and the same time, since they are the wealthier countries, they have most technologies, facilities, their disease impact or disease burden in that population is relatively low. But on the other hand, the scenery is completely opposite to the other areas where we found that the malnutrition, the environmental degradation, they do not have the right access to water, all these water stress, all these conditions, they suffer. And the malnutrition is one of the major problem. So 40% in the poorest countries, we find a number of health-related problems. Nearly 2 million people around the world die every year, and others are made sick or disabled as a result of exposure to smoke from fuels burned in indoor cook stoves. Now we are talking about the natural resources. I have already mentioned that the sustainability has some key features, has the key indicators, and the very important thing is the natural resource, and some of which are the renewable and some are non-renewable. So still we depend on the fossil fuel. And these fossil fuels, they are non-renewable. And not only they are non-renewable, they are causing lot of GHG, the greenhouse gas emission. So the population who do not have the recent facilities, they still have to depend on this agriculture, forestry, and for their livelihood, they are struggling. They have to use all those things for that basic fuels, and it's causing indoor air pollution. Indoor air pollution often becomes more, 5 times more than the outdoor air pollution. Recently, we are still in the COVID pandemic, and we have threatened by the number of death we have experienced and we have seen. But the true data is air pollution related death is even more, which we have not noticed. So whatever it's the we have to think for the sustainability in a bigger scale, in a holistic way. So it starts from the individual, it starts from the population, and if we think up to this impact, it comes in the population, into affluence, into technology. These three factors cumulatively causes impact. And the tragedy of common, we have heard this term, there's a tragedy of common. We think that the dilution is a solution to pollution, but no, this concept is no more valid. So we have to think that individual is the basic, the demands of life that will create a societal changes, societal determinants of health, and that ultimately will lead to the environmental determinants of health. The ecological footprints are very, very important. It's not important to just copy the westernized things, because we have already we have seen that the western population, the wealthier population, they are actually using more of the resources, but definitely for the technology they can withstand this problems. But the underdeveloping countries, the population, they have more population definitely creates a lot of different problems, because more the number of population, then its impact would be bigger. So we need to have this an equity in terms to reach a common future, common earth, to if we have to live in sustainable way, if we have to think that yes, we cannot, some some people can afford to move to the different extraterrestrial in other areas. They are thinking to having some land in other areas, in moon, in Mars. But if we think that earth is a place where we have to live, then it has to have a holistic way of living. And the health is an important part of this sustainable lifestyle. It is very, very intricately linked. And to have good health and well-being, we should have certain important goals. And from Indian perspective, considering the rural backdrop, we see there are several challenges. There are several challenges which need to be addressed. So we have to reduce the global MMR to less than 70%, so that we can take care of lives properly, and all preventable deaths under the age of 5 years. We have to take care of the epidemics, reduce the premature mortality. We have to strengthen the prevention of the treatment of substance abuse, reduce the deaths. And another thing is the prenatal toxicity is very, very significant. So we have to think this entire exposure and how these environmental components are linked to health. These two important things are going on. One is the epidemiological transition and the environmental risk transition. Environmental risk transition, for this environmental risk transition, the exposures are coming from household, community, and global level. So it has an immediate impact to delayed impact, which we call about the acute toxicity and the chronic toxicity, acute exposure and the chronic exposure. Most of the things we get to the chronic exposure, and which has a delayed effect, prolonged exposure, come up with a number of diseases which we cannot immediately identify. So after a prolonged exposure of 40 or 50 years, we can see the deterioration, and we can see even the death more due to cancer. We have seen that the epidemiological transition, that how the communicable disease, maternal exposure, perinatal, or that I have already mentioned, there's a prenatal exposure, nutritional component. All these are important factors, and also it's leading to the a number of non-communicable diseases. As lifestyle is very closely linked with the health, unsustainable lifestyle is very, very dangerous. Just for an example, it is estimated that 30% of non-communicable disease deaths occur in individuals under the age of 60 in low and middle income countries, as opposed to 13% in the high income countries. So we have already seen the equation that impact is dependent on few important factors, on which one important attribute of it is population, another attribute is affluence. So the scenario, like in India, where population is high and affluence is low, we have a number of inabilities to protect the diseases or to take care of these non-communicable diseases. So we have to take a holistic goal, holistic approaches, and to meet these major challenges that meet primary healthcare needs, particularly in rural areas, control communicable disease, to protect vulnerable groups. The vulnerable groups are more those who are at the young age and the ages more than 60. We have to take the urban health challenges. Here we find that in the rural areas, they do not have very good health facilities. They have to go in a long way to reach to get any facilities. So the technology plays a very important role, that how telecommunications and other facilities, by developing some sustainable healthcare group, we can kindly mute, so that we can have this access, the primary need of life. We can provide this healthcare support. We have to reduce the health risk from environmental pollution and hazards. So for that, there are so many policies are there, but not only the policies, we need to monitor. And now coming to the green healthcare mission, I will say that since I have already mentioned that health has a big connection with the sustainability, and in healthcare, healthcare is very, very important, providing the main support. They are providing us the main support, the lifeline. They are in the lifeline, but still in different healthcare units, we see that the carbon footprint is significantly high. It comes from the transportation using the fossil fuel and the anesthetic gases, drug manufacturing factories, the food processing units, air conditioning, freezing unit, heavy electrical equipments. Those are used in the hospitals and the healthcare units. They causes greenhouse gas emission. And here I must mention the technology that we are always talking about: the population, the affluence, and the technology. All these three are the important parameters or the important attributes which causes this impact. And when I was studying this green healthcare regarding work health and sustainability, I have found that several articles from western countries, that where they have taken this green care healthcare approaches has been considered in uh, in Western countries. Here we have found several literatures, and they have shown that how they have minimized the greenhouse gas emission. But here in India, we have not found enough baseline data. So this part has to be improved. So whenever we talk about the healthcare, health facilities, we know that we do not we have to increase more basic fundamental healthcare facilities to provide that, or that will come in a proper ratio with the population that we have. But not only in the number, we have to take care in the quantity as well as the quality. In terms, we have to think that the healthcare practicing sectors, if they consider these green healthcare approaches, that it is not only it has been taken care of the well-being of the patient and the workers, those who are working in this healthcare unit, but it aims to reduce the environmental vulnerability, because the instruments that we are using over there, the transportation facilities, again it is dependent on the fossil fuel, the gases, having the VOCs, the drug manufacturing units, and we have enough biomedical waste. So that needs to handle properly. More the population, definitely more will be the biomedical waste, and that has to be done systematically in the facility. We have to increase the number of biomedical waste recycle facility that which will take care of this waste management, biomedical waste management, this part. And in this COVID pandemic, we have already experienced that whatever the requirement, we do not have that much of facility. And again, incineration causes a huge amount of greenhouse gas emission. So in future, to have some green healthcare mission, we have to take care that how solar panel, these facilities can be taken care, some battery operated ambulances, so that we can reduce the fossil fuel driven services. We have to take care a holistic way, not only prioritizing the individual and the society, also we have to prioritize the environment. Otherwise, sustainable society, sustainable environment will not be there. And I want to emphasize today one special aspect, that is the water resource and sustainability. Water is definitely our lifeline, and we all know that's only 3% water. Although it's a blue planet, the 3% is the fresh water, and among that is available drinking water is 0.024% only. And in that, 54% of India faces high to extremely high water stresses. And even if we do not take care about the water resource management properly, we will be in serious trouble in upcoming days. We have to think about the water footprint. As just like the carbon footprint, we have to be very aware about the water footprint. That and the another part, we know about the green water, blue water, and the gray water that comes from the industry, blue water that is available in the surface and the groundwater, and the rainwater as the green water. But apart from this, there is virtual water which we miss out. That is, in whatever we are using, how much water is needed to procure that, to have this ultimate that is coming in that. Just simple, in a food that we are eating, a single here is an example, a single dish of pasta, what is the footprint of that? So it's the actual water use, and that is the cooking water that is using to make that pasta, and the virtual water is it's in the farming, processing, and transportation. So you can understand that environment sustainability, it's a holistic thing. Each and every aspect is interlinked with each other, though we understand it physically or it's virtually. So the strategies, the solution, the technology plays a very, very important role, because we have enough good technology, and therefore we should be aware of all this virtual water and the demand, the individual demand, that the demand of our choice, whether it's a consumer behavior, we have more in the consumerist way of lifestyle. We go through that, we have to buy new things, these are the new trends, we are buying new clothes, we are having that ready to eat foods. So we never think that how this consumer behavior actually creating an impact on overall. So all these things have to be incorporated properly to understand these sustainable things. Otherwise, the sustainable goals will not be reached. I will end up with showing few work from the arsenic research. As I have already mentioned, that water is very important part of our lifeline. So therefore, in each and every cases, we need to know, or we have to think about the technology that takes its optimum use. Over exploitation due to high number of population or its demand, each and every time that these things are coming and they are interlinked, that more the population, more is the demand, and depending on our demand, we are using we are in a demand of having more crops, and for that we are utilizing the underground water, or we are spraying more on pesticides, which are causing the underground water contamination, or due to our over exploitation, the water table going down. So all these are creating an implications in this vegetables and crops. It's coming up there, and this is staying there as a residual amount. So these are creating toxicity on us. So ultimately human are suffering, but by the process that we are doing. So if we have only anthropocentric idea, the sustainability will never be achieved. It has to be biocentric and in a holistic way. These are some pictures from the camp where we have collected the water samples as also the biological samples, that is urine, blood samples, to know that what is the level of arsenic contamination from the environment and also the population who are exposed. We have gone through the arsenic metabolism, and it is very alarming that whenever arsenic it is in the groundwater, once we consume it, converts in the inorganic to organic phase, and it affects our system in a different way. It causes damage in our DNA, it alters our epigenetic profile, it causes certain genes to be upregulated and downregulated, which causes carcinogenic pathway. It ignites that carcinogenic pathway. So, and it's not only arsenic, that we are living in an environment where there are several exogenic factors, physiological factors, physical factors, physiological factors, all these environment and the other environmental factors, actually by this epigenetic interaction, they give signals to our gene, and it causes an overall impact. So far we are talking about the individual, economical, and societal and environmental, these three parts. But apart from this, there is another important part, which is our genetic part. This all together creating an impact, or they are interrelated to each other. And arsenic is such a heavy metal that we are uptaking it from our environment, and it is affecting multiple organ in our system and leading to cancer. These are some affected faces from our field camps, where we found this, even these small children. I have already mentioned this, the prenatal toxicity is one of the very important window, and you are also observing this adult population. So depending on the window of exposure and the duration of exposure, the outcome matters. So water, which is our lifeline, due to the anthropogenic activity, wrong utilization, and we are depleting these main resources, our water table is going down, we are contaminating our the aquifers, and that's why even the crops that are growing on it is having the poison. So we are eating poison on our plate, and this is due to our activity. We have to remember that we have a common future, that I have mentioned in my first slide, that we have a common future. So we have to take the responsibility, otherwise the poisons, they are actually crossing our placenta and affecting the generation next. So it's not only a single generation, it is affecting two generation down downstream, this effect is remaining in their signal. So it's important to focus from our diet, from our entire environment, from the individual to society. This has to be controlled at these two levels so that the ultimately environmental goal can be achieved. There are pretty examples from our field that the population having similar exposure, they have different outcomes, all due to these environmental and genetic interactions. We have seen that the people that who we have more arsenic in their rice, that I am just explained, that we are having poisons on our plates, knowing or unknowingly, because we are not growing the crop, so we don't know that from where it is coming. So the main thing is that when we are polluting our water, when we are polluting our natural resources, it will act as a boomerang and it will hit us badly. So we have found that the individuals having having this arsenic contaminated rice, it is causing the genetic damage, and this has been very widely circulated. This paper has been most cited article so far, and the European Union has changed their regulations and they have a threshold limit. So here from Indian perspective, what I find that we have very lack of information regarding the quality, and until and unless we don't know the quality, that what we are eating, the impact that is happening on us, we cannot control. So I don't want to go into detail of this, just giving a glimpse that the solution is to change in the safe water. There is no more solution. We have to provide the safe water. So as I have already mentioned, that it is also coming through the irrigation. So if it is not drinking, it's not only the drinking, so irrigation also giving us this toxic outcome. We are having this. So holistic solution has to be there, otherwise we cannot address a pathway. We today in the pandemic, we have seen that in COVID pandemic, the plastic use has been increased due to more, we are not going to the places for avoiding contact on this. And so, but the plastic, we are giving more and more online orders, we are getting foods in the plastic packets. They are causing another hidden pandemic, or another pandemic may be waiting for us, which we are ignoring right now. So all actions are interrelated to each other. This is the entire research that we do, that the workflow, we take the samples, we study the networks, that how this arsenic is hampering in our system, what are the cellular deregulation that they are doing. This is the epigenetic mechanism with which we understand the environmental signal and the genetic signal, who are interconnected between this. These are the epigenetic moieties. Their changes in these histone proteins, they alter a certain gene to express and certain gene not to express. So our genetic, all these data are actually making you understand that how these environmental change can impact on the cellular changes. These are the documentation of this. They are changing at our each cellular, subcellular, and the molecular level. They are causing changes in the DNA damage. They are not allowing DNA repair to occur properly. They are causing changes in the mitochondria, and this mitochondria, through mitochondria, you know that mother passes the mitochondria to their offspring. So if there is any changes in the mitochondria, so the prenatal toxicity, through the prenatal toxicity, it will be transferred to the next generation. So mother needs to be protected. The proper diet, proper nutrition, proper health care should be focused to mother to give a to give a proper child to born, so that the prenatal toxicity can be reduced. So there it has to be a sustainable solution to provide in this rural area. I have already mentioned that there are several challenges that we face from the Indian scenario, because we have a big population size, we have less affluence in the rural areas, less facilities do we have. So we have to provide them some very common facilities, the alternative source of water. We have to educate people regarding the rainwater harvesting, the co-precipitation, how that solar irradiation, due to solar irradiation, certain germs can be deactivated, and some simple ways so that they can sustain their lifestyle. Along with this high throughput technology, for which we are doing this epigenetic research so that we can come up with the epitherapeutics, and we are trying to make a chip with which we can easily identify the population who are more susceptible, because until and unless we can understand who are more susceptible, we can provide them more protection, so that we can have a better future. So this is our sustainable low-cost primary healthcare model for arsenic research, and I hope in future we can train more local people, we can reach more local villages, and can by creating this arsenic health kiosk, and give some solutions to this important problem. So this is my research lab, and thank you so much for giving me this opportunity. Thank you.
Thank you, ma'am, for enlightening us in such a pertinent manner with your thoughtful deliberation. We are really mesmerized with your reverential voice. You have nicely pointed out the tragedy of commons, which is intricately related with the present day theme. Now, moving on to question answer session. We have a few questions from the participants. The first question is,
yes, I can see one. It's on Greta Thunberg. I hope the chat box. Presentation is not visible. Someone has wrote that the presentation is not visible.
No, it was clearly visible. Our first question is, do you think that in developed countries, the role of developed countries is much higher in towards sustainability than the developed?
Definitely, because this is very clear that already those are the developed country, they are using more resources and they are polluting. So these there are so many summits are going on for this same thing. So it's not only the theory, it's not only the knowledge. Definitely the sustainability will come only when we will do some real act. It's not only the promise, we have to keep these promises on.
Oh, sure. That means you want to say that polluters should pay for the pollution.
Absolutely.
Okay. Thank you once again. Now I would like to request.
Thank you so much. Thank you so much for giving this opportunity and share with all of you. Thank you so much.
Okay. Now I would like to request our colleague, Dr. Vinita Dotto, to continue the fifth technical session. Okay, what will Vinita?
So, am I audible, sir? Audible. Okay, thank you. I am Dr. Binita Datta from Department of Chemistry and Secretary of this conference. I welcome you all in this fifth and final technical session of this 2-day long conference. And now it's time to listen from some of our emerging researchers and faculties from our participants. And our first participant is Jimmy Devbarma. Mr. Jimmy Devbarma, are you here with us? Can you just respond? Mrs. Okay. Are you here with us? Please respond. Okay, maybe not present. So we are moving to our next presenter, Dr. Sanchita Dotto. Dr. Dotto, are you present here?
Yes, ma'am, I'm present.
Ma'am, can you kindly just start your presentation?
Yeah.
Yeah.
I'm audible and visible, right?
Yes, ma'am, you are audible and visible.
Okay,
Do you have presentation, ma'am?
yeah. So, good afternoon, everyone.
Do you have any presentation? You can
Yeah,
start.
I had presentation. Yeah.
Yeah, please start with that.
Yeah.
Dr. Sanchita Dotto, she is Research Associate of Indian Council of Medical Research from Department of Pharmaceutical Technology affiliated to Jadavpur University. And she will tell us about the class 1 histone deacetylase, a potential target for cancer therapy. Ma'am, we are eager to hear from you, ma'am. Please continue with your
Yeah, I'd like to share my screen now. Please check if everyone can see.
Yes, ma'am. Please proceed.
Can you see my screen?
No.
Can you see my screen?
Yeah, ma'am. Just starting. Yes, ma'am. It's visible. Please proceed.
Okay. Yeah, let me just enlarge it. Okay. It's not becoming full screen. So should I continue like this?
Yes, ma'am. Whatever you feel comfortable. You can just press F5 for presentation. It's not working.
Okay. Yeah. Is it okay now?
It's okay. You just proceed.
Yeah. Okay. So as you have already introduced me, so there is no more need to give any introduction. So now I'm going to my research topic, the paper presentation topic, which is class 1 HTEX, which is a potential target for cancer therapy. Now to start the presentation, I would like to just brief about cancer. What is cancer actually? Cancer is actually abnormal growth of cells. Abnormal growth of live cells in any part of body, the cells can start growing abnormally. This special situation is known as cancer. As we all know that cancer is a very severe, cancer is actually the second major cause of death globally after heart disease. Heart disease is the first, then it comes cancer. Now, there are many causes of cancer, like viral infection and some radiation exposure. Many more are there. But among all these, the first and most worrisome cause is genetic alteration. So these genetic alterations, how these genetic alterations help to progress cancer. These genetic alterations usually activate the oncogenes and suppress or deactivate the tumor suppressor genes. That is how the genetic alterations progress the cancer. And what type of genetic alterations usually cause cancer? The first type of genetic alteration that causes cancer is DNA methylation. And then it comes the modifications of histone. What kind of modification? Acetylation, it may be acetylation, methylation, phosphorylation, different kind of modifications in histone also can lead to cancer.
Ma'am, sorry to interrupt, ma'am. Your slide is not changing.
Yeah, I will change. I will change it. It's not complete yet.
Okay.
Yeah. What is histone? Histone is a type of protein that give a support for chromatin structure. To feed the DNA is very long. To feed that long DNA inside the nucleus, it is histone which give the compact structure of the long chain DNA. Now, now histone modifications that usually causes cancer. Among them, acetylation status of the histone is in recent years. Acetylation status of histones is very too many research work is going on about this type of modification, acetylation of histone. The acetylation level of histone is maintained by two enzymes. One is histone acetyl transferase and another one is histone deacetylase. Now, histone deacetylase are a family of enzymes that deacetylate the lysine residues of histone proteins. Now, if this HDAC or histone deacetylase, now this HDAC, if it deacetylate the lysine proteins, what happens? How it lead to cancer? This deacetylation of lysine residue on the histone protein increase the ionic interaction between histone and DNA. How? Because if deacetylation occurs, then the ionic interaction increases between the histone is positively charged and DNA is negatively charged. So a very compact structure of chromatin is formed by this deacetylation method. And if the chromatin structure become too much compact, then the DNA strand become unavailable for the different transcription factor to bind. And if transcription factors will not bind on the DNA, then further synthesis of many proteins will not happen. And this is how this deacetylation leads to suppression of many important proteins, and which usually help in suppression of tumor. So as the proteins does not synthesize, so the tumor suppression does not occur and cancer progression happens. Now, here I am going to talk about this HDAC group of protein. In human body, till now, 18 different HDACs have been found. These, according to their structural homologies, these 18 HDACs have been classified into 4 different groups: class 1, 2, 3, and 4. So here, I'm just going to tell a little bit about this class 1 HDACs. So class 1 HDACs are composed of 4 different HDAC enzymes: HDAC 1, HDAC 2, HDAC 3, and HDAC 8. These all are localized, these all 4 enzymes are localized only in nucleus, and they are zinc dependent enzymes. In their catalytic side, what is the meaning of zinc dependent? In their catalytic side, they have zinc ion, where they bind with the histone proteins. Now, what is the role of all these class 1 HDACs? Before going to the role, I want to tell that in recent last few decades, this studies about all these class 1 HDACs is going on. And why? Because in different kind of carcinoma cells, like gastric carcinoma, colorectal carcinoma, or breast cancer or leukemia, very high expression of these class 1 HDACs have been found, which draw the attention of the researchers to look into this matter respectively, to look into this particular group of proteins deeply. Now, why this expression increases? and how this higher expression of class 1 HDAX helps in cancer progression is that this, if the expression of this HDAX increases, then they will be deacetylate the histone more and more. And if they will be non-stop continue deacetylating the histones, then the DNA structure, chromatin structure will becoming compact more and more. And if they become compact more and more, then the DNA will not be available to further synthesis of tumor suppressor proteins like CDK1, cyclin dependent kinase 1, which is a very important protein in cell apoptosis. So all these type of proteins will not be synthesized. And if all these type of proteins will not be synthesized, then cells will not go for apoptosis and they will continue growing non-stop. Now, if they will be growing non-stop, then what will happen? Then the cells will now start invasion from one tissue to another. Cells will start moving from one tissue to another. They will start metastasizing. Metastasizing means they will start developing from one body part to another body part. They will start developing from one. If they will start developing in brain, maybe they will start developing another set of cells, non-stop tumoric cells in liver.
To interrupt you, ma'am, can you just proceed a little faster because we are just running.
Yeah, definitely. And so all this invasion, metastasis will lead to cancer progression. So because of all these causes, for last few decades, the research is going on for a target-specific inhibitor, target-specific class 1 inhibitor, which can be considered as a breakthrough in cancer treatment. So class 1 HDAC specific inhibitor are designed targeting the zinc ion containing catalytic domains of the HDAC enzymes. And mostly the derivatives of some chemical compounds like hydroxamic acid, benzamide, and short chain fatty acid derived compounds are used for designing these inhibitors. Now, some of the class 1 HDAC specific inhibitors that are in research right now are panobinostat, belinostat, and SHAHA, etc. But all these inhibitors are having one common problem, that is selectivity. What is that selectivity? Selectivity means if we are designing these inhibitors, they are not only inhibiting the class 1 HDACs, but they are inhibiting some useful, important proteins of the body also, which are leading to adverse side effects. That is why, and another issue about all these inhibitors are the toxicity. The toxicity, if these inhibitors, the chemical compounds that are used to develop these inhibitors are not so much friendly with human body, they are causing toxicity. So further advanced research is going on for in level to phase 2 trial for all these inhibitors so that it can be used in human body. And we have to work in keeping eye on this fact that we have these inhibitors has to be less toxic and perfectly selective for the class 1 inhibitors. So the search is still on for this. So this is all I have to say.
Ma'am, thank you so much for your valued presentation. And if any participant has any question for her, please. If there is no question, ma'am, thank you once again for your celebration. And yeah, we are proceeding to our next speaker. So there is a little change.
Thank you very much.
Dr. Shumon Kumar Guho. Are you here, sir, with us?
Yes, I am there.
Yeah, thank you so much, sir, for being with us for this year too, because sir. So, sir, our keynote speaker last year, and we remember you, sir, and so much.
Yes, thanks.
And can you kindly start you?
So, can I
Dr.
start?
Suman Kumar, yeah, you start your presentation, and I'm just introducing. So Dr. Suman Kumar Guha, he is a scientist from Variable Energy Cyclones, Department of Atomic Energy, Government of India, Kolkata. So he will enlighten us with a very interesting talk about the safety management in VCC, his institute during the COVID-19 pandemic, aiming towards the sustainable health and happiness in the workplace. I think it's a very interesting and good talk. We are eager to hear from this. Please.
Whether my presentation is visible, ma'am?
Just wait for 2 seconds.
I cannot see anything from here actually, from this side.
Oh, okay. Can you just stop and start your presentation once again?
Is it coming?
Until now. Yeah, it's started to show.
It is coming, huh? You can see.
Yeah.
Okay, then I'm starting. First of all, that I want to thank the organizing committee of eConsus 2021 and my respected session chair, Dr. Vinita Datta and Dr. Manas Pal. And I must thank Dr. Rajrupa Ghosh for providing us this opportunity to present our work in this very great occasion. So this type of seminar is always helpful for sustainable development and for making awareness among the common people. We all know. So basically today we are going to describe that what we have done for the last almost 2 years in our institute for maintaining the health and happiness in our workplace. So safety management in VCC during COVID-19 pandemic aiming towards the sustainable health and happiness in the workplace. Now, what is the VCC? VCC, you know that VCC is a premier research institute under Department of Atomic Energy of Government of India. We basically deal with our atom and the splitting of the nucleus, and for that we do various types of experiments: nuclear physics experiment, accelerator technology experiments, accelerator physics experiments, societal activities are also there, allied researches are also going on here, detector technology. So this is VCC basically. So our institute is a big institute. It is located at the heart of the city, Kolkata, and it is almost, the area of the institute is, on the office area is almost 15 acres. And if we consider the office and our residential area along with the SINP, Shah Institute Nuclear Physics area also, so it is near about 25 acres in the heart of the city of Kolkata, Salt Lake Sector 1. So we have to maintain a very strict regulation during the entire COVID lockdown period, because all of you know that you are also having the opportunity for working from home, but in our case there is no option for working from home. So it is basically we are working in our office place only, from our office place only. So we have to maintain the safety regulations as defined by the Government of India and state governments for maintaining the restrictions of this COVID-19 pandemic in our office and our allied and our neighborhood. So for that, what we did our basically preventive maintenances taken during the pandemic, and we will go the next slide, managing our resources, and then third slide is basically public awareness drive in our office and the neighborhood. So in detail, if we go, so it is a little bit slow. Just a minute. So this is the slide. So first is that preventive maintenance taken during the pandemic. This is as I told you, that it's a big, huge office and comprising of various buildings inside that office for different research facilities. So there are basically almost 1 lakh to 1,50,000 cubic feet area of that local common areas. That means lift, lobbies, and etc. So we had to sanitize all these things. So first of all, that from since 13th of April 2020, when we started our office, so prior to that, we did a thorough sanitization using 49.9% activated hydrogen peroxide solution, along with that 0.01% silver nitrate solution, percent to percentage concentration. So for that sanitization of that entire volume first, and then all the you can see that all the activities what we did. So along with that, all the departmental vehicles, our ambulances, or all the lobbies, our guest rooms, everything, and including security offices, etc. So after that, we did a thorough sanitization drive for our residential area also. So residential area, you can see that it is that just located at the heart of the city. So we did a thorough sanitization for the residential area also by ourselves own, and these are all our own staffs and our own team. We developed, we trained them. After that, that local Vidhannagar municipality, they also helped us a lot for this sanitization process. And this slide is not moving, I don't know why. Yes. So this is another slide that Vidhannagar municipalities, they are also helping us for sanitization of our residential complex, because the no outsiders are allowed in our complex. So they are doing the sanitization in our residential area also. So these are all coming under preventive maintenances taken during pandemic. So we started like that only. So after that, when our office started, so slowly we started our canteen, our all the common facilities like that. So before starting all these things, so we distributed all the mask, gloves, sanitizer for the each and every staffs for the canteen and the common areas. We insisted for the individual mask, social distancing, individual temperature monitoring. You can see that temperature monitoring is going on at the main gate of VCC, whether he is in vehicle or whether he is coming on his own, or each and every person we are insisting for them for the social distancing. And you can see that the preventive maintenance during the pandemic, that effort for social distancing in the canteen itself. So our canteen, we basically run our canteen for the last few months with the strength of only 10 to 15%. So you can see that we never allowed any person to sit nearby or in a table, two or more person in a table, single table. And now you can see that another thing, that in our preventive maintenances of the logistics management of the VCC store itself. VCC is we are having a huge stores, and we are having a lot of components which are coming from the outsides of the India. Something is coming from Europe, something is coming from USA. So these are all we sanitized, and we maintained a specific sanitization scheme for each and every material which we are receiving, which we were receiving from during this pandemic period. And after that, you can see that managing our resources means we started our office with all the meetings means our high level meetings, our national level or international level meeting, or the promotional interview review committees. All these things we did through all our web meeting. There was no physical meeting we took place, although we were coming to office regularly, but everybody of us were attending meeting from its own places. And this is our managing under the part of the managing resources in-house development. What we did, we developed the ethyl alcohol based hand sanitization and sanitizer for the distribution among all the officers and staff members and the contractual personals. So as you know that we are working here almost more than 1,500 people each and every day, they are attending the office. So we developed our own sanitizer, we distributed this thing in regular basis for each and every sections. So now come to the next slide. Just a minute. So this is the managing our resources in-house technical development. We using the technology of that microcontroller based Atomica 2560 based sanitization walkthrough. This is a total area of 1005 cubic feet area, we made a sanitization walkthrough by ourselves own, and for the 20 seconds the sanitization shower will be there for the each and every people who is coming from outside to entering into the VCC office campus. And for that, after that, we developed this PC is basically slow.
Connection.
Just can you see this slide?
Yeah. The slide we can see.
Is not going to the next slide actually.
Oh. Let's wait for the connection is.
Yes. Now this is our under the again it is under the management of the resources, in-house technical development using technology. We have developed that huge number of hand sanitizer vending machine and installed in the various places of VCC. You can see our director is giving demonstration of the first unit what we developed in our office and installed. After that, this is the you can see that the managing our resources when we started our canteen. So we stopped basically all the handling of the cash, because you know that the handling of cash is basically the contamination of the germs and all these things is very high. So we introduced a contactless card system for each and every payment and billing system. After that, we under the managing our resources, we conducted two consecutive COVID vaccination camps for our employees, their family members, and you can see that the lots of old people we have given them the facility for this COVID vaccination camp, and almost 400 numbers of beneficiaries were there, and we conducted this thing in our residential area in Salt Lake. And after that, under the public awareness drive, so not only this thing that managing our resources or the preventive maintenance, we tried to aware the people about this particular disease and how to restrict the spread of particular disease. So for that, that we had a massive tree plantation drive aiming towards the clean and clean VCC during this COVID-19 lockdown period. And after that, this again it is coming under public awareness drive. You can see that we have inaugurated one immunity garden in our VCC campus itself. So there are basically 18 to 20 numbers of different types of different species of plants which are having basically medicinal plants which are having a specific immunomodulatory action, and with the help of government of West Bengal Medicinal Plant Board, so we developed this thing in our VCC campus, and total 400 numbers, almost 400 numbers of saplings we planted there. And now after 1 year, it was developed in February 2020, February 2021, and almost after 1 year, it has grown, and it is very beautiful, and people are very much appreciating this thing. And we have basically aimed to aware the common people about the normal immunization process, or the immunization process how he can develop by himself on using these natural resources is the main aim of us for developing this immunity garden. So another thing is that you can see that during this just before this there was a that during Amphan May 2020, so during this pandemic lockdown period, so we had this Amphan, and we had a devastation in our campus. So lots of trees were uprooted, and among them there was a family member of VLCC that was a 50 years old mahogany tree. So it was uprooted. You can see that his picture is there. So we took an initiative to make it alive and kept it with us, and to make to give the message to the common people that the tree after its death also it is alive and it is staying with us. And after that, what we did during this pandemic period, we had a nationwide awareness campaign for for the restrictions for the contamination content for the the restriction of this particular disease. And with the aim for that, we had a nationwide awareness campaign, online drawing and essay competition for the different age group students. and we had a huge amount of responses among them, and they participated in these things. We gave them award, and their name and their essay were published in our website of the Government of India, and we provided each of them, we provided the e-certificate, and finally it was very much appreciated among the different age group of people. And you can see that just after 1 year or just after 2 years of this entire period, and how we have dealing with this thing, and this is our green and clean VCC inside this heart of your city, and our Swachhata committee, that is the thing, and that Swachhata committee VCC, which is working under the Swachh Bharat mission project of Government of India. And on behalf of Swachh Bharat Swachhata committee VCC, I would like to thank my co-authors, Sri Taposh Hamanto, Sri Taposh Bhaumik, Dr. Subhashi Chattopadhyay, our director of VCC, and all the Swachhata committee members and the staffs members of the VCC for maintaining this thing, and for the restrictions or the for contention of the spread of this disease inside the VCC campus. And we really succeeded, and we are working since the last 2 years without any very much very outgrowth of this particular disease inside either in VCC or in our residential campus. So we are aiming towards the sustainable health and happiness in our workplace. So we will be working with the same motto. And thank you very much again for giving us the opportunity for presenting our work in your this very nice e-conference. Thanks a lot.
Thank you so much, sir. It was a wonderful talk, and enjoyed a little bit more because these values are known for me, because I was a PhD student from Shaha Institute of Nuclear Physics. So it's building. I have had a lot. Um, uh, thank you. So this institute I have visited many times. All the canteens and everything is known, and so many memories are just coming and striking.
Thanks. You come again. You come again.
Yes, sir. Pardon, sir.
You come again. You will have a good time here, just for a visit.
Thank you, sir. And one audience participant from our audience, Dr. Mohua, she raised hand. Are you here, ma'am? Do you have any question, please? Dr. Mohua, she raised hand during the presentation. No, maybe she's not here right now.
Okay, thanks a lot.
We are just moving to the next participant. Dr. Are you here now?
Yes.
Dr. Ngos, she is assistant professor from Bidhan Chandra College, Asansol, and activity of polysaccharide from natural and structural diversity. Please start your PowerPoint presentation and please proceed.
Thank you, ma'am, for your kind introduction. Good afternoon, everyone. The topic of today's presentation, the antiviral activity of sulfated polysaccharides from natural products, a review of extraction, structural diversity, and the structure activity relationship. The outbreak, this single frightening term, refer to some of deadliest pandemic in history. Those are measles from measles virus, Black Death from plagues, smallpox, Spanish flu, and recently SARS, HSV, HIV, and now we are struggling with coronavirus. So list is very long like their history. In history, 2000 years ago, the herpes epidemic struck the Roman kingdom, and the King Tiberius took a decisive action. He banned all kissing. Not surprisingly, the king's solution was not effective, and the human civilization was experiencing a full-fledged epidemic. So far, doctors all over the world have managed this situation by providing a mixed therapy known as HART, highly active antiretroviral therapies. It uses multiple drugs that act on different viral targets. But still, this therapy is facing some challenges. Those are, first, today's active drug may not be active for same patient due to evaluation of drug-resistant mutant virus. And secondly, the poor bioavailability of these drugs. And thirdly, the toxic side effects of this compound is too much. So there is a need of new kind of drugs which have novel mode of action. And recently, unfortunately, many compounds having antiviral potency in cell culture have been detected in recent years. And among them, sulfated polysaccharide from natural origin draw special attention of pharmaceutical chemists. The sulfated polysaccharide from natural origin have been found in many marine alga cells, some human cells, and animal cells, and some in bacterial cells also. So, sulfated polysaccharide can be easily extracted from those natural sources by using different kind of solvents, and reported by Cassidas et al., Feldman et al. In 2000 and 1999, and many other work also reported. But the question arises, are the sulfated polysaccharide source limited in these sources? The answer is no. Sulfated polysaccharide can also be obtained from land plants by two-step process. First, extraction of polysaccharide, then sulfation of that extracted polysaccharide.
Ma'am, sorry to interrupt, ma'am. Can you hear me? Ma'am, your slides are not changing.
Oh, sorry.
Okay. Do something.
So now it is visible? Hello?
Yes, it is clearly visible now. Please continue.
So sulfated polysaccharide can be extracted from natural sources easily by using different kind of solvent. But sulfated polysaccharide can also be obtained from land plant by two-step process. First, extraction of polysaccharide, then sulfation of extracted polysaccharide. And the Saha et al. Reported in 2010 that the polysaccharide extracted from these plant source, and then sulfation occurs using SO3-PY complex. The similar work also reported by Saha et al. 2011, Sinha et al. 2010, and Bandyopadhyay et al. 2010 also. But these process is a very much time consuming and costly process also. So recently, scientists have generated sulfated polysaccharides from the land plant using one-step process, skipping the extraction process. And Rai et al. In 2013 generated sulfated polysaccharide from rice bran, a waste material, by changing the reaction time 10 minutes, 6 hours, and 36 hours to get a different kind of sulfated polysaccharides. Mukherjee et al. Also use this one-step protocol to get sulfated polysaccharide, and they utilized the different natural sources like E. dulcis fruits, Moringa oleifera flowers, and Spirogyra ssp to get sulfated polysaccharide, namely P1801, MOS, and SSS. Now the structural analysis. The sugar composition analysis showed that P1801 contained solely glucose residues, indicative of glucan sulfate. And conversely, the MOS contained galactose, mannose, xylose, glucose, and arabinose as major sugar residues, indicative of acidic heteropolysaccharides. And other polysaccharide SSS contain xylose, arabinose, rhamnose, glucose, and galactose as major sugar residues, also indicative of another kind of acidic heteropolysaccharides. So sugar composition analysis showed that showed the structural diversity of the polysaccharide from various plant source. And now the detection of sulfate group. The FTIR spectra showed the strong band near about 1250 centimeter inverse and 810 centimeter inverse, indicative of the sulfate group. Now, the degree of sulfation, which defined as the number of sulfate groups per monosaccharide residues in the polymer. And P180, the DES of P1801, MOS and SSS have the value of 1.5, 1.4, 0.9. And now the position, the glycosidic linkage position and the location of the sulfate group also determined by Mukherjee et al. Using linkage analysis. And they provide the structures of P1801. This is the structure, and in these like these other sulfate polysaccharide structure also be analyzed by other group of scientists. Now after structure come to function. See that P1801 MOS and SSS showed the strong antiviral activity, and the activity decreases from P1801 to MOS to SSS, which lie parallel with the degree of sulfation. See, with increasing degree of sulfation, activity increases. So degree of sulfation is also important factor for the also important factor for the antiviral activity. And Bandyopadhyay et al. 2011 also reported that sulfated polysaccharide showed strong antiviral efficacy towards HSV1, and here the activity also dependent on sulfate group. See, stronger the sulfate content, higher the activity. And the Saha et al. Reported that the xylogalactofucan F2 showed the activity which increased after further sulfation of this compound. And this is the activity of alginic acid, which contained high amount of carboxylic acid groups, but its activity again increased after sulfation. That implies that nature of anionic group is also important for antiviral activity.
Ma'am, can you please wrap up within 2-3 minutes?
Okay. I'm just summarizing. The Rai et al. Reported that sulfated polysaccharide exert concentration-dependent antiviral activity. See, with increasing concentration, here in all the samples, the activity increases. And here in the experiment, drugs are added in all the cases, and see that the prominent effects are seen on the pre-adsorption and pre-adsorption post stages only, which infers that inhibition occur at viral entry stages. And with this view, this is a commercially available cytotoxicity assay was done by Rai et al. 2013, and the cell were treated with drug, that is sulfated polysaccharide, for 3 days, and then the MTTI yellow strain was reduced by the enzyme of a live cell to a blue color formajan. And see, more the intensity of blue color, more is the number of live cell. And we can see from this that all the sulfated polysaccharides have passed the exam of toxicity. So all the compounds used here are not at all toxic. Now the summary of this review work are Sulfated polysaccharide can be obtained directly by extraction or by chemical modification from different natural products. Sulfated polysaccharide from different natural origin have different novel molecular structures. Sulfated polysaccharide have been shown to exhibit antiviral activity, and the important factor of such compounds antiviral activity are degree of sulfation, molecular mass, and structural diversity. The main mode of antiviral activity is inhibition of viral entry. Sulfated polysaccharide do not show any observable sign of toxicity or side effect. They are water soluble, and experimental evidence indicates sulfated polysaccharide might become increasingly important in drug development for the prevention of sexually transmitted disease in the near future. These are the references of this work, and thank you for your kind attention.
Thank you, ma'am. Do you have any question from audience? Anyone? Any question from audience? Jenny, I thank you, ma'am, for your insightful lecture.
Thank you.
Thank you. And yes, now we are now moving to our final presenter of this from participants, and then we have one keynote lecture, and Dr. Adino Dikari is already is with us. And welcome, sir, from our organizing team. And now our last presenter from our participant, Mr. Ravi Kumar Shah. Are you with us, sir?
Yes.
Can you just put the video or presentation on? So Mr. Ravi Kumar Shah, he is not academician. He is treasurer of an NGO named Ehsas, Feel the Desire of Nature and Society. So we are expecting another wonderful lecture, and he will tell us about the role of environmentalists, NGOs, and government to increase forest and tree cover, affecting reforestation of ecosystem in India. So wonderful. We are eager to hear from you, sir. Please continue.
Thank you. Hi, good afternoon. Is it visible? Thank you. I am Ravi Kumar Saw, Treasurer of Ehsas FDNS. First of all, I would like to thank the organizing committee, UB College, to give me the chance to present my paper. Ehsas Feel the Desire of Nature and Society is an NGO located in Asansol, working for the enhancement of unprivileged society and also awareing and working for the development of natural resources by planting tree, awareing about the sustainable use of natural resources. Coming to my paper, the title of my paper is Role of Environmental NGOs and Governments to Increase Forest and Tree Cover Affecting Restoration of Ecosystem in India. Introduction. During the early part of the British rule, trees were cut without any thought to export and appropriated forest resources for revenue generation. But after some time, the British regulate and conserve. In 1855, Lord Dalhousie framed regulation for conservation of forest in the entire country. Due to rapid population growth, depletion of natural resources, deforestation, increased industrialization and agricultural growth, the environmental balance is disturbed and various aspects of human settlement like quality of air, water, sanitation, health is badly affected. According to the report published by National Geographic, forests cover almost 30% of our land, but they are disappearing in an alarming rate. Deforestation causes climate imbalance and climate change, as because the forests are the lungs of our planet, it increases greenhouse gases emission and causes global warming. Since the industrial revolution, the global annual temperature has increased in total by a little more than 1 degree Celsius. A planet that has never been hotter has recorded 9 out of 10 warmest year from 2005, only since 1880. This indicates that due to human intervention, the greenhouse gases are emitted rapidly and resulting in increasing temperature. Now the climate scientists have concluded that we must limit global warming to 1.5 degrees Celsius by 2040. Increasing human activities have resulted in changing our climate. Climate change has dramatic effects on natural environments, plants and animals. Many environmentalists and NGO come forward to work on environmental issues to increase forest and tree cover, ecosystem restoration, etc. Objective. My objective is to find out the importance of present tree cover in India, to understand the important parameters to restore forest ecosystem, to understand the role of individual organization and government in environment protection, to find the possible solution which human beings can do for nature. This paper is done by the secondary and also by the observation, interview, government data and internet search. My findings. After the analysis of government data, the forest cover in India in 1991 was 19.39%. In 2019, it is 21.67%. If we see the state-wise forest cover percentage of tree cover in northeastern states of India and Uttarakhand, it is more than 40%. Assam, Madhya Pradesh, Chhattisgarh, Himachal Pradesh, Jharkhand, Odisha, Karnataka, Tamil Nadu is moderate tree cover ranging 20% to 40%. Rest percent in agriculture-based states and states with urbanized city like Bihar, UP, Maharashtra, Punjab, Gujarat, Rajasthan, West Bengal. State-wise tree cover. Tree cover are the tree patches excluded from forest. We can see the change in tree cover in many states of India in this map. State-wise percentage of tree cover in the period of 2001 and 2017. You can see the positive growth in Arunachal Pradesh, Chhattisgarh, Delhi, Goa, Gujarat, Himachal Pradesh, Jammu Kashmir, Jharkhand, Madhya Pradesh, Maharashtra, and some northeastern states of India. And the negative growth are seen in the Assam, Bihar, Haryana, Karnataka, Odisha, Punjab, Tamil Nadu, Uttar Pradesh, West Bengal. Discussion. There are many reasons which affect the forest and tree cover across the country that have direct influence on the forest ecosystem. That is settlement. With the increasing population, humans are cutting down trees thoughtlessly across the country. Vast amount of area are captured for building flats and commercial market. Industrialization is a major issue, causes degradation of forest and tree cover in India. Now this day for the growth of country and employment generation, industries are expanded all over the country. Next, agriculture. According to the FAO, agriculture causes around 80% of deforestation. According to the same report, 33% of agriculture cause deforestation is a consequences of subsistence agriculture, such as local peasant agriculture in developing countries like India. Population growth. India is a developing country because of less of awareness and education in past decade. Uncontrolled population growth in India causes increasing settlement and intensive agriculture in India. Demand of Indian timber and forest resources. Forest contributes to the world in terms of timber production and other forest producers. From the British rule in India, trees are cut down for timber and paper production. For extraction of many natural resources, forests are degraded in India. There are some parameters which may control the degradation of forest, and it helps to restore the degraded ecosystem in favorable condition of forest growth. That is afforestation. Sustainable human settlement. Sustainable settlement is one with an ample supply of green space. In the case of town and city, this includes park located inside it, green belt that surrounded it and protect its boundaries. Environment sustainability. Conserving natural resources. It may be conserving natural resources, creating more efficient project and practices, develop alternate sources of power, replanting forest, preserving wetland and protecting natural areas from resource harvest. Awareness on environment protection. Carelessness on natural resources is a major negative point for our green environment. So it is very necessary to aware and giving proper knowledge about the sustainable use of natural resources. And community development by the local people strengthen the activity for afforestation and ecosystem restoration. Community by the proper knowledge may help to make the activity more easy and reliable. There are many individual environmentalists and various are working on environmental protection. We are well known to few names of environmentalists like Sundarlal Bahuguna, propagator of the Chipko movement, Medha Patekar, who has initiated Narmada Bachao Andolan, Jadav Payam, the forestman of India. He has created 550-hectare long man-made forest all by himself. Rajendra Singh, the waterman of India, he run an NGO called Tarun Bharat Sangh, and Uma Shankar Mandal, the mangrove man of Sundarban, who is working on ecosystem restoration by mangrove plantation and community development since 2009. They had set an example of the role of individual in environmental protection.
Can you hear me, sir? Can you just wrap up
Yes,
within
yes,
next?
yes, ma'am. Okay. There are many NGOs like Green Yatra, ISA Foundation, Purvasa Eco Helping Society, Chalo Party Foundation, the Youth Force, Welfare Initiative Foundation, also working for the development of environment. Some government programs initiative working for the development of environment. There are many schemes like National Green Corps, Eco Club, National Action Plan, Climate Change to pursue sustainable development of course that meets economic and environmental goals at the same time. To make people aware of the value of coastal habitats, Swachh Nirmal Tat Abhiyan, Secure Himalaya project to biodiversity, land and forest resources in the high Himalayan ecosystem. Green Chain campaign. Green Chain campaign is launched by the ASAS on the day of International Forest on International Forest Day. Green Chain campaign can reasonably interpreted more than one possible way. One who can plant and influence others to do the same. Our main point of Green Chain campaign influence to plant more trees in open space, awareness on recycle and reuse of plastics, sustainable use of natural resources, awareness on reducing the use of plastic, promoting the use of eco-friendly products. This is some activities by the ASAS, FDNS, some individual other concerned groups and NGOs. Conclusion. Overall discussion clearly shows that increasing forest and tree cover is a positive sign for our environment, but this change is not enough to tackle climate change and different natural phenomena and disaster. Our study focuses on more tree plantation in open and abandoned space. It focuses to work on expanding forest cover all across the country with the help of local individual, NGOs, involvement and government initiatives. This is my reference. Thank you.
Thank you so much, sir, for your presentation. And it's a very good topic to cover, and we all should discuss about this reforestation, and we all should take part in this process. And thank you so much
Thank
for
you.
covering this topic in this conference.
Thank you so
And
much.
now was our last lecture, and this lecture we are also waiting for this Dr. Atin Odhikari. He will be delivering the next talk, and he will be introduced by our esteemed colleague, HOD from our Department of Botany, Dr. Sanjeev Pandey sir. So I'm handing over this session to Dr. Pandey sir. So please take it. Okay. Yes, sir, you are audible, sir.
Thank you, Vinita madam. So it's my privilege to introduce one of my senior colleague in Bose Institute. Once upon a time, I would say, it's 24 years back. It's very happy to get him back. Thanks to Almighty that I rediscovered him a couple of months ago when he was delivering lecture in Durgapur Government College.
Sir, sorry to interrupt you, sir. You are not audible right now. Have you just? Okay. Maybe there is some little technical problem. Just for a few seconds, please wait.
sorry for interruptions. I was saying that it's my great privilege that I got Dr. Atin Adhikari, who is my senior dada, I would say rather. We were working in Bose Institute 24 years back. And suddenly after about 20 years, he was missing. I suddenly found him when he was giving a talk in Durgapur Government College. Thanks to Almighty for that. Dr. Atinda, you were working, you were waiting for quite some time. Sorry for that, because people were taking 2 or 3 minutes more for their allotted times. So I would now introduce you to the platform of eConsus 2021. It's a long list of achievements. Dr. Atin Adhikari has more than 10 years experience in environmental and occupational health research areas at University of Cincinnati College of Medicine and at the Jianping Shu College of Public Health, Georgia University, Georgia Southern University. He was previously involved with a broad range of research projects related to exposure assessment of air pollutants, including airborne particulate matter, microorganisms, and microbial contaminants in indoor and outdoor environments, microbial exposures among community and in agricultural workplaces, relationship between respiratory allergies, microbial contaminants, and air pollutants, and inactivating of microorganisms relevant to biodefense. He is currently working on PPE compliance among workers and other occupational health-related issues. He has supervised 3 important projects as the principal investigator, supported by CDC, National Institute of Occupational Safety and Health, National Institute of Environmental Health Science, and United States Housing and Urban Development, and several DoD projects as the co-investigator, and authored or co-authored 67 peer-reviewed articles of original research. He has reviewed 58 national and international research articles for scientific journals and served as a panel member and reviewer of Ngos disease, disability and injury prevention and control, special emphasis panel, Center for Disease Control and Prevention, National Institute for Occupational Safety and Health, and the Johns Hopkins NIHS Center in Urban Environmental Health Pilot Project Research Grant Program in Johns Hopkins University. So it's a long list of introduction. It will continue further, I think. So I would rather request Dr. Adhikari to share his thoughts to start his lecture. People have been waiting to listen to you.
Thank you very much, Sanjay, for the great introduction. And this is a great privilege for me to with you, all of you. And good afternoon, everyone. And I think this is a multidisciplinary audience, and we have some students, we have faculty members, we have seniors. So definitely, this is a great opportunity for me. Let me first share my slides with you.
Yes, it has come, Otinda.
Okay. Okay, so today the title of my topic is global warming, climate change, and sustainable health. And this research area is actually not my specialty. I am working in many other fields, but I am teaching this topic. So what I will do, I will explore some newly emerging areas which might be exciting for all of you. So we have 2 primary learning goals today. Number 1 is developing an understanding of the roles of different environmental pollutants on global climate change, and how those changes are related to environmental and occupational health effects. And then we will discuss sustainable health promotional efforts and how those are specifically applicable for work-related diseases, and we will also address some environmental hazards. And we have 6 primary learning objectives. Number 1, discuss the short-term and long-term air pollution-induced climate changes in the atmosphere at local, regional, and global level. And why I am starting from the very beginning, because we have some junior students also here. And then we will discuss roles of greenhouse gases and air pollutants on radiative forcing, how that's changing the Earth's surface temperature. Then we will discuss the distributions of these pollutants in the Earth atmosphere at global level. After that, we'll discuss in more detail about the health effects from climate change, and then understand the significance of climate change with respect to occupational health issues. And finally, some sustainable health promotional approaches which are related to adverse impacts of global warming and climate change. So, of course, all of us are familiar with atmospheric effects of air pollutants. There are many different types of effects, with some of those effects are short-term effects, and which might be the visibility or some changes in our urban climatic conditions. And there are some regional effects, and finally the global effects. We are more concerned about global climate change in this lecture, but all other effects are also interrelated. So if we think about the presence of too much dust particles in the air, which can reduce our visibility, and at the same time they may have some kind of promoting effects or reducing effect for some climate change related impacts. So there is a very complex relationship between air quality and climate change. Air quality can be impacted by climate change, and also conversely, climate change can also impact air quality. For example, we know that ozone is a serious issue in the global climate. Ozone level is increasing, and at the ground level, ozone is hazardous for us. And in the upper stratosphere, ozone is again protecting us from some hazardous radiation, solar radiations, and other radioactive radiations. And if we think about particulate matter, we know that that's also another important outcome from the air pollution. But some of those particulate matter, they have warming effects, and some of the others have some cooling effects. One example is black carbon. This is a particulate pollutant from combustion that contributes to the warming of the earth. But if you think about sulfate aerosols, those are also particulate matter, but those are cooling the Earth's atmosphere. And how that's happening, I will explain later in another slide. Now, that's why climate changes can result in impacts to air quality. Atmospheric warming is associated with climate change, and that has potential to increase the ground level ozone in many regions. And the impact of climate change on other air pollutants like particulate matter, those are less certain, but the research is underway. Like if we think about the sulfate aerosols, these are some emerging areas, and we are undergoing new research endeavors. If you think about the United States, in the United States we have some kind of temperature increasing temperature patterns, and particularly the southern states in Texas, in Florida, Arizona. There we are seeing that the temperature is gradually increasing, and by the 2100 we are expecting the temperature will increase about 100 degrees, above 100 degrees Fahrenheit, so close to between 40 to 50 degrees Celsius. So that will be a serious problem in the next few decades. Now, if we think about the impact of climate change on human health, there are many different types of impacts. One is in this figure is adopted from CDC, and this is very interesting. So at the center, we have rising temperatures, we have extreme weather conditions, we have rising sea levels, and we have increasing carbon dioxide levels. These are very well-known effects from climate change. And these are the outcomes on the atmosphere: increasing air pollution, changes in vector ecology, increasing allergens. Water quality is also another issue. If we have excess floods, excess sea level increase, that could be a problem. Water and food supply impacts. If we have higher temperature, can increase the drought conditions. These are already visible in Africa. And also environmental degradations, extreme heat, they have other types of problems. Now, if you think human health problems, those are here outside. So we can expect asthma and cardiovascular disease from air pollution, excess malaria, dengue, encephalitis, hantavirus infection, different kind of viral infections, Lyme disease, chikungunya. These are all related to different kind of disease vectors. So if their ecology is changed, then that will also create problem for us. Now, increasing allergens, increasing pollen grains, increasing molds, that's also already reported. In USA, if you go to the Midwest area in the upper states, then you will see the pollination seasons are changing, and number of pollens per flower, those are also changing. Overall, the pollen burden in the air, in the atmosphere, those are changing, increasing respiratory allergies and asthma. Now, if we have problems with water quality, then we may have cholera, we may have cryptosporidiosis and other bacterial diseases. And water and food conditions could be related to malnutrition, diarrheal disease. And also, if we have extreme heat, then that can cause heat-related illnesses and death. Cardiovascular failure could happen if we have extreme heat-related exposures. Now we have some data here. This is also from data collected from American Public Health Association. Already we are seeing that 42,000 cases of West Nile virus is in the US since 1999. And that's the cause more than 1,000 people, 1,700 people, they have already died from that. And there are many other vector-borne diseases. Those are also increasing, like tick-borne diseases like Lyme disease. Those are also expanding, and Lyme disease has doubled from 1991 to 2013. So these are some climate change related health effects which are already visible in the United States. Now, if we think about individually how climate change can affect our health, number one, this can elderly people and children, they are more vulnerable to heat-related illnesses. And of course, the morbidity and mortality from excessive heat, those are also reported. And at least 37 US states, they saw record high temperature in the summer of 2010. More than 7,000 deaths were already reported. So these are some problems. Now at individual level, we may have dehydration problems, we may have heat stroke, we may have aggravated cardiovascular problems, and aggravated respiratory illnesses. Now I will discuss a little bit about the basic theories and basic concepts of climate change. So these are the primary determinants of global climate change. Number one, how much energy we are receiving from the sun, and how much sun's radiation is reflecting back to the upper atmosphere, which is Earth's albedo, absorption of incoming solar radiation, and absorption of outgoing thermal radiation. And there are many other mechanisms. One is quasi-periodic oceanic temperature changes, increasing sea salt with decreased Arctic Ocean salinity, and variation in solar radiations, orbital variations. They don't have a major role on climate change, but they are also influencing climate change problems. Now, if we think about the overall solar radiation, which is constantly coming to the Earth, that's 13,070 watt per square meter per second. And satellite measurements indicate that solar output varies in the short term, so we have some kind of changes in those radiation patterns. Now, greenhouse effect, all of you are familiar with that. So this is our Earth's surface, and we have layer of greenhouse gases here. And all of you know that if we have solar radiations are coming, and when they are reflecting back, at that time actually they are generating some higher wavelength infrared radiations that can increase the surface temperature more efficiently. And these are the most common greenhouse gases. Primary greenhouse gases are carbon dioxide and water vapor. And also we have other gases like methane, nitrous oxide, tropospheric ozone, CFCs, perfluorocarbons, and sulfur hexachloride. Now in this chart, you can see the concentrations of these greenhouse gases affected by human activities. So most important greenhouse gas is carbon dioxide. And you can see the contribution of this gas to global radiative forcing is 56%. But we cannot ignore methane and halocarbons. They are also contributing. And of course, we have to think about water vapor in the atmosphere. Though that's also another important greenhouse gas. If we think about carbon dioxide, the concentration is increasing. Now, if we get consider the estimate, it's 31% since 1750 and increasing 1.5 ppm per year, and annual increase is 0.4% up to 2007. Now, in recent years, we have some gradual decrease, which is good news. But still, the contribution to global forcing is about 56%. So you can see in this chart that how the carbon dioxide gas emissions are changing. And since 2006, now we have some decreasing trends, which is good news for the our human population. Now, methane is another strong infrared absorber gas, and methane concentration also increased 150% since the 18th century. The annual increases have been 10 to 15 parts per billion volume per year, but we have some variable data. Similarly, we have nitrous oxide, and these are the different sources. Methane, you know that methane is coming from paddy farming. That's one of the most common sources. Nitrous oxide also coming from different sources, and it's increased 16% since 18th century. CFC, now the CFC is controlled. We have some regulatory measures against that, but CFC can stay in the atmosphere for a long time, and its contribution to global forcing is about 13%. Tropospheric ozone, the contribution to forcing is about 8%. Now, I think some of you are already familiar with ozone depletion trends in the stratosphere, which is creating ozone holes, and we have excess UV radiations in polar regions due to that problems. Now, if we have excess UV radiations, that could be related to skin cancers and many other diseases. Now, ozone and UV radiation changes, those are in this chart, you can see the relationships. So if we have higher concentrations of UV, that's related to lower concentrations of ozone. So these are the minus 60, minus 50%. Now, as I said earlier, that water vapors are also one of the important greenhouse gases, but we are most of the time paying more attention to carbon dioxide. Why? Because all these atmospheric gases, they actually absorb not all wavelengths. They absorb some specific wavelengths, and for other gases, those wavelengths could be transparent. So, for example, the absorption patterns of water vapor, those are blue peaks. And for carbon dioxide, here, those are the pink peaks. So you can see in this chart that how those are absorbing in different wavelengths. Now, for carbon dioxide, why it's so important? Because you can see the overlaps here between 12 and 15 micrometers. And this window is actually releasing the temperature to the upper atmosphere. The temperature absorbed by water vapor, that's going to upper atmosphere through that window, and carbon dioxide is blocking that part. That's why we have carbon dioxide is so important greenhouse gas. And if we have higher carbon dioxide concentrations, that's also increasing the global temperature. Now, what is the radiative forcing? I said earlier that carbon dioxide is playing a major contribution, 56% to radiative forcing. That's the balance between absorbed and radiated energy determinants, which determines the average global temperature. Now, if the radiative forcing is positive, that warms the earth, and if it's negative, that cools the earth. So usually the common trends in radiative forcing is increasing trends. That's why we are more concerned. But if we have excess aerosol concentrations, some of the aerosols, they can cause negative forcing because they can reflect sunlight, and that's also in the long run can cause climate cooling. So here you can see the radiative forcing components, and carbon dioxide is here, and also we have other different kind of particulate matter, aerosols. So you can see the when carbon dioxide is playing positive role in the radiative forcing, methane is also playing positive role, nitrous oxide, all of them are positive, but aerosols, they have actually negative role. I already talked about sulfate aerosols, so they can they have some opposite influence. Now this is the Earth's energy budget, and here you can see that this is the incoming solar energy, which is absorbed by the land and oceans. Some of that is reflected by the atmosphere, some is reflected by the clouds, reflected by Earth's surface, and then radiated to space from clouds and atmosphere. So water radiation from the water vapor is very important step at this point. And if we have excess carbon dioxide, then all these processes will be influenced, and we may have higher atmospheric temperature at the ground level. Now, in the aerosols, there are huge amount of dusts also present in our climate, and these are mainly from deserts. Those are solid particles, and depending on the presence of this dust on different surfaces, they may have different kind of effect. For example, if the sand is present on, it may have a cooling effect or it may have warming effect, depending on the presence. So when for example, sand particle, when the sand is present on forests, then it's lighter particle, and then it can actually cause cooling. But when the sand is darker than snow or ice on some surfaces, then it will create some opposite trend. Now in this figure, you can see the overall how the global dust is moving from one place to another. These are mainly from Taklamakan, from Libyan desert, from Sahara. These are actually the dust belts, major dust belts in our atmosphere, and all these dusts are moving from here one place to another. Now, depending on when they are on the top of snow surfaces, then they have different effect, and when they are present on the forest surfaces, then they have different effect, as I said in the last slide. Sulfate aerosols are also very important, and they are produced from sulfur dioxide or dimethyl sulfate, and they are also mostly coming from fossil fuel combustions, and usually their role is cooling. And here you can see how the sulfate aerosols are distributed on the Earth's atmosphere. Black carbon particles, which are a component of soot, those are originating from combustion processes, and they warms the atmospheric layers, but also shades and cools the surface below. So here you can see the black carbons are heating the atmosphere, and sometimes those are cooling. Now there are many other changes in the atmosphere, from clouds and precipitations, quantity of droplets, size of droplets, and their reflectivity. All these factors influence the climate change. Now, if we think about different types of aerosols, natural aerosols act as common condensation nuclei, and polluted air, in the contrast, they contain much higher concentrations of water-soluble particles. And that means pollution-rich clouds tend to have more numerous but smaller droplets. And if we have small droplets, that make polluted clouds brighter. Now, if we have brighter cloud, that leads to scattering of more light and become more reflective. And brighter clouds in turn block sunlight from reaching Earth's surface and shading the planet and producing net cooling effect. So this cloud brightening effect, that is called cloud albedo effect. So you can see that how aerosols are causing cloud albedo effect and reducing the atmospheric temperature. So overall, when the gases are increasing the temperature, aerosols are actually helping to keep the temperature under control. And here you can see overall clouds are how those are the sunshine is reflected back from the cloud surfaces. So these are small droplets, and here you can see large droplets. So if you have small droplets, actually, then we have more reflection of sunlights. Now, what are the evidence of for climate change? We are always talking about climate change, but sometimes we don't have enough evidences. It's already reported that 0.2 degrees Celsius per decade is anticipated. But if we compare all this data with satellite measurements, then we have some discrepancies. So definitely this is because the problem is all these measurements, those are based on some specific sites, and also sometimes we are using some models, and these models may not be accurate when we are considering the measurement for global levels. And these are some common evidences for climate change: retreat of glaciers, decrease in sea ice levels, and also sea level changes increase about 10 to 20 centimeters over the past century. And there are many biological changes which are already visible. Now here you can see how the global surface temperature is changing. Since 1980 to 2000, we have actually higher trends of atmospheric temperature. Although it's very small level from 0 to 0.4 degrees Celsius. And this type of small measurement, how that's applicable in the global atmosphere, is also it's a debatable issue, but most of the people agree that the global surface temperature is increasing. Now, at the same time, we have global increase in water vapor, evaporation, and precipitation, which can increase the floods, and also the coastal areas, they might be inundated, and we may have many other poor water quality related issues. So all of us know that these are the future climate related projections, that we are expecting more recession of glaciers, more increase of the sea levels. Projected increases are 1.5 to 2 millimeters per year. And also we can expect more extreme weather events, higher maximum daily temperatures, more hot days, more intense precipitations, more risks for droughts, increase in tropical cyclones, peak and mean precipitations. Now, if we think about the environmental impacts, our water resources are affected by that. Agriculture is affected if we have more drought. Forestry is affected. Coastal zones and marine systems are affected if our sea level is increasing and we have more floods. And human settlements are changing. In Africa, already those are reported that people are moving from one place to another. Biological communities, including disease vectors, they are also moving from one place to another. And these are the serious environmental impacts. Water resources are affected from changes in rainfall, stream flow, and groundwater recharge, increase in the number of people living in water-stressed countries, and increasing flood frequency and magnitude. Now, for agriculture, the crop growth is reducing because of the increased drought conditions and expansion of arid or semi-arid zones. And another problem is if we have more carbon dioxide, sometimes we have stimulation of crop and forest growth. Now, environmental impacts include also sea level rise, and including increasing storm frequency and severity, increased flooding, accelerated erosion, loss of wetlands, and saltwater intrusion, and these are already visible in coastal zones. Now, human settlements, as I said, those are already changing. Now, if we think about biological communities, poleward movements of temperate species and species changes due to increased arid and semi-arid areas, changes in phenology like bird migration or plant flowering, those are already reported. Initiation of migration, nesting and breeding times of birds, those are also related to climate change. So to summarize, these are the key weather and climate drivers for human health. Increasingly frequent, intense and longer lasting extreme heat, which worsens drought, wildfire and air pollution risks. Increasingly frequent extreme precipitations, intense storms and changes in precipitation patterns. Rising sea levels that will intensify coastal flooding and storm surge. Some existing health threats might be, we already discussed about those health threats like allergic diseases, cardiovascular diseases, and heat strokes, dehydrations. These are some problems. But not everyone is equally at risk. The children and elderly people, they have higher risk levels. And also the people, those who are working in outdoors, for example, construction workers, agricultural workers, they are also at higher risk. Now, important considerations also include age, economic resources, and the location of the problem. If we have climate change in the floodplains or coastal zones and urban areas, then we may have different levels of effects. In this chart, these are very nicely explained. So you can see the problems of human health are at the center. Climate change is here. And you can see stratospheric ozone depletion, changes in global elemental cycles. These are all interconnected. And UV radiations are also increasing. And all of you know that if we have excess UV radiations, that can be related to cancers and other skin diseases. Now we may have some depletion of our fisheries, freshwater changes and depletion if the water quantity and safety is changed, and declines in phenotypic genetic materials and diverse different types of agricultural services, occupational services, that also can change. So all these problems are interconnected and they are, in the long run, they are related to human health. Now in this chart, you can see that what can we do against all these problems. Either we can mitigate those or we can adapt with those. Now in this conference, we are talking about sustainability. So for sustainability, we may have different kind of sustainable approaches by which we can fight against climate change related issues. Now, at the individual level, as I said, that these are the major health-related problems: asthma, respiratory allergies, airway diseases. And if we think about the mitigation and adaptation, then we need new research, and that those research areas should address the relationship between climate change and the composition of air pollutant mixtures. For example, how altered pollen counts or and other effects of climate change, they jointly affect the severity of asthma. And we can produce different types of models to identify the populations who are particularly at risk. So these are, as I said, the problems with children is different, problems with elderly people are different, and these are already shown in different places in the US. For example, in New York areas, it's already found that the climate change is projected towards sending asthma and allergies. So these are the different counties in New York, which are coastal counties, and you can see that they have higher levels of asthma compared to others. And at the same time, they have higher levels of increasing temperatures in those areas. And ozone concentrations are also very high in those areas. So global warming is also making the spring allergies higher in many areas. And at the individual level, what we can do? We can definitely, these are some kind of recommendations from doctors that you can take antihistamine tablets at night, or you can consider nasal washes, which are a natural alternative. So these are some kind of natural alternatives which can be used for against spring allergies. Spring allergy is not a major problem here in India, but in the US, these are a serious issue. Starting from March, the overall pollen load in the air is very, very high, and thousands of people suffer from allergies and allergic asthma. Now, already we are seeing longer allergenic pollen seasons, and you can see in different US states, we have different types of changes. So these are the red means we have changes between above 25 days, and these light blue color, these are no changes at all, and light yellow color 5 to 9.9. So these are at different places we have different types of allergenic pollen seasons. And ragweed, you know, that's a very common allergenic pollen type. Now in agriculture, we have problems. We have excess floods and increased heat stress. So these are the different results from different US states. Now, if we cannot control the climate change, then we are expecting by 2100, we may have the temperature change will increase more than 15 degrees Fahrenheit in different areas. Actually, in most of the US states. So heat stress, cardiovascular disease, and stroke, these are all related. Climate change may exacerbate existing cardiovascular disease by increasing heat stress, increasing the body burden of airborne particulates, changing the distribution of zoonotic vectors. That's why we need new research that addresses the cardiovascular effects of higher temperatures, heat waves, extreme weather, and changes in water air quality on the health. And this new information should be applied to development of health risk assessment models, early warning systems, health communication strategies targeting vulnerable populations. And also these risks are resulting from climate change, which could be offset by reductions in air pollution levels. Heat-related morbidity and mortalities are also reported, and we need different kind of effective risk communication strategies. We need to identify the vulnerable populations, and then prevention strategies, and of course we can apply different kind of sustainable health and sustainability models here. Vector-borne diseases like Lyme diseases, dengue fever, West Nile virus diseases, these are also well-known problems from climate change, and these are increasing day by day. And we need new research to enhance the existing pathogen vector control infrastructure for that. So you can see how the climate change is affecting pests. Number one, when we are applying, we know that we are using a lot of insecticides, and many of them have different effects, but the insecticides are not working because due to increasing resistance to insecticides. So if we have these problems, then of course we have the disease vector levels can be increased. So the insects may have additional generations. Some insects can grow bigger in warmer temperatures, and finally they can actually play more important role in spreading the diseases and also attacking on crops or the people. If we have floods, then we can expect more diarrheal and waterborne diseases. And these are some common examples like salmonellosis, campylobacteriosis, and of course there are many other parasitic diseases, viral diseases, and bacterial diseases which are also related to poor water quality. Now, if we think about different infectious diseases, these are in this chart we have that, and these are the different kind of diseases starting from schistosomiasis, malaria, cholera, dengue, and hantaviral disease, Rift Valley fevers. Many of them are viral diseases and bacterial diseases, and those are occurring in parallel to environmental changes. So the pathway of effect is also changing, and depending on the different pathways, we may have either increasing or decreasing patterns of infectious diseases. So in most cases, those are actually increasing the diseases, but in some cases, for example, urbanization, urban crowding, and that's reducing the cholera because we have improved sanitation, improved hygiene. Now, if we think about COVID-19, climate change is also related to COVID-19. Many people are working on that. One study from the US, they already reported that small increases in particulate matter was associated with about 8% increase in the COVID-19 death. Cancers could be increased from UV radiations. Now, climate change could affect those are all general effects. Now we'll discuss about specific effects on workers. Now you know that agricultural workers, construction workers, and here in many other occupations, people are spending a lot of time outdoors. So they are more exposed to solar radiations and also heat strokes and high heat stresses. So in this chart, you can see how these are the different hazards and different kind of exposures, and these are the occupational health effects from that. We may have heat stress, we have more fatigue, increased chemical tolerance, we may have more respiratory disease, more cardiovascular disease, and if we have UV radiations, then skin cancer, eye effects, and immune dysfunction could be resulted from that. Now we have extreme weather that can cause mental stress, more traumatic injuries, vector-borne diseases. Those are also a problem in allergies, asthma, infectious diseases. These are all interconnected here. Now, if we have industrial transitions from one job to another, sometimes we don't know much about that, how the climate change will affect there, but more musculoskeletal disorders, cardiovascular disease, mental stress, these are all problems among the workers. So we have some evidences which are already available for climate change and related occupational health hazards. So, for example, ozone and particulate matter with longer, warmer seasons, those are already reported. And they may have asthma and other respiratory disease may have affected by increasing ozone levels and particulate matter concentrations. Now, eye effects, skin cancer is affected related to ambient UV exposures. Now, finally, we have to think about some social and behavioral issues, how the climate change is creating problems for that. Now, you know that health disparity is a serious problem now. It's not that all people on our earth is equally receiving health benefits. So mostly the people of low socioeconomic status and some people of some specific races, like in the US, black people or Hispanic people, they have many problems, and they may have different levels of climate change related health problems. Now, that's why we are expecting more social determinants which are related to climate change related health, and there are some disproportionate distributions of those determinants. And some vulnerable populations, we need to identify them, and we need to explore that what kind of behavioral changes or social changes we can do against those. So here we have some suggestions from other researchers. So you can see these are the preparations for climate change. And number one is the interconnectedness of humans and the natural environment. We can increase that. More time spent in the natural environment has shown to improve the mental stress, increase the resilience power, self-esteem, and reduce the depressions. So if we have these problems, then we may consider the interaction with natural environment. Now we need some new ideas and new frameworks from the sustainability field which can be applicable here. We need some paradigmatic shifts in health promotion, thinking and acting in the context of climate change. And of course, we need new partners for that. We need to develop initiatives to address climate change by shifting beliefs and attitudes, because many people, particularly if you go to rural areas, those who are working mostly in the outside outdoors, they sometimes do not believe about the climate change. So we need to promote target-oriented communications. We can target agricultural workers or construction workers. We can advocate for organizational and community policies. And of course, we have to promote the behavior change, particularly if we want to fight against heat stress and other kind of heat-related problems. And finally, we need to ensure access to mental health for those who are experiencing more psychosocial consequences of natural disasters, particularly if we have flood, if we have storms which are coming from the climate change, then we need some interventions for mental health interventions for those people. So to conclude, ozone depletion is the primary contributing factor for increasing UV levels on Earth's surface. There are many greenhouse gases which are creating problem for us, but carbon dioxide is most important, and aerosols may have some negative radiative forcing effect, which can reduce the climatic temperature. Now, the fraction of total forcing due to carbon dioxide is increasing, and we are expecting about 75% increase in the next several decades, leading to increase in global temperature, sea level rise, increase in storm frequency and severity, increase in flooding. Those are all anticipated from global warming. Increased risk for asthma, allergy, heat stresses, vector-borne diseases, and diarrheal diseases. These are all anticipated. And effects of climate change on workers' health and safety are inevitable in agriculture and construction-related occupations, and we need to specifically focus on those workers. Several social and behavioral issues are related to climate change, and we need specific health promotionals and sustainable approaches to address all these challenges. So because we don't have enough time, I had to run very fast. Now I welcome your questions. If you have any questions, I can answer those.
Thank you so much, Dr. Adhikari, for your beautiful, informative lecture. It was almost all inclusive about climate change. You talked about carbon dioxide emission. You talked about ozone layer. You talked about the effect of aerosols on our health. You talked about different diseases and the relation of these diseases with climate change, and how we can tackle those diseases also by having our behavioral changes. You also talked about aeroallergens, pollen allergens. You were an expert of that, I know that. Yeah, from very beginning, from both institutes. You also talked about different microbial diseases, which currently we are facing one of them, which is most devastating experience for everyone, and the learning also. From your lecture, I think the youngsters will can think about different problems to work on. You have given indications at several places. I saw that there are areas or questions where you can pursue on those areas so that the world can be made a better place to live. Thank you so much, Otinda, and I wish that we will be having you in such platforms in a more frequent way. Yeah. Now I will invite questions, of course, as you said.
Thank you very much.
Has any questions? You can ask questions. It's an opportunity for you. You can ask questions. The Adhikari will answer that.
Maybe you can share my email address with them in the future if anyone is interested if you have any questions or you want to do some collaborative research works, I am always very open to that.
Dr. Adhikari, I would rather ask you to write in the chat box. I don't remember right now. In the chat box, if you write the email, your email ID.
Okay, sure.
If you are not able to ask right now, you may talk to him over this email. You can send your question or any other problem or queries, whatever you want to get answers through to this email ID. He will be happy to answer that. Thank you, sir, and or rather I would say, for being with us. And I will be in connection to you later on. And if you are busy, you can leave this platform according to your convenience, or if you are free, you can. Now we will be moving to valedictory session. So it depends upon your time and space, how much. Yeah, you'll get.
I will stay for some time and then I will leave. But thank you very much, Sanjeev, for inviting me. I am really very glad to be with all of you, and definitely this is a great opportunity for me.
No, it's actually our opportunity.
I will be happy to come back again with you.
Like you. Thank you so much. Thank you.
Yeah.
I will give the mic to Vinita Madam to continue with the valedictory session.
So, am I audible, sir? Continue, sir. How is the screen? Yeah. Is it okay? Remove the. Welcome again. And with this end of this fourth technical session, we have arrived at the final session of this conference. On the beginning of this valedictory address, I, Dr. Benita Datta, the Secretary of eConsus 2021, on behalf of the entire organizing committee of this conference, I would like to thank all the eminent speakers from India and abroad for their gracious presence in this event and enriching us with such wonderful academic sessions during this 2-day long international conference through this virtual platform and making it a success. Thank you all. I'm sure that you all had 2 days of ample, productive, and insightful academic sessions. Today, Dr. Shubir Dasgupta, Professor and Head of Department of Zoology of Maulana Azad College, has conveyed us about various bioactive compounds extracted from skins of different organisms and their effects on physiological systems of interest. In the next lecture, Dr. Gargi Bandyopadhyay, research scientist from Department of Biology, Yeshiva University in New York, has beautifully explained the critical subject of cancer biology and its relation to the stemness of the biological system. The next invited speaker of this conference today, Dr. Saurabh Sheel, the assistant professor from School of Art, Ocean and Climate Sciences of IIT Bhubaneswar, in his insightful lecture has described the different kinds of ocean resources and how to utilize them to attain the sustainability in true sense. In our fourth session of this conference, we had distinguished researcher and academician, Professor Shobboshachi Bhattacharya from Agricultural and Ecological Research Unit of Indian Statistical Institute as the first keynote speaker of this last session. And he shared with us his insightful research on population ecology and its cooperative nature as a fundamental aspect of population dynamics. The next speaker, renowned researcher, and our valued invited speaker, Dr. Pritha Bhattacharya. She is the Assistant Professor and Head of Department of Environmental Science and Epigenomics Lab of University of Calcutta. She has kindly covered a much fundamental topic of this time, the environmental sustainability and its linkage to health. Her informative lecture was included the discussion on the main three pillars of sustainability, namely the social, the environmental, and the economical factors that is related to the humankind. And lastly, our session, the last lecture of this session was by our keynote speaker, Dr. Atin Adhikari, and he is an associate professor and Department of Biostatistics of GMT College of Public Health of Georgia Southern University, USA. And in his insightful lecture, he addressed one of the important global issues right now, the global warming, the climate change, and the sustainable health. He discussed about the critical role of aerosols and albedo effect, the realization of the seriousness of the current situation of global warming, and the incoming danger we must feel and we must act upon it for the mankind, that the global warming and this climate change, this includes the health effect on mankind like diseases, allergen pollens, and the airborne diseases, the heat stress or the cardiovascular diseases that may come as a result of this climate change. And he has hinted at how we can just cope up with the situation. And in the end of this valedictory session, last of all, I would like to add one thing, that we must have to be concerned about the fact that environment and development are for the people, not people for environment and development. Thanking all the valued speakers and participants for participating in this virtual e-conference, for making it a grand success. Now I please may request Dr. Sudipto Das, the convener of this conference, and Assistant Professor of Department of Mathematics, and our IQSC coordinator of this college, to propose the vote of thanks. Dr. Das, please take over.
Thank you all, and good afternoon to everybody. Rather a chilly one, as I gathered that all of the participants are from the Northern Hemisphere. On behalf of Banwarlal Bhalotia College and as a member of the organizing committee for eConsus 2021, a two-day international e-conference on revisiting strategies for sustainable development, I convey our humble and heartfelt thanks to all of you for your active participation, which made this conference such a wonderful and cherishable experience. Firstly, I would like to thank all the keynote and invited speakers who kindly accepted our invitations to deliver such illuminating lectures and share their thoughts and opinions on the various aspects of the prevailing situation of development and sustainability, about the problems, the concerns, the imminent dangers, as well as the plausible solutions to overcome them. We were fortunate enough to hear from such eminent personalities: Dr. Kaushik Roy from University of Utah, USA, Mr. Kumar Rana from Institute of Public Health, Kalyani and Asian Development Research Institute, Patna, Professor Apurva Ratan Ghosh from University of Burdwan, India, Professor Diwakar Tiwari from Mizoram University, India, Dr. Vijay Kumar Bharma from ISRO, Bengaluru, India, Dr. Arindam Gupta from University of Burdwan, India, Mr. Shagnik K. Bhaduri from University of Louisville, USA, Dr. Mollika Dastidar from Jawaharlal Nehru University, India, Professor Shubhit Dasgupta from Maulana Azad College, Kolkata, India, Dr. Gargi Bandyopadhyay from Yeshiva University, New York, USA, Dr. Saurabh Shil from IIT Bhubaneswar, India, Professor Shobbeshati Bhattacharya from Indian Statistical Institute, Kolkata, India, Dr. Pritha Bhattacharya from University of Calcutta, India, and Dr. Atin Adhikari from Georgia Southern University, USA. Thank you, sirs and madams. We were deeply enriched by your discussions and really looking forward to hear from you in near future. We also convey our deep gratitude to all the presenters who deliberated on such diverse topics, through which I am certain that we, all the participants, got quality exposure to enhance our knowledge. And of course, it is our great honor to thank all the participants of this conference. Over 700 people have registered for this conference so far, and your active participation has made this event to be such a success. We sincerely hope that we will receive more such responses and active participations and collaborations from your side for our ventures in near future. It is the right opportunity to convey our gratitude to Dr. Amitabh Basu, Principal BB College, Asansol, and Chairperson for this conference, who always encouraged us and provided the support whenever was needed. Thank you, sir. It would not be possible for us to engage into such venture without your continuous guidance. I am also thankful to all the respected colleagues and the students of our college for their support and enthusiasm, which gave us further motivations. And now the time has come for a huge round of applause to all the members of the organizing committee whose relentless efforts have made this event fruitful. I convey my heartfelt thanks to Mr. Rajesh Das and Dr. Subharti Sarkar, who are the joint conveners for this conference, Dr. Vinita Datta and Mr. Kajal Maji, the joint organizing secretaries for this conference, Dr. Arnab Ganguly, Dr. Animesh Mandal, and Mr. Kuntal Bakuli, who were the members of the technical committee, Dr. Rajrupa Ghosh, Dr. Sangita Lahiri, Ms. Jadkua Jamal, Dr. Sandeep Chatterjee, Dr. Devashish Mandal, Dr. Manas Pal, who acted as anchors and moderators during the conference, Dr. Sanjeev Pandey, Dr. Malay Kumar Ghodui, Dr. Amalendu Samanto, Mr. Santosh Kumar Bhagat, and Mr. Mohammad Arif Akhtar, who looked after other technical aspects of the conference. Thank you all, each and everybody. We are also thankful to all the non-teaching staff members of our college, especially Mr. Tribeni Ruidash, Mr. Rajiv Prasad Gupta, Mr. Soumya Chatterjee, Mr. Srikanto Laik, Mr. Kanai Ruidash, Mr. Julfikar Ali, Mr. Sohel Ahmed, Mr. Bir Bahadur, Mr. Anup Jadab, and the others, as they have ensured the smooth functioning of the conference through their diligence and hard work. My sincere apologies for all whose names I may have missed in this list. As we are well aware of the evolution of human civilization and the so-called development and their catastrophic effects, most prominently manifested in the global climate change, it is high time for us to discuss about the sustainability of our future generation alongside the global trend of further developments. It seems that we have to radically redefine the idea of development to achieve any kind of sustainability. In the words of Dr. Debal Dev, the famous seed warrior of India, we have to initiate and practice developmentality, a prescription of development which can also be sustainable. As the ongoing global pandemic has exposed our vulnerability, and as human interactions with nature and natural habitats are rapidly deteriorating, it seems that the doomsday of the sixth mass extinction is looming ominously. Our only hope of survival lies in unified efforts from all of us on various fronts. It's time for action now. We have to dissolve all the barriers and restore humanity to fight for the global cause. As we often forget, and here I am borrowing the word from Carl Sagan, the legendary American astronomer, we often forget that Earth is just a speck of dust suspended in sunbeam, just a lonely pale blue dot inside a vast enveloping cosmic darkness. And most importantly, we often forget that it is the only home we have ever known. Thus, it becomes our duty to protect our beloved home at any cost. We are at a peculiar juncture of human evolution in the sense that we know now way too much about this imminent global catastrophe. Now, the only question remains, will the future generation remember us as the saviors or as the destroyers? The choice is ours. And with these words, I will be signing off and would request Mr. Rajesh Das, Joint Convener of eConsus 2021, to take over from here for some important announcements and to officially conclude the conference.
Am I audible? Yes, you're audible.
Yes, sir.
I mean, visible, right? Yes. Thank you, Sudipto. Dr. Sudipto Das, Coordinator IQAC, Banwarilal Bhalutia College, Asansol, for your unwavering support for the conference as like the previous year. Along with my joint convener, Dr. Shubharti Sarkar, I would like to thank all the participants, without which the beautiful people, this conference wouldn't have been possible. And I thank to our colleagues for their support as well. As a final note, please keep in mind that the full paper submission deadline is February 20, 2022, and we will convey the same to you over mail. Thank you so much, all of you. Good night and namaskar.